conference participants. Welcome to the 4th International Conference on Trias Advances in Research and Progress, Patient-Derived Tumoroids and Humanized Mice, in the beautiful city of Hyderabad. I am Nikita Naik, Organizing Secretary for this conference along with Ms. Ramya. We are gathered here to exchange ideas and open discussions on patient-derived tumoroids and humanized mice. And it is indeed a golden opportunity for each one of us to enrich and enlighten ourselves. Without further ado, let's dive into our promising schedule. I cordially invite our esteemed chief guest, Professor Yaj Meduri, Vice Chancellor of Mahindra University, Vice Chancellor of the stage. Mahindra University. I would also like to invite Professor Rajendra Singh School of Life Sciences, Dean of School of Life Sciences, Dr. Radenna Pallu, Executive President, Federation of Asian Biotech, Founder of Oncosic Bio and Akasta Health. Now, I would like to invite the esteemed guests on the dais, and Ms. Ramya and Dr. Lily to lighten the lamp. Thank you.
वक्रतुंड महाकाय सूर्यकोटि समप्रभा वक्रतुण्डमहाकाय सूर्यकोटिसमप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा।
The lightning of lamp symbolizes the dispelling of darkness and the victory of knowledge over ignorance. Now, I would like to invite Dr. Manusmriti Singh to introduce our esteemed guests.
Good morning, everyone. Welcome to Mahindra University. I hope you've had a quick breakfast, all of you, and we look forward to the day. It's our pleasure and honor to be hosting the 4th International Conference on 3 hours advances in research and progress. The title theme for this year's conference is Patient-Derived Tumoroids and Humanized Mice. This is an annual event organized by Dr. Suresh Pusala and their team, and we are very honored to be hosting it at Mahindra University. Again, it's my pleasure to be saying few words about our Vice Chancellor, sir, although he told that he would demote me if I say too much. So, our VC, sir, Professor Yej Meduri, he did his BTech honors degree from IIT Kharagpur and completed his master's and PhD in engineering mechanics at University of Wisconsin-Madison, USA. He is known to have set up premier institutes in India in past couple of decades. To name a few, he was the founding Vice Chancellor of VIT University, Vellore, and the ex-Vice Chancellor of Bennett University, Greater Noida, and JP University of Information Technology. He firmly believes that education should be an immersive and transformative experience and champions personalized learning, experiential education, and real-world industry collaborations to ensure that our students are ready to tackle the challenges of the modern workforce. He's very approachable, and we are really happy to be guided under his leadership. So thank you, sir, for being here. I would request you to give the inaugural remarks for the event.
Morning, dignitaries in the dais. Ladies and gentlemen, welcome to Mahindra University. As my colleague Manu pointed out, I do hope that you'll have a whole day of very interesting discussions, because yesterday evening I was talking to some of the big scientists around here. You know, in education, one knows about the 3 R's, but in life sciences also to look at 3 R's is quite fascinating. We were also debating about what is humanized mice yesterday evening, you know, that's why. But these are questions, I think, which you all guys are very much into, not like us novices who stay away from deep research in life sciences and tumoroids, etc. But suffice to say that we are delighted to be hosting this event. This is, I think, the first of the ones which Professor Chauhan and his team are planning. Life sciences is a big focus area for us at Mahindra University, and we do hope that we can ramp up and build up on it over the next couple of years. We already have around a couple of hundred undergraduate students, around 15, 20 PhD scholars, and around 24 faculty, and but we'd like to grow this up, you know. I think our facilities are starting to show up. And with that, with the research projects flowing in, the publications getting done, and the research work getting done, I do hope that CLS and MU get identified on the map of India. But delighted to see you all of here, and I do hope that you have a great time. Welcome, welcome once again. Thank you very much.
Thank you very much, sir. And now I would say a few words about the Dean of Life Sciences. So Professor Rajendra Singh Chauhan is a prolific researcher with 6 patents granted by the Government of India, 23 PhD awarded under his guidance, 110 national and international publications, and over 25 crores of grants from different Government of India granting agencies, including DBT, DST, DRDO, ICMR, amongst others. He is also recipient of prestigious national awards such as Jawaharlal Nehru Academic Award of the Indian Council of Agricultural Research, Government of India, and Pran Vohra Award of the Indian Science Congress Association for his significant research and contribution to the areas of agricultural biotechnology. And it's my pleasure to be working under his guidance, and we are thriving, I would say, because we just started last 2 years, and we are really ramping up. We have really good research labs. I would also request the participants, company members to visit our labs, assisted by our lab technicians and PhD scholars. We'll be very happy to show you around. And I would request Chauhan sir to please grace us with his remarks.
Thanks, Manu. Very good morning to all of you, and welcome to Mahindra University. I extend my heartiest welcome to the distinguished panelists, speakers, and the delegates. This is our first international conference, and I'm especially thankful to Dr. Suresh, who considered Mahindra University as the host for doing this international conference with us. So thank you very much, Suresh. Well, as Vice Chancellor Sir pointed out, that Life Sciences we started in 2022, so we are 2 months behind completing 2 years. And within these 2 years, we have done significantly well. Right now, we are running 2 undergrad programs. So I'll be briefly, describing like what all we do in Life Sciences. So we do a BTech Biotechnology, 4-year degree program, and then BTech Computational Biology, that's again 4 years program. And we have close to 150 students in both the programs. We have masters, which we are running with Dr. Reddy's lab. So that we started last year, and 30 of their employees, they are doing masters in biotechnology with a focus on biologics, because Dr. Reddy is pushing towards the biologics area as well. And we're planning to run another master's program, that's biomedical data science, that we are starting in the fall of 2024. We have a very strong research culture at Mahindra University, and that's probably exceptional to a private university. We are currently having 20 PhD scholars in life sciences and close to 150 across the university. And all the PhD scholars are funded 30,000 scholarship per month, along with the boarding and lodging with the air-conditioned rooms. So that's something which is excellent for the PhD scholars. We have around 20 faculty. They are all in life sciences. They're all trained in the finest institutions across the world in different areas, whether it is medical biotechnology, agriculture biotechnology, industrial biotech, and computational biology. And with the support of the Vice Chancellor, we have this seed grant funding because these young faculty, they are very desperate and passionate for starting the research. So almost every PhD scholar, every faculty has been funded around 10 to 15 lakhs seed grant funding. So in 1 year, we have got around 1.5 CR from the Mahindra University. So that's the grant which is provided to the faculty. Our undergrads are also very talented. 2 of our undergrad teams, they have recently won the first round of DBT BIRAC EU Fellowship. So one team has already made presentations in TNU Coimbatore, and another one is going to do it next week in Agriculture University Dharwad in Bangalore. So we have a deep talent, whether it's faculty, PhD scholars, and the undergrad students, and we generate new knowledge. That's what we have started now through research. But to translate that knowledge, new knowledge, into wealth or the money so-so, we need the industrial support, because industry is full of the avenues, the problem statements. So we look forward for industry collaborations, and that's one of the advantages being in a private university, that we do not really have to undergo through any bureaucratic hurdles whenever we set up any collaboration with the industry. So I welcome the all the industry professionals who are here to please join us in taking it forward, as we have with Dr. Suresh. That's the beginning. And I'm sure that in the years to come, it will go a long way, because that's the need of the hour, to really take the advantage of industry academia collaboration, which is missing in the country as a whole. So with this, I once again welcome you all. And as Manu pointed out, that if you have time, please do visit our labs, undergrad labs. Those are really world-class, done. And our research lab is going to come in the next few months. So with this, I once again wish good luck for the conference. I'll be here throughout the day. And once again, welcome you all here. Thank you very much.
Thank you, Dr. Chauhan, for such a wonderful welcome. Now, I would like to invite Dr. Sridhar Vallatti to introduce Dr. Suresh Pasala, founder of Oncosic Bio and Akasta Health.
Thank you. I think this is my first occasion to introduce Suresh formally, even though I know him for more than 6 years. Everybody knows him, but I thought let me give an introduction for the benefit of various audience that are available here. So Suresh is a trained veterinarian from Tirupati, and then he made his career to go to US and did his PhD from Iowa State University. And then later on, he went and worked as a postdoc at St. Jude's Children's Research Hospital. I think that's when he started applying his science to healthcare, seriously. And then he was also did a residency at University of Maryland in Washington. And then he became the chief of comparative medicine at NIH, which is a very prestigious position. And then he made his entry into industry. He worked with BMS, Bristol Myers Squibb, which is a famous pharmaceutical company. And he also was in the senior leadership team at the BBRC, which is very well known in India. He has various accolades, which I don't want to go more in detail in the interest of time, but he has publications in Nature and he has got patents. And he's also an advisor and member at AAA LAC Council and a vice president of LASA. That gives him the enough credentials for the global recognition. And I think he decided in 2018 that he wants to become an entrepreneur. So I was happy to host him at University of Hyderabad's Bionest as a founder of Akasta, first Oncosic Bio, which you are seeing now, and then later immediately started another company called Akasta. And he's a consultant to VCR Park in Vizag. It's called Vast Cancer Research Institute, and one of the members from there will be going to talk to you guys very soon. And he's also a member and an advisor and a member in the Reagent Biosciences. And with this, I want to stop there, but he has got other active roles in BIRAC, DBT, he's also an angel investor. With a short introduction, thank you very much for this opportunity.
Thank you. Thanks, one and all. First of all, warm wishes, greetings, and thanks to the Vice Chancellor and the Dean, who I consider as good friends now. And we do want to host this event annually, international event at Mahindra. The facilities here are state-of-the-art. Innovation is at the peak here. And I would like to have industry academic interactions here, as well as very famous people like Uday and Ajit, who are all here. And my colleague from VCR, Mandar, is also here. So we'll have deep discussions on this going forward. And starting a company and actually feeding many, many lives, including the animals that we talk about, in a very humane manner is what my goal is. In doing so, we came with the 3 R's, which is reduce, refine, and replace animals. That's where the alternatives come in. So we respect the animals for the research. We use them, utilize them. And the FDA is asking us to do that, but along with that, the alternatives. So there are a lot of good companies that are coming up in India and US that are looking at alternatives and advanced animal models. This conference is the 4th international conference. We have 90 registrations to be here today in the auditorium. We have 150, so this campus is a little bit far, so they'll take time to come, and I have at least 45 people on their way right now, so sending me messages. And we have 150 registrants online who are signed up for live cast events. So they are in attendance from 10 o'clock on live. And we are also broadcasting this on YouTube for the next 30 days, 1 month. And for that, we have close to 900 registrations. So we don't end it right here. So the word about we are doing it in Mahindra, we are doing this conference in India for the 4th time is going to be there. And it's well publicized in LinkedIn. And so the idea is to promote alternatives, 3 R's, science, and then bring good places like Mahindra to the forefront of the Indian ecosystem of advanced research. So thanks for your innovative discussions yesterday and 2 months back. And my good friend Manu, who introduced me to Mahindra, and we pulled her from Delhi along with others. So we look forward to a lot of great scientific interactions, sir. And with that, I welcome you all to this conference. Looking forward to a great day of learning on alternatives and advanced animal models. And we have one esteemed guest in Lily, and also Krishna, my good friend. Both are from the industry. They both represent two major mouse distribution, breeding, innovation, transgenics, humanization. So they are into the forefront of that. So Jackson obviously is the pioneer in this one. And we have several other companies like that. So looking forward to Krishna's talk, which is going to be spellbinding, I'm sure. And also Lily, who is coming from Janvia, but she has been in India for so long and started NCBS and so on. So I think the mouse programs she started, she's a very valuable trainer. So we like to have all these interactions going forward for long. So I didn't want to miss any name here, but all the speakers are going to be here. And Uday is going to conduct an exciting panel, which usually Uday makes it much more like T20 instead of 5-day test match. So it's going to be really exciting, and he'll put people on the spot and create small fights. So we're looking forward to that, Uday. Thank you. And you have a nice day, and lunch, snacks, everything available. And for hosting us here today, thank you once again, sir, and Mahindra University. I want to give a souvenir.
Now I would like Dr. Suresh to give souvenirs to the esteemed guests. Thank you. It is my honor and privilege to stand here and welcome all of you to this exceptional event where innovation meets inspiration. Without further ado, I would like to briefly introduce and talk about the future of cancer research using tumoroids and humanized mice. So last few decades have seen many different types of cancer model, including patient-derived cancer models. Preclinical models being introduced currently focus on enhancing translational efficacy from preclinical setting to a clinical setting with integration of 3R principle. Such cancer models include organoid cancer model, cell line cancer modeling, Drosophila melanogaster cancer modeling, and patient-derived xenografts. So today I'll be talking about tumoroids and humanized mice. Tumoroids are patient-derived cancer organoids which mimic tumor microenvironment in a controlled settings. And in a human body, so tumor microenvironment consists of different types of cancer and non-cancer cells depending on type of cancer, and different types of immune cells as well, as you can see in this schematic of lung tumor microenvironment. In a human body, when a tumor is treated with a therapy, the results that you see is a cumulative effect of the interactions between the tumor, the treatment, and the components of tumor microenvironment. So tumor microenvironment, some of the components have to assist in tumor progression, while others have anti-tumoral effect. In this representative example, the lung tumoroid very well reproduced the histological feature of the primary tumor, an adenocarcinoma, which was confirmed using various stain. For example, positive staining of thyroid transcription factor 1 and negative staining of P40, confirm it is an adenocarcinoma and differentiate it from squamous cell carcinoma. In addition, positive staining of thyroid transcription factor 1 and Ki67 also confirms the proliferative character of tumoral cells. So these tumoroids can then be used to generate patient-derived organoid xenograft, PDOX. The cells extracted from the biopsies of PDOX can then further be used in the formation of organoids. So the cells from PDOX can improve the cancer cell selection by removing fibroblast and normal cells without the loss of heterogeneity. That's why you have this double organoid. And so tumoroid research, there are several tumoroid organoid cancer research applications, such as evaluating immunotherapies, assessing primary and metastatic diseases, exploring genetic disease drivers, tracking tumoral cells, identifying predictive biomarkers, informing the in vitro to in vivo transition, and investigating drug responses, thus revolutionizing the drug discovery workflow. Just like tumoroid mimic tumor microenvironment, humanized mice have genetically engineered functional human immune system, thus mimicking the human context. There are various models to develop humanized mice and to mimic the human immune microenvironment. We will hear a great deal about it from some of our speakers. There are various PDX models for various cancers in the works and currently in the market, such as prostate cancer, stomach cancer, intestine cancer, breast cancer, and pancreatic cancer. Despite numerous advantages to using tumoroids and humanized mice over traditional cancer models, there are several challenges and bottlenecks that need to be addressed. There are some immunological limitations and require further development and optimization in humanized mice model. Other challenges include high cost of establishing the models, lack of optimization, standardization, and quality control measures, given the field is relatively new. However, the synergetic application of these technologies offer a holistic view of cancer progression, a better understanding of drug development and personalized treatments, and optimizes the predictive value of preclinical studies, thus accelerating the identification and validation of therapeutic strategies. In conclusion, I would like to reiterate that the integrated use of 3R principles, tumoroids, and humanized mice represents a transformative approach in cancer research, promising improved translational relevance and personalized treatment insights. Now, I would like to invite our first speaker, who needs no introduction. They have left an indelible mark on the field of humanized mice. Dr. Krishna Bhagavatalu serves as the Director of Business Development for the Jackson Laboratory in India, Southeast Asia, and the Pacific. With over 2 decades of experience in life sciences and healthcare, Dr. Krishna held senior roles in Agilent Technology, Teconic Biosciences, Intertrek, and Global Value Web Technologies. He possesses expertise in various life science domains, including food testing, molecular diagnostics, in vivo models and services, data analytics, and personalized medicine. Dr. Krishna holds an MBA in General Management from the University of Adelaide, a PhD in Clinical Bioanalytical Chemistry with specialization in molecular medicine from Cleveland State University, and a master's in microbiology from Osmania University, India. He's a member of the Royal Society of Biology and holds membership with ANZLA, FABA, and SALAS. Today, Dr. Krishna will give us an insight into JAC's model and services in advanced therapeutic areas. Please give a warm welcome to Dr. Krishna Bhagavatulu. Yes.
world is moving to. So before I get into the details, I also want to share with you a little bit about today what is an insight or what we do at JAX and what JAX models are, and also the services in advanced therapeutic areas. So the Jackson laboratories humanize energy mice models that can help you also characterize and also do a lot on the validate your immuno-oncology therapeutics. So I'll give you a little bit insight into how we work and what we do and what Jackson Laboratory is all about. As an organization, what is our mission and how we work with worldwide researchers. So our mission mainly is to work with researchers and also to discover precise genomic solutions for disease and empower the global biomedical community. So when I say empower the global biomedical community, it is so relevant in today's world that all the researchers around the world are working with the quest of trying to discover something, make a difference, and how they can really create an impact in trying to provide better solutions for better life and better health. So Jackson Laboratory is an independent, non-profit biomedical research organization that is almost a 100-year-old organization that leverages a unique combination also of how we work on and working with empowering and also creating the scientific excellence through innovation, through research, through education and discovery. Those are four important strong pillars of the organization, which I also heard our speaker from Mahindra University speaking about how the university is moving towards creating and establishing research facilities. Similarly, what we also do at Jackson Laboratories is trying to create our own internal grant funding research facility who programs groundbreaking discovery research, and additionally we also offer mouse models for preclinical studies and education to support the worldwide biomedical research community. And also, JAX has a global experience, and that can be seen here in this slide, where you can also see how we are spread across the world and how we have an unparalleled experience as your partner, whether it be in drug discovery, drug development, drug evaluation, drug efficacy, so on and so forth. As a suite of personalized offerings, including novel cutting-edge services for early preclinical drug discovery, we have provided, like I was talking about the preclinical research models and services as well. So our models are almost shipped to around 64 countries, I think, across the world. Where you can see this is represented with the light blue regions across. I think it's more than that basically. And then also we have in India ADLT as our authorized partner. And then you can also see our locations with the blue dots here. In the US, we are having our own facilities in Bar Harbor, which is in the main on the up right. And then we have a lab in Connecticut, and then we also have in Sacramento. And we are also spreading across globally. And now the next part of the presentation that I'm going to be discussing with you is about the miles-associated mouse services that are very significant and very important in the world that we are working on with. JAX mice are the industry standard for animal research. They also focus on using precise genomic solutions, mainly to better understand human diseases. And that's where the 3R also comes into the picture. So we also very focused upon 3Rs, and therefore we also provide continuous research and development on understanding what could be the precise models that are really applicable for doing your ongoing research work that is so critically important and relevant. So our rigorous animal health programs and also stringent genetic quality standards make sure that the data is reproduced and it's validated for all of your experimental work that you're doing. And that I think is very, very important in the way we do. And that is why JAX mice is also known to be the gold standard and also the best characterized mice that is widely used. More than 80% of all of the mouse-related research publications cite strain sourced from JAX. So we also have over 13,000 strains that are cryopreserved. That's again another contribution for 3Rs because we don't really try to have mice that is just not relevant, but we also see that there's a future and there's a requirement, so we cryopreserve them. In that way, whenever there are any researchers who want to really work on those, we help them to cryo-recover and we provide those. So in that way, we have over 13,000 strains. Let me walk you through a little bit about how we do also custom model generation, which is also another important area of interest for researchers on using the specialized models, how they can really work on their models. So with JAX, what we also do is try to do a genetic modifications and with variety of technologies, be it DNA microinjection or be it lethal embryonic cell manipulations and microinjections, or even using some of the latest technologies such as CRISPR and also PROS. So the process you can see here, how a custom model is generated, where you can see it starts with the project plan and then goes on to generation of and validation of the models, and then also the ESL modification and screening, and then goes on to the different stages, and then comes to the stage of validation of N1 animals. So this is a very interesting project that takes almost around 30 to 40 weeks for you to focusly getting on how you want to get the model of your choice of interest. So while working with JAX, we provide you also regular updates along the way, including the founder package, and also the mice are guaranteed to be a germline competent and genetically validated. That is something which I think is also important because what we are seeing globally is a lot of researchers are moving towards specialized models. And they want to custom create, they want to really design, they want to use a model of their interest. And that is where I think particular organizations that the JAX comes to really big help. Along with that, we also have a lot of husbandry support where our experts manage simple and complex projects on time and within budget by maintaining colonies that are important, by providing breeding services, aging services, cryopreservation, cryo-recovery, cryogenic services, expansion services, and genome scanning. Similarly, what JAX also does is a lot of work with veterinarians. So tomorrow, I think when we see a lot of interest coming across in terms of how the trends and research is developing, and Suresh coming from a veterinarian background understands this more of how it is important to do a lot of surgical procedures with expertise and experience. So that is where JAX works with researchers on providing those kind of services. And also JAX, what they do is a lot of work on education. So they travel across the world, they work with research universities, and they work with veterinarians by providing a lot of surgical procedures, surgical services on how expert surgeries are done on these in vivo models. do a lot of preconditioning services where there's a lot of custom preconditioning solutions, diet-induced models, and FPS models. So what I think I want to also spend a little bit time on sharing with you is about the journey of how a drug development is done, and also to give you a little bit of insight onto what cycle goes by and what approach an organization take in doing this process, which I think also would be very interesting for researchers here, where a lot of areas of interest in advanced therapeutics are these days, and what we see is, as we all can imagine, is more oncology related, especially cancer or immunology related, where there's a lot of immunogenic responses that are also important to understand and to study. And at the same time, there's also neurological impact that creates also an alterations in our day-to-day life. So therefore, there's a lot of research happening in these areas, including the metabolic and genetic disorders. So we have a preclinical and clinical data and observations, and that leads into the genetic modifications and data interpretations, and where we then create a specialized models that are of important and interest, be it PDX, humanized mice, and then also genetically modified mice, or even for that matter, induced models that are really also of interest. There is a lot of new and cutting-edge capabilities and expertise with mouse models of these human diseases. The major advantage of doing the study, is also how we can try to look into de-risking and also to designing your drug development work, especially like I was talking about in the areas. You can also see here the array of also key activities that goes into this process, which play an important. And also I think we have a lot of experienced expertise professors, for example, researchers. Our neuro division is run by Dr. Stacy Risso, who also has been working with researchers like you around the world, and mainly to do the services and also to models to develop. And also we have done a lot of work with organizations such as Roche or even Pfizer, where they also have been using this level of model design expertise and experiences from JAX. Now I'm going to move a little bit into also talking about what are the different therapeutic areas and in vivo services that are going to be also important, and also I think are going to be relevant for today's presentations that we're talking about. When we look into neuro, I was talking about, so JAX also does a lot of work on, regardless where you are in your discovery phase, there is a lot of work that goes into, especially in providing you really specialized neurological models that are relevant to study a wide array of preclinical research models and also understand about neurological disorders. We do see a lot of work happening, which I think would be of help for students here and for researchers here also, is also to design and develop their focus in the coming areas, especially in the area of gene therapy, which is gaining a lot of significant interest to create the genes or to understand and explore the genes that can be also manipulated and also help in providing the therapeutic solutions. That is where a lot of interest is coming in gene therapy, where those therapeutic genes are modified and studied on mice models to see how they can really impact the human models or human immune systems. So we also do a lot of work in small molecules, nucleic acids, protein replacement therapies, antisense therapies, and also pathway modifications. Some of the areas how we do specialized models and studies that are also going to be helpful for you when you're doing your experimental design is to look into the general behavior and then also a fine motor behavior, electrophysiology, and motor. These are different aspects of how we try to really focus, be it in the cognitive areas or sensory areas or anxiety-like behavior, social behavior, and psychosis. So these are some parameters that we take into when we are working on these models. That's something also researchers here can look into when you're working on to your neurological models and applications. And this is to give you an insight into how the various techniques and also various age groups of mice are working on, be it from neonatal to adult, on the different doses that we use to do different timelines to study the neurological disorders. And also here you can see, to give you a little bit of insight into how the data is evaluated and is studied. This is also about the amyotrophic lateral sclerosis studies that was performed. NFL is a serum biomarker of neurological injury and degeneration that can be measured from blood. or CSF and tracked longitudinally in the mice here. This marker is increased in patients with ALS and other neurodegenerative disorders that can predict the prognosis of patients with ALS and Alzheimer's disease. So this is just to give you an insight into how the studies are conducted. Similarly, also there's a lot of translational large molecular pharmacokinetics. This is also becoming of interest, which I think also would help some of the researchers in their journey on discovery. FcRn studies, which are offered from JAX, comprises two models, FcRn PG32 and 276 mice, that mimic specific aspects of the human IgG physiology. It provides the ideal solutions to assess the half-life of different antibodies and also of antibody variants in early phases of drug development. Furthermore, these models actually also provide a most translatable studies for you on how you can do your therapeutic antibodies and understanding, and also in timely and cost-effective manner. That is very important. We are also working a lot with other institutions in the FcRn models, especially with some of the top institutions like Dana-Farber is working on these models as well. And also to give you an insight on the pharmacokinetics, how we do this through the standard steady arms with vehicle and test article, using precise therapeutic injections through IV or intraperitoneal route. And then also there are multiple blood collection time points that are collected, and also plasma is used for the processing, or serum that is used for processing, especially again to do your clinical observations, which also are important, like I was talking earlier about, using whether it is body weight or also conditioning score, body conditioning scores. And then you do a lot of antibody analysis through ELISAs, and then you move on to PK analysis, whether it is pharmacokinetics, including half-life, Cmax, volume of distribution, and clearances. This is just to give you a glimpse of what is being done on that. And moving on to the immuno-oncology or immunology space, there is also a lot of work that is happening of interest, where humanized, of what we were talking, especially that goes with the theme of the conference, on using humanized models. Now, what exactly is the humanized model and how it is developed and designed? Basically, it's also something where you take the human cells and that are engrafted onto the mice, and then there are different models that are there onto, especially with 334 and PBM, which are also of our interest. And then there's a lot of preclinical models that are studied, whether it is oncology, whether it's infectious diseases, whether it is autoimmune and inflammatory diseases. So there are a lot of things that goes on to this, whether it is graft versus host disease, or applications onto lupus studies, which is gaining a significant interest, multiple sclerosis, type 1 diabetes, and psoriasis. This is just to give you a little bit of insight. And then how we do a lot of work onto assessing the biomarkers or histopathology, necropsy, or clinical observations. The data here gives you an insight into how the lupus efficacy studies are conducted. You can see here, this is an experiment that is done to do a proteinuria analysis, and how this increases by 13 weeks of age treatment, basically with the cyclophosphamide treatment, and then also beginning at the 8 weeks that inhibits the increase in proteinuria in MRL LPR mice. This gives you a spontaneous mouse model insight which recapitulate clinical features of human SLE and are used extensively in preclinical efficacy studies. Similarly with the oncology, by combining the oncology and immuno-oncology studies, what we are also seeing is the work that is happening on the PDX tumor, be it to do a cell graft, cell line engraftment, xenograft, immuno-oncology, toxicology and efficacy, and also thereby going on to proof of concept. Recently, when I was there traveling and meeting some of our researchers in Australia, what made me also of great interest in having a conversation is how the researchers are really moving from basic work to translational work. They are doing a lot of work on PDX models and PDX or synthetic models, but then they have challenges using those models, and they can't really replicate what they're doing here. The next stage, what they are looking at is humanized models and how they can really create a platform to mimic the human immune system. And that is where I think humanized mice really come into the picture of providing you a data that is really relevant for you to understand how it can be helping you in designing your molecules of interest or therapeutic molecules of interest, or taking you your research to the next level. So how we do is an experimental process here designed for you to evaluate some of the parameters that we do in the assessment. In the study that we are showing you here gives you also how we have studied a specific bispecific T cell engager and a monoclonal antibody on tumor growth. On the left is the mean tumor volume that you can see here, and the right is the body weight, to give you an insight onto how the molecules are really engaging the monoclonal antibodies and how the tumor growth is evaluated and assessed in the oncology model. Like I was talking to you about the humanized mice, which I think is also very, very important in the way we are moving on to 3 Rs, where there is also a very important specialized advanced models that are gaining a lot of interest globally, which is also where I think India is playing a very, very big role, is on trying to create models that are important. Now, when I say create models that are important, and why are these important? As we were talking about the oncology, the relevance of oncological cases that we see is significantly increasing, be it in the breast cancer, be it in the myeloma, be it in the glioblastoma, so a lot of other areas. Now, for studying that on human systems and trying to get a human responses is simply practically impossible. And that is where the humanized mice come into picture, where the process, like I was talking earlier, has created a groundbreaking solutions and the platforms with thereby mainly to do a human cell engraftment, to evaluate the compounds and to treat a variety of human diseases. From oncology, what this is also moving is into other areas, be it infectious disease, microbiome, to study the gut flora, and also how that is really having therapeutic significance.
I'm almost done. But yeah.
Okay. Yes, so this. Actually, this is my last slide, so yeah, but still. So yeah, so we do have a lot of work on happening, like I was talking about, to treat advanced diseases. So one of the interests, apart from cancer, is also the way it is moving towards other diseases, infectious diseases, metabolic disorders, microbiome, and all, where I think the humanized mice are also becoming something as a cost-effective models to stimulate trials or evaluate multiple drugs alone or in combination, and where you produce also reliable predictive data. When I say reliable predictive data, you know why that is important. It plays a significant role because when you want to take your molecule of your interest or whether you want to do a steady analysis, what is important is the reproducibility. At the same time also where you can take the data and what you can really use the data for. And when you want to use the data for, you need data that can create also an idea for you to understand what you can do with that, whether you want to publish it, whether you want to evaluate your molecules of your interest, be it small molecules, be it large molecules, whether you want to take that studies to a clinical trial level or you want to take it into other platforms. For all of those, as we know, the difference between mice genome and human genome, for students here, how much is that? Can anyone answer? What is the similarities between mouse genome and human genome? I should have actually asked you this first in the beginning, but maybe for the research students who are here, for the benefit of them, just want to understand if anyone has an idea. What is the percentage of similarity between the mice genome and human genome? Sorry? 70-20%. No, it's actually 95%. So it's 95% similarity between human and mice genome. It's only the 5% difference that makes it mice and that makes us human. So the 5% makes a big difference, right? So that is where I think that is also one of the reasons why a lot of researchers like to use in vivo models and they start with mice. So, but then when you're moving on to the levels of advanced models, like I was talking about, we have a wide array of humanized models now. In even humanized itself, you have different models, be it CD34 or PBMC. The difference between these two, CD34, it is a robust in vivo platform
mainly to analyze the safety and effectiveness of potential new drugs to modulate the immune system. So these are advantages because they also have a long-term studies, especially in the way the cell lines are maintained, the T cell maturations, and also the T cell dependent inflammatory responses. So that's what makes it also a very interesting model for researchers to use it, whether you want to study human cell biology, immuno-oncology, or even infectious diseases. And if you look at the other ones, and then you also have a PBMC model, which is a humanized PBMC mice. It's a quick engraftment of adult peripheral blood mononuclear cells, and then also they enable short-term studies requiring mature human T cells. So that is where researchers also use these models. And then they're also used to study and evaluate compounds for T cell modulation, which plays a very important role in the immune response. So there is a very interesting talk that is scheduled follow-up towards the end of today's sessions, where my colleague, Dr. Charu Gupta from the US, she will be presenting on how to guide for selecting xenograft and mouse models for preclinical cancer research. So stay tuned till the end of the day, I think the last session, where Dr. Charu will be presenting more about that. And it was great giving my talk and presentation, and especially interacting with here. I really like Dr. Suresh's work, what he's doing in creating an impact in the scientific community, and we are honored to be here at Mahindra University as well. If there are any personalized discussions or questions that you have, I'll be here all day to discuss and talk to you one-on-one, and then also to have some kind of understandings on how the research work can be taken forward. With that, I want to thank you all for your time. It's great coming over here and being a part of this event. Thank you. Sure. Yeah. Sure.
So my question is, you talked a lot about oncology models. Do you have any DDH models for in vivo models for AML, CML, TNBC?
AML and then TML.
That's one set, AML, CML.
Yes.
And then TNBC as well.
We do have, yes, absolutely. I will share with the details with you, and I can also discuss with you, so that you can also look into those models that can be of your interest. But are you looking into?
Fee for service, or is just a license we have to get a license?
I'm sorry.
Is this a fee for service model, or we have to, like traditionally sometimes we have to do a licensing deal?
I don't think this involves licensing.
Okay.
You can readily take them available. Sure.
All right, thank you.
Yeah, welcome. Sure.
So this is Srinivas from Accubasis. I have another question which is related to pharmacokinetic and pharmacodynamic correlations with PDX models to the clinical data. Do you have any experience? So the purpose of using patient-derived xenograft is to counteract the heterogeneity that is coming in the phase 2.
Yes.
That's the reason, so we are going to more humanized in patients.
Yes.
So do you have any data which can explain the correlation between the preclinical to clinical, where the success rate is more than 80%?
Yeah, so I don't know if we have the data that is comparing between preclinical to clinical one, but at the preclinical level, we have a lot of researchers at JAX who also does a lot of work with our other researchers or clients like you on providing those kind of studies. I'm sure we have a lot of work on that, and some data is also available, and some data is confidential, which was not published. But yes, so there is a lot of work happening, and that is where, like you rightly mentioned, also people are moving towards humanized ones. Because I had a similar interaction also with another researcher at other university, and they are also working on that platform, and they wanted to also move on to humanized to kind of also overcome the challenges in terms of the pharmacokinetics data, what they're evaluating and comparing it with clinical outcome.
So that's continuation to my question. So basically, when you are comparing the pharmacokinetic and pharmacodynamics, you take the tumor growth and then we'll see any kind of pharmacokinetic parameters.
Yes.
But when you go for clinical studies, we need to understand the biomarkers which are similar across the different kinds of patients.
Yeah.
Especially when there is heterogeneity.
Yes.
So, I mean, I think you might have done some standard compounds where we are seeing certain kind of.
We have. I can share that with you.
Okay.
Absolutely.
Yeah,
Okay.
I'm coming that 2022 to Nobel laureate, Dr. Paulus, who made the missing link between human and where whom we have come. And the last part of the things what we are doing, it is surprising to know all disease coming from the lethal drug sequence and integrated in the human. Let's say for HIV or COVID, even ibuprofen doesn't work to all, even the miscarriage and all those things. So I'm telling when you are doing this humanized mice, are you looking into those things which could be like some to lead to a great things to that?
So if I understand you correctly, I'm sorry I couldn't hear clearly, but I think what I understand is that you're asking me if, are you having any specialized humanized models to study the precise drug effects on individual, like some drugs works on some and some doesn't work on others. So do you have any models to evaluate that? Was that the overall gist of your question, if I may ask you? Disease specific? Inherited.
This is the sequence when you're doing this humanized things. Promoter will be the same.
Yes.
Are you putting those sequence or something there?
That's a very interesting question, actually. I've never heard so far anyone talking about.
Just because you follow this 2022 Nobel Prize.
Yes.
This has been done very nicely.
Yeah, absolutely.
Why this, our Indian and Pakistan thing, 50% those sequence there which is prone to COVID, but not to HIV?
Yeah, definitely. Definitely, because that is where I think the personalized genomics comes into picture, where these days what is also happening is they are trying to create the solutions or the personalized genomic solutions where certain drugs or put a gene map to individual and see how he responds to certain diseases and metabolic disorders. And what is happening is, in my system, trying to design and develop that is practically not feasible, but that is happening also, I think, in some parts of the world. But at present with JAX, I think what we are doing is more focused on therapeutic disorders like metabolic disorders, inflammatory disorders, oncology disorders. But that is more of a personalized solutions what you are guiding towards. But I think that will be also very interesting for researchers who wants to understand that in specialized human study races, basically, be it any kind of Caucasians, Indians, or African Americans, and those kind of diverse, wide background, ethnic backgrounds, and how they respond to certain diseases. And that is where I think a lot of genetic research is needed. Definitely that would be very, very helpful for researchers to carry it forward onto these models, applying them as a base for that and develop those solutions. But that will be very, very interesting, actually.
Thank you.
Thank you.
Thank you for all your questions. Thank you, Dr. Krishna, for your insightful presentation.
Thank you. Thanks to all of you.
We are truly grateful for sharing your invaluable insights. I would like to invite Dr. Mandar Kulkarni to present a token of appreciation to Dr. Krishna.
Hi everyone. So there's a half an hour tea coffee break. So you're not supposed to bring the tea or coffee inside. Please have it outside, and then you can resume the session, which would be soon. Lily would be taking over. So there's a 2 or 5 minutes break to bring it or have it, and then please come back here. Thank you. After the break.
We'll start at 10:30.
Yeah, 10:30, 10:30, 10:31 we'll resume.
Check, check, check. Check.
Check this. 1, 2, 3, check.
Check, 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 check.
మూడు వస్తున్నాయా?
Check, check, check.
Check testing. 1, 2. Test. 1, 2. Check.
Check. Check, check.
సరే రెండు
check.
మైక్స్ మాట చెక్ చేసాను.
Check, check, test.
Check testing.
Check, test.
1, 2, 3, 4.
Check, test. 1, 2, 3.
Check.
Care Resource Center established at the National Center for Biological Sciences in STEM campus in 2014 in India. Dr. Lily has been instrumental in organizing and conducting high-end international hands-on workshops and training programs in mouse genome engineering, assisted reproductive technologies, mouse embryo handling, and developmental engineering development for Indian students and scientists. With over 20 years of experience in laboratory animal sciences and mouse model engineering, Dr. Lily currently serves as Head of International Operations for Zenvier Labs, a key player in international biomedical research, providing access to quality, well-defined animal models and associated preclinical services. Please give a warm welcome to Dr. Lily as she will talk about Zenvier Labs, a new partner for your preclinical research studies.
Yeah, can you hear me now? Okay, great. Yes. So, good morning, end of morning, I guess, to everybody. And I would like to take this opportunity to really thank Suresh, Nikita, and Manu and the whole team for inviting me, and obviously for Mahindra University to host this. It's a great pleasure for me to be here. And like Nikita said, I've been in India for 10 years. I say this because it's not already obvious sometimes. So I know the Indian animal science community quite well. I've been involved in different training programs, setting up facilities over the years here. And I'm really glad that I got this opportunity with Zenvier Labs, which is a French company. So I'm back to my French roots because I am French originally. But expanding their activities here in India, and I really hope to interact with many of you in the very near future in this regard. So I'll present a little bit who we are and what we do and how we can really support your preclinical study research. So Zenvier Labs is, like I said, a French company. It's a family-owned business still to this day. And it started in 1960 by Roger Zenvier, and this is why you have the RJ nomenclature on our animals. It's in the middle of France, and he was just working in his agriculture environment over there, and stumbled upon this article that said that there was a really lack of professional laboratory animal providers in France and in Europe compared to what was done in the US and elsewhere. And so he took this opportunity to get trained and to start really establish high-quality laboratory animals for the French community and later on for the entire Europe. And now it's the grandson, Thomas Zenvier, who is now the CEO of the company and running the company in Laval. This is the Zenvier Labs site. It's a unique site in Europe. It's the biggest breeding site in Europe, located here in the west of France. And you can see here the facilities here, over 230,000 square feet of animal facilities, 3 million rodents. So we do only rodents. And providing customers worldwide. So most of our customers to date are in Europe, but we do provide quite significantly amount of animals and models throughout the world, and more recently in India as well via our partners Samitech. And I am here to hopefully increase those partnerships, and really happy to interact with the community again under a different hat, but with the same enthusiasm, and really appreciative of all the work that's done in laboratory sciences in India. So to give you a glimpse, because we are not yet well known here in India, but we are very well known in Europe and the rest of the world. We have major clients, including Indian clients, and so we have data that we can share from these clients in case you you need a little bit more convincing in the quality and the purpose of our animals. So in terms of services, like I said, obviously we have, like many of the international breeders, a wide variety of laboratory animals, mice, rats, hamsters, and jailbirds. I'm not going to go into details of all of what we have. We have the website and the ranges there. All the classical strains are there. More specific strains also for human diseases are also there. What I'll focus on today is on the topic of this conference, is mostly on our different immunodeficient animals and the related humanized animals that we are provided designing for your preclinical research. We have a service, obviously, to create new genome engineer models, to create the model upon request. I have to say that all of our models in our services are license-free, so I have to insist on this because that's not the case of some other providers. So when you purchase an animal or service from us, it's not linked to any IP or licensing issues. We also provide, and this is a big service that has increased a lot over the years, is custom and contract breeding services. So many, because our facilities are really high standards and providing high quality animal breeding management. And so when you start working with these a little bit more high-end animals, you need facilities that can manage these. Not everybody has that in their facilities, so we can breed and on demand the animals, either if they come from our repository or from an external source as well. And obviously other services to basic surgeries and related samples from the animals that we can provide as well. So in terms of quality, this is something obviously that we are very, very attached to and take great care of. In terms of genetic quality, we want to make sure that, like many of our colleague breeders, the animals that you obtain from us have a really well-defined genetic identification, and that genome is, that background is stable over time and over space. And so we have a very rigorous program for all of our stocks at Janvier Labs, where we have very well-defined cryopreserved nucleus stocks, embryos usually, that we recover on a regular basis after 10 generations. That's roughly every 5 years. And we do regular genetic monitoring testing to ensure that our stocks do not drift over time or that they don't get genetically contaminated with other animals that, animals can jump from cage to cage sometimes, hopefully not, but by doing all these tests, we make sure that what we provide you down the line here is the same genetic quality that in the beginning. Our quality control does not only stop at the genetic quality, we also do an extensive immunophenotype, and this is really relevant to not only our immunodeficient animals, but all our stock. We have a very strong immunophenotyping team at Janvier Labs. And this is very important because by doing this, we actually have noticed that depending on the genetic background, the different substrains, you can have different immunophenotype profiles. The percentage of different immune cell types are different depending on the stocks that you're using. And that can be really relevant before you start for some of your studies to know exactly what the immunophenotyping profile of your animals are. All our animals are maintained under really strict SPF, even SOPF for the immunodeficient animals in our core breeding standards. So we exclude all the pathogens that may interfere with the animal's health, with the human health, and obviously with the research down the line. The uniqueness of our site, the fact that we have a unique breeding site and that we routinely come back to the original cryopreserved embryos via embryo transfer makes us quite unique in the sense that we have a very controlled and defined microbiota over the space and our time. So not only does the genetic background does not change, but our microbiota is extremely stable over time because of the way we maintain our animals and because of the fact that we are coming from one unique site. Of course, we have scientific support and a very unique transport system throughout Europe, to have an end-to-end service providing of our animals. So now I'll dive in more precisely in the topic of today and present to you first a few of our immunodeficient animals and how we've used these animals to do humanization of these substrains and how these humanized mice are generated and the importance of them for preclinical assays and downstream translational work. So this is not an exhaustive list, but the immunodeficient models has grown over the years from the basic nudes to the really more complicated strains now. So the most model being used now for grafting most of the time is the NXG, which is our version of the NSG. It's the same, it's called differently because JAX has a proprietary on NSG, but the mutations are identical, and I'll show you some data on these models to show that they actually work really well. And we have also some really even more efficient models now that have been not only mutated to be immunodeficient, but they have been also humanized transgenetically to have some of the human HLA factors expressed, for example, which increased even more the possibility and the efficiency of human cell engraftment and make them even, the possibility to use them even longer for the experiments. So just rapidly on our NXG, so it's the PRC IL-2 gamma-2 mutation as well, and these are readily available, and these are the gold standards for basic humanization, and now I'll talk a little bit later in the talk on even more advanced models that are derived from this one that are also used. Like I say, these are license-free, so they're ready on the shelf, and there's no licensing issues when you buy them from Janvier Labs. So just a proof of concept, just quickly to show you how these work. So what you see here on the left panel is NXG animals that have been grafted with some AML leukemia cells, liquid tumor cells, and you see that over time the animals do really well get the tumor growth really well. And what the clients have done here is that they've tested one of their chemotherapy reagents, and by doing so, the animals that have been treated but grafted, the tumor does not grow. So this is just a proof of concept that these NXG work well for these purposes. There's a lot of paper. I can provide them on demand, and you can find them online as well. These have been used extensively with our partners for CAR T cell analysis and development as well. So now let's go and dive in directly in the heart of the topic of today's seminar, humanized immune systems. So to answer one of the questions that was asked earlier, when we talk about humanized mice, we have to be careful and be precise because they are transgenic humanized mice, meaning that genetically we have added the human gene in the animal or replaced the mouse gene with the human gene. Here, this is a little bit different. What we are doing here, we are removing the immune system of the animal, so to speak, and we are replacing it with the human immune system, okay? And there are mice actually where we can do both, so we can have combinations of both. So these HIS model, which are humanized immune system mouse models, so we start with animals that are immunodeficient, meaning that they will be able to engraft the human cells, donor cells. And we have different flavors of these. I'm not going to go too much in detail right now on this, but a little bit later. But the importance here is that when you're using these humanized immune system models, you're bringing human relevance to earlier stages of your drug development process. And this is why these have been developed initially. So these make them much more translational than the non-humanized version of the simple NXG, for example. And so you can really eliminate trial and error on the different components, the drugs, or the tumor therapeutic agents that you're testing on these humanized immune system, humanized system. So how do we do this? So like Krishna said previously, there's different versions of this. I'll take here the more stable one, which is humanizing via engrafting human CD34 cells from the cord blood. So we take our NXG immunodeficient animals in this case, and the first thing that we have to do is to remove the immune system of the animal. And so we do this by two different methods, depending on the model that we're using. Either we gamma irradiate sublethally the animal, so that kills all the remaining cells of the mouse, or we do the immunoablation via chemicals, which actually works very well with busulfan. You can't really read here. That is injected IP. And this for the NXG actually works very well, and it's actually much more robust than gamma irradiation as well. So depending on the models that you're using, you can use either gamma irradiation or busulfan treatment. And at the end, so now you have an animal that has almost depleted of its own immune system, but you need to provide it the donor system, which is the human one. And so we have banks of human CD34 cells obtained from the cord bloods of the human, and these are injected immediately after ablation, and it takes a few weeks, 14 weeks, for the full humanization of the immune system to be stabilized and efficient. And so we usually do, in our case, a quality control after 15 weeks to make sure that we have a minimum amount of humanization in our animals, so that we just don't give you animals that have failed the humanization process. So that's very important. This is why you have to wait a little bit, 14, 15 weeks. Although if you have we have clients that want them immediately after the humanization process, we also can provide those, but they will not be quality controlled, right? So that's different models. But these are actually very stable. You will see in the next slide. Usually if you do get the animals that have been quality controlled, meaning that we guarantee that they have at least 25% of their H45 cells that are human, but usually it's much higher. And they're stable for another 15 weeks to up to 30 to 35 weeks after humanization. So you have a long time to do your experiment, which is the difference between humanizing with the CD34 cells or the PBMCs, right? The PBMCs, you can humanize much earlier, but you have a much shorter time to do your experiment because you have this host versus disease reaction that will kick in, and the animals, you will lose the animals after a few weeks, right? So depending on what you're doing, either you opt for the CD34, which is much more stable, but it takes a bit more long time, or the PBMC if you want a quick analysis, but it's a very short time, the analysis that you can do. So like I said, we always do the quality control before and after of the different immune cell populations by FACS cell sorting. And this is an example to show how stable it is. So this is 14 weeks after engraftment. So we have this peak and then it stabilizes, and it's stable for almost 30 to even, we have clients who have reported that they've used the animals without any problem after 35 weeks, okay? So you have a long time to use the animals. This is important because we have clients who say, yeah, but you ship up the animals at 16, 17 weeks. They are shipped here. We ship them to Korea, by the way. That's the furthest his animals go, but they work very well. We have very happy clients in Korea using these animals also. But yeah, so they arrive. We have to wait 1 week for the animals to recover. So basically, we're not starting an experiment after week 19 or 20 after engraftment. Is this going to work, yes or no? The answer is yes, because you have another 10 to 15 weeks to do your experiment. So there's no problem with that. In the case of H44 humanization, obviously PBMC is much shorter. Okay. So another version of these immunodeficient mice, like I said, have been engineered even more to also express the HLA factors, the human HLA factors. And so these can be humanized as well and have been used mostly for vaccine assays and therapeutics. Sorry. But yeah, that's another variant of it. The procedure is the same, it's just that the animal background is a little bit different. So yeah, on the shelf, we have the NXG immune system, the 34 available, and the other ones can be on demand, but these are available on the shelf. So you can, ask us today. You don't have to wait the 15 or 17 weeks of humanization. So if you order them today, you can eventually have them shipped out of our facility next week. Of course, if you don't ask too many, but in the 50, hundreds, usually that's possible. Either you want them early without the QC, that's possible, or at 15 weeks after the quality control. And depending on the different backgrounds of the models, the applications will be different. And I think Krishna's colleague will talk about this this afternoon. So now we're moving forward, and the immunodeficient animals used for humanization is a field that is growing and very actively, the development, the R&D on these animals is growing very fast because we understand the requirement, and some of these preclinical assays have very precise demand, and obviously sometimes the humanized mice need to be adapted depending on the studies that are done. So we are moving forward now, and these are things. So we are we have designed this upgraded model of the NXG that is also mutated for the B2M, which makes this, like I was saying, when you're grafting PBMCs, you will have a very short time point to do your studies. With this animals, actually, the host versus pathogen issues come much later. So in this case, you have much more time to be able to do your work. So this is an interesting model where finishing up the proof of concepts on this, but these will be available in a couple of months to order as well. To answer Suresh's question this morning, this is a very exciting model where this one is the highest level of immunodeficient that you can have in a mouse. And this one is so immunodeficient that you don't actually need to gamma irradiate or treat it to deplete the mouse immune system. You can graft your human cells directly without immunodepletion. So this is very interesting because obviously immunodepletion has some consequences that can impair some aspects of the results. So we are really excited, and this one is almost ready to go. It is ready to go, actually, as of today. So please contact me if you're interested, and I'll put you in contact with the people who can provide even more detail. And and we're ready to collaborate, to have, since these are the first ones that come out of our facilities, to have test batches and things like that for you if you're happy to work with us on validating these animals. Oh, sorry, I was having it here, but so this is the c-kit, sorry, animal. Okay, so why order our animals? I mean, I've explained all about the genetic quality and microbiota quality, but in terms of the HIS animals, our quality assurance also is this really, really in-depth immunophenotyping before the quality control after. We have full access to the donor, so the cells that we use for engraftment. So CD34, if you're with one donor, you can engraft maybe 6, 7 animals at best. So if you're going for big batches, this will require multiple donors. So if you're really conscious of having donors that have similar profiles, that we can pre-select the donors for you as well. Or if you want donors that have an enriched subcell population, because this is what is going to be useful for you down the line in your studies, that is also possible. So we really can work and communicate with you. We have a whole, our immuno team over there is really that they have access to all the CD34 data that we will share with you. After the quality control, obviously, also from animal to animal, there might be some different levels in terms of the subcell populations that are more humanized than others. So we can pre-select the animals that are adapted really to the subcell population that interests you for your work. So these are really personalized services that we do that are, in some cases, actually included in the price of the animals to a certain extent. So we really want, and this is part of the 3 Rs as well, to be able to provide really animals that are as most adapted to the work that you're going to do so that you can actually extract data from the 100% or 90% of the animals, because there's a lot of work and expenses for you and for the client and for the breeder to actually design these animals. So we want to make sure that they are used as optimistically as possible. And this is why we do all these quality controls, and we communicate a lot with the client to make sure that we provide the animals that are as most and best adapted to the studies that you're going to be doing. Janvier Labs is actually part of the Janvier group now. Over the years, Thomas Janvier and the Janvier team leadership have been partnering and acquiring different startups and biotechs all over the world. And this really expands our portfolio on the different types of services that we can provide as well via the Janvier group things. So JC Discovery is actually Janvier Labs. This is all our immunophenotyping and our custom model design partner. Most of the activity is actually done in Janvier as well. Cardiomedics and physiogenetics, so people who are working in metabolism disorders or cardiac-related disease, they have a full platform dedicated to these types of services. Obviously, it's always linked to the animals that we provide and that we work with them on these things. MyMab is very interesting. They have a very cool platform, really high throughput to do monoclonal antibodies. And obviously, they can test and profile them for you as well. And I'll talk a little bit about Eucharist here, which has, because it's really related to the talk today. So Eucharist via Janvier Group, or Janvier Group via Eucharist, if that's the right way to position it, actually provides now humanized animals that are liver humanized, okay? And I'll go a little bit into detail into what that is and how this is kind of and this is unique to the Eucharist model, there are a few other companies who make this, but not at this level of quantity. So what is the FRG liver humanized animal? So I got these slides from Rob, from Eucharist, who actually presented this a few days ago. Basically, it's mice that have human livers, okay? And that means that the hepatocytes of these livers are human, they are derived from the human, and they're going to produce all the metabolism that they're going to produce and treat is going to be human-related, okay? So this is really, really a game changer in terms of metabolism and liver-related disease analysis. So FRG, what does this mean? So F is for the Fa. Okay, mutation, I'm almost done. RAG and gamma, so these two help in the engraftment, and this one actually prevents the mouse hepatocytes to be put in place, okay? So this is how they work. They have also a molecular switch to make sure that the engraftment of the human hepatocytes is optimal. And so very similar to the immune system humanization process, what this does is that they have a bank of human hepatocytes that are competent, and they have a whole service to be able to culture and revive human hepatocytes in other mammalian species. So we're talking about humanization, but we can do monkeynization, pigization, I don't know how you, all the things depending on different applications. But yeah, so you have your donor hepatocytes, here human. You engraft them in the FRG model that has been depleted from the mouse hepatocytes and enhanced the possibility of engraftment of the human donor. So this takes a little bit while, but then your liver is almost fully populated with human hepatocytes, and it's very stable over time. You have up to 180 days to do your experiment. And these are also readily available on the shelf, meaning that you don't have to wait 180 days for the animals to be provided to you. So this is my last slide. So obviously these animals have very different and very exciting applications, and many, we can provide many papers and references if you're interested on these animals. They have been used really broadly in the States and different pharma and CRO partners that have been working with Eucharist over the years. So obviously the different applications to test different gene therapy, obviously the liver disease related, like I said, which is very interesting, that these hepatocytes have a human phenotype. So that means that they have a human metabolism, and you can treat all your toxicity really, this is translatable now. You're using your DMK and toxicology studies in a human liver in vivo, basically. So where it's really translation into the second part of the talk here is that we have, Yirkes has clients that actually use these humanized liver models to actually test the organoids. Okay, so this is where the two can come from, to actually have a proof of concept that your organoids work as good as a human condition. Then you can, graft these organoid cells into the FRG mice and see if they repopulate properly. And this, you have your, the validation is there. So instead of going, into clinical data and studies that would cost a lot, you can actually have this pre-validation in these humanized animals. And so what this means is, again, with the importance of these humanized animals, whether they're transgenically humanized, immune system humanized, here liver humanized, and some cases, so Eucharist tells us that they're working on a model that will be liver humanized and immune system humanized, so we can combine the two. So this really brings human relevance to all the earlier stages of the drug development process, right? This is really where the 3 Rs come into place, that you have really specific models now to answer and to screen your different components. And as we were discussing last night about the importance of failure, okay, these models actually allow you to fail faster. Okay, you're allowed to screen the drugs and the components that don't work much faster, and even though these are expensive, and much cheaper than if it was an actual clinical trial down the line. So failing faster is actually the purpose, and by doing so, you actually use less animals, and you're more specific in your studies as well. Okay. On that note, I will finish here. Be here all day. I am in Bangalore, so you can call me. I can come and meet you in your different areas in India if you're interested in discussing these further. And we have other services, but today was really focused on this. Thank you very much.
Thanks, Lily. It's wonderful talk. I'm quite inquisitive about your organ transplanted humanized model. Are you or Janvi working on any other tissue other than liver as a regenerative scope?
Yeah, so not as directly, but via the Janvier Group partners. We have partners who are doing urogenital organoids, and we see how the platforms can be translated. So these are in the process. It's not there yet. The organoids are, the urogenital organoids, but these...
So the humanized c-kit mice, are they available to be shipped to India from France?
Yeah.
And what was your experience on how they are when they come here, or on their status of immune compromising?
So I don't have the data yet because they're not, it's actually being validated now, but I'm happy to put you in contact with the people there, and you can ask these questions. Some of them is not yet, it's confidential. So, but
sure.
We can get in contact, and I actually put you in contact with the people who are actually doing the work, and they'll be able to answer the questions much more reliably than I am.
And I don't want to put you on the spot, but can you give a range of the cost, not exactly, of the c-kit mice?
I don't know.
Okay, thank you.
Actually, I do. I have it there, but I don't know right now, so I can give it to you later.
Yeah, very nice talk. Thank you. Quick question, and I'm going to put you on the spot. Where does regulatory stand at the end of the day on these models being acceptable to them? I mean, in one way to look at these models these are neither mice nor human. So the drug is going to be so confused as to which way do I go. So where does regulatory stand on accepting these models? Do you know?
That again, I don't know, but they're much more accepted than the non-humanized version assays. That's for sure. Yeah. So, I think Suresh might be the one able to answer these questions. He's much more linked to these regulatory concerns.
Okay.
But it's really, I know that the FDA and the European Commission is really pushing for the validation of these. So and this is why there's so much development, because they understand and they see the value of these animals, especially, like I said, to fail faster, basically, right? And this is a huge concern from the whole industry, and that's
no. So I think the proof of the pudding would be you test 10 drugs that have failed and 10 drugs that have worked in humans. Do these models fare better than plain garden?
Yeah, that would be very interesting. So we're not a
I
CRO.
think that's the study
exactly.
we're looking
so
for.
We don't do the work ourselves, right? We provide
okay.
the animals, right?
Okay.
So, but we partner. We have very big pharma groups who work with us.
Okay.
So these I can. For some of these questions, we do have the contacts of the scientists in these pharma groups who have done the work, and we can put in contact, but we have a conflict of interest and confidentiality issues that we cannot pronounce ourselves on these things.
Right.
But that is very interesting information, obviously. If you go back, but these are very expensive. That means you're wasting a lot of money to prove something that has already happened.
But it's chicken and the egg. Without validation,
exactly,
I wouldn't spend the money,
no,
right?
No,
So,
these are very important questions, of
okay.
course.
Thank you so much. Yeah.
Hi Lily, thanks for the great presentations. I have one technical question. So you put a lot of effort in generating these mice, right? You cannot propagate them, so they're all one-time use.
Correct.
And why?
If they're not genetically, the ones that are genetically engineered, I mean, it's in the genome. Whatever is genetically engineered will be transmitted, right? But this is cell or tissue engraftment. That's not going to go through the germline.
Okay, thank you.
Yeah. This is where these are expensive animals, so expensive experiments, but less expensive than a failed trial.
I really answer the question which is related to,
Oh, sorry.
So, which is related to FRG model, especially hepatocytes. What kind of advantages we are getting over normal human hepatocytes? Did you compare that kind of advantage and disadvantages? Normally, human hepatocytes is basically for drug metabolism in phase 1 and phase 2. But now it's very cost effective now. I think you don't require to worry about it. And moreover, the translation from clinical to preclinical is almost established there. So what is the advantage of FRG model to bring into the science to understand the metabolism way here?
Yeah, like I said, it's a pre-screening process and a validation process, right? But if you want more detail on the FRG, I'm happy to put you in contact with the Yercuris team, who have all the client data and all the assays on these information. They have a lot of we have a whole list of publications and things like that we can provide you. But yeah, I didn't really quite understand your question. The advantage compared to what? To
normal human hepatocytes for drug metabolism.
You mean cells?
Yes.
In culture?
In cultured cells.
Yeah, but here you have the whole in vivo environment and
no, that I think we need to have a comparative data to convince
those data I'm sure are there that we can provide. Yeah.
So in addition to the drug metabolism studies, where are you actually placing this FRG model? In toxicity or any other areas?
In all the areas, many other areas, in gene therapy, in metabolism, in NASH, MASH, translational organ testing, vaccine as well. Yeah.
So is there any, in addition to, continuation to his question, so what is the regulatory standpoint of you for accepting these hepatocytes?
In the US, they're very well accepted. This is the standard model for all these types of diseases.
No, IND, you can put in IND application that, we have conducted this assay for
yes,
FRG
for some
model.
applications, yeah.
Oh, that's good then.
Yeah, following up on that, the question comes is that these are humanized for one protein or one cell, humanized only for one protein or one cell line, etc. But the rest of the mouse is the rest of the mouse.
It's still a mouse.
Yeah,
it's still a mouse,
still.
yes.
So
it still have
it's
a
not
tail
human.
and ears.
It's not a human.
No, it's not.
I mean, I've been involved in so many IND discussions. We go there at the IND, is mouse looking like a human, or a rat looking like a human, a dog looking like a human? And we don't know, because the data from all these three species is different compared to what we are predicting for human, that we don't even know the outcome of it is. So the question comes is that, as people are asking, regulatory validation, you have to show the clinical validation of these models, and to show that, yes, my model is actually clinically acceptable model. Until you do that, these models are models, that's it.
Yeah. I mean, the FRG has been shown to be an acceptable model, for example. For many
I mean,
aspects,
acceptable in terms of the preclinically, you want to test it as a model.
That's the aspect of preclinical. Clinical is human. That's the next step. You cannot. You will not replace the clinical data. I mean, at the end of the day, but these high-end and very precise models allow you to be much more efficient on your preclinical studies. That's what the advantage is. Okay? Yeah. But you still need humans. I mean, at the end, this is for the human, right? So, yeah.
Thank you, Dr. Lily, for that insightful presentation. We are grateful for sharing your valuable insights on Hanware Labs as a new partner for our preclinical research studies and the various models Hanware Labs provide. It was very interesting to learn the process of production of these models and the rigorous quality control measures they go through. Now, I would like to invite Professor Chauhan to present a token of appreciation to Dr. Lily and Dr. Manu also. To invite Dr. Manu to take over. Thank you.
Okay. Thank you, Lily, for the wonderful talk. So it's my pleasure and honor to introduce Dr. Mandar Kulkarni, who's our next speaker today. So Dr. Mandar is a seasoned leader with life science-focused strategic and business development experience over 2 decades. Dr. Mandar was an executive team member of a seed-stage startup in New Haven and has served in leadership roles in several precision oncology companies in India, where he routinely collaborated with leading institutes across the country. Currently, he serves as the CEO of VCR Park, Vyas Cancer Research Park in Vizag, which is an amazing facility. Please do go, because I have been, so I tell others also to visit, for their unique incubator accelerator to support startups, international collaborative projects, training programs, and clinical research. Please give a warm welcome to Dr. Mandar. Today, he will give us his valuable insights on the evolution and refinement of immuno-oncology mouse models. Can we have the IT support, please?
Why is this not clear? Oh, yeah. Okay. Okay, thank you.
Good morning still. Pleasantries at the end. I want to, in the interest of time, I want to get right into it. I hope to address 2 questions through the progression of the slides that I want to show. The first question is, why do we really need to humanize mice? This was touched upon, and I hope to convince you that there are 3 reasons why we need to do that. One is probably the most straightforward. The mouse immune system is different from the human immune system. Therefore, you need to have a high fidelity reporting mechanism in mice for whatever pathology or disease you are attempting to address. The second reason is probably a little bit more nuanced. Almost every pathology, I'm going to focus on oncology, but almost every pathology interacts with the immune system. And therefore, there is a need to humanize the immune component of the mouse for us to again get a read of what the pathology is leading to, and is there a way for us to exploit the ability of our immune system to attack the pathology and restore normalcy in the individual. And the third reason is to fail fast. This was discussed or has been part of the theme since the morning, and this has to do with the toxicology. There are a lot of regulatory discussion happening, but my sort of overarching response to that is when skid mice were created, same questions were asked. When we had NSG mice, same questions were asked. They are used today. Being at the forefront of cutting-edge technology provokes these questions, and I think it's a healthy discussion to have. And I believe that these will evolve over time and be accepted by regulatory for sure. It's simply a timing issue. So those are the 3 reasons why I believe humanization is important, specifically in the context of oncology, and a specific smaller reason as it relates to oncology will come up through the slides. The second question towards the theme of this conference is, how far are we from alternatives? And I'm going to be presenting a very exciting technology that potentially has an alternative in our grasp to answer some of the questions that have been brought up, right? So I'm going to take a step back and talk about VCR Park a little bit for probably 5 to 8 minutes. Why VCR Park? Clinical oncology is practiced, surgical, chemo, and radiation. But the field of oncology in the last 20 years has dramatically seen a transformation, and there are so many different areas of oncology that need support. To add to this, India harbors a disproportionate burden belonging to the low and middle income countries of cancer. And unless we address cancer in our own country through our own efforts, it will take much longer for us to be able to address our burden. So it's important for us to think about the Indian-ness of cancer. And therefore, we are focused on translating ideas into products and services in India at VCR. That is our vision and mission. Interestingly, US has set the benchmark over the last 50 years, where 50 years ago, 80% of cancers in the US were clinically presented in late stage with poor outcomes. In the last 50 years, that number has gone down to 20% with poor outcomes. So they have favorable outcomes for almost 80% of their cancers just by the function of the ability to detect them in earlier stages. We today are at 80% late stage detection, and I don't think we have 50 years before we can make a dent in that number. So we have to be proactive, passionate, and committed to this cause today. India traditionally has been very good at manufacturing, distribution chains, late phase clinical trials, and more recently biosimilar efforts. What India lags behind to the Western world is the innovation axis, specifically the translational axis as well. So that is some of the ideas that we have incorporated into the being of VCR Park, and we hope to address these questions over the next decade or so at VCR Park. How do we do this? So like I mentioned, we surveyed India, and the first 4 steps of the value chain going from discovery to commercialization involve a lot of different stakeholders coming together to be able to accelerate the path from idea to clinical development. So what we did is actually reached out to high quality strategic partners across the country who are doing these things at global standards and integrated them into our platform, so to speak. So we have physical presence, but then we also have strategic partnerships with really high quality partners who complete this value chain and accelerate the path to clinical development. We are led by a visionary leader, Dr. Himabindu Gaddipati. She is a hemato-oncologist trained at FDA, NCI, has an MBA in tech transfer management. You already know me, but we are also supported by a large team. You see Suresh here. He's our animal model expert because of all of his extensive background. We have a pathologist, drug development expert, as well as a nanotech expert on our core team, but then we also work with several organizations across the country. There's hospitals, there is academic centers, there's strategic partners that I mentioned. We are recognized as a Bioness center in the country under Department of Biotechnology. And at our incubation center, so far we've been able to incubate 11 companies, most of them working in oncology. All of those circles have faces to them. These are of most note is the group of this key opinion leader oncologists who run early phase clinical trials and are at the epitome of their own practice at their own centers. And just to name a few, Tata Memorial Kachchar Cancer Center, Mahindra University, etc., etc. So we're working with the best of oncologists across the country on the clinical side as well. So as I mentioned, we have physical presence in our core lab, which is state of the art. Manu has been there. I invite all of you to please come and see what we have put together as a translational platform. In addition, we have a local hospital that is connected to us directly. This is a cancer hospital which gets us a lot of clinical access to retrospective samples as well as prospective enrollments. And with the strategic partners of Biobank, Jugnu is here today. Small animals, we work with Hylesco and hope to work with JAX and Hanviers. We're able to incubate startups, but then also attract late preclinical and clinical stage technologies from the West, which can have a big impact on Indian cancer scenario. In addition to this, like I mentioned, through the key opinion leaders, we're able to scale the studies that come in across 10 different sites in the country. So we essentially take a view of how big the study is and how diverse the population needs to be, where do we need to go based on the more localized incidence of different cancer types in India. So this is our physical footprint. We look like this in Vizag. We have a lot of high-end capabilities built into our core facilities. I'm not going to belabor, but just to say we have a confocal imager for high content imaging, in addition to some microscopes. We have several molecular platforms, quantitative real-time PCR, plate readers, etc. We have advanced pathology, Roche Ventana, autostainer, and fax machine. We have genomics at our own facility. We have some protein characterization instruments available in our facility. And we are currently working on a partnership with Thermo to add LCMS to our facility. So this will complete our translational facility to quite cutting edge extent to be able to accelerate many studies at VCR Park. So the next phase is quite exciting for me and potentially for all of us. This is our hospital. It is NABH accredited and CDSCO approved to conduct clinical trials. So in short, we were the first truly private company under Bionest. We are the only Bionest focused on any disease area, which is oncology in our case. We have strategic partners. We have a hospital, and we have incubated 11 startups at VCR Park so far. So I guess this is duplicating. Apologies for that. So what now? What do we do now? How do we take this to the next level? We wish to become a center of excellence for oncology and immuno-oncology from late discovery to phase 1 clinical trials. How do we do that? There's proteogenomics, there is advanced models. I'm going to talk about one of them today. Early phase clinical trials, phase 0 and phase 1. And I'm going to talk about phase 0 today. AI/ML expertise through our strategic partner Persistent Systems. And potentially sometime next year, we want to be able to integrate cell-based therapeutics as a service into our platform. We have a universally connected lab through Thermo. This is in progress right now. We're putting this platform together where all the instruments are connected on a single platform managed by sample manager LIMS to have tracking and continuity of all the samples, be it research or clinical, coming into the lab. This is our clinical trial model. In the pre-precision oncology era, clinical trials were relatively simple. There used to be a sponsor and a CRO working together, and the CRO would manage all the different requirements for getting samples from the biobank, pathology, all the clinical sites where patients needed to be enrolled. In the current scenario of precision personalized oncology, there's too much happening. There's a lot of chaos. All of the stakeholders are talking to each other, and this has become critical. So our model simplifies all of these activities, and because we have consolidated several stakeholders on a single platform, they all still get paid their share, but we are able to synchronize and facilitate all of those interactions as more seamless compared to a traditional CRO. And we have our own employees as clinical research coordinators placed at the clinical site where enrollment is happening, and therefore there is a lot of direct oversight and synergies and consistency with all the data that's required to be generated for the clinical trial. So this is the traditional timeline, and fail fast is the mantra these days, and therefore I'm going to pivot into translation. How do you translate drugs with preclinical profiles into early phase clinical trials with higher success rate? This is a little bit of a historic review of cancer and immunology evolving in parallel. So on the left you have some milestones, not exhaustive, some critical milestones in oncology, and on the right you have some critical milestones of immunology. Cancer was first defined, this is not a typo, 170 by Galen, and immunology came into its being 18 centuries later, technically. So the first principles of phagocytosis and immunization were described by Koch and Ehrlich in 1894. So clinical oncology existed for a long, long time before immunology evolved and was clearly defined as an immune system. Over the years, Ehrlich coined the term magic bullet based on his infectious disease work and wanted to apply that to oncology. His time was not long enough to do that, but he envisioned the magic bullet for cancer as well. But then on this side, over the years, we realized that there are so many oncogenes. Cancer is complex. Cancer interacts with the immune system. And the success rate of the most effective targeted therapeutic even today is 15 to 20%. Even with PDL1 therapies, which were the next horizon about 6 to 8 years ago, the success rate or the progression-free survival for patients is still between 15 to 18%. So there is still a lot of unknown interactions between cancer and the immune system that we must interrogate for us to improve the success rate. And fail fast comes from the idea that only in 2007, the need for stimulating the immune system to attack cancer cells was propagated. So we're talking 17 years ago. The ability to do that came in 2015. Lily spoke about the NXGC kit mouse, which does not require irradiation. That came about 9 years ago only. So in the historical context, we are really at the cutting edge of interrogating cancer in the context of immunology today. So we have to be able to utilize these models in the right way, in the right context, and accelerate our understanding. There's academic understanding, there's translational understanding, and there's clinical understanding. So all the effort of humanizing these mice is to be as faithful as possible to the human condition. And this is probably the more subtle reason of why we must humanize. The rest of the mouse may be a mouse, but we realize that immunology and oncology overlap very closely. And there are several still unknowns in the immune system and the cancer state where this can be exploited. There are neoantigens that are in infancy. There are several temporal mechanisms. So CTLA4 and PDL1 act at different times of the maturation and the attack on the cancer cells. And there are studies, James Allison has shown some of these studies, the Nobel laureate, that the timing of when you treat with anti-PDL1 versus anti-CTLA4 dictates whether there is progression, clonality, recurrence, all of those different things. So it's a very exciting time to engage in immuno-oncology, right? So this is very hard to read, but the take-home point was already conveyed by Lily and probably going to be expanded on by Charu a little bit later. The point here is there is age, gender, the question you're asking, how many weeks your experiment has to go, which immune compartment is important to you for your assay, what downstream assays are you doing academic or drug discovery. All of these questions have to be carefully answered before you start looking at the catalog of what mice are available. So designing your science and question specifically before going to Janviers, I mean, take their help, Janviers and Jacks, but make sure that you're going to get the answer you want by answering these set of questions. There is a lot out there, and there's a lot of complexity out there, right? So this is, these are some of the other considerations. Do you need only males, only females? What age should they be? What genetic background, what immune background you want? Again, this will be elaborated by Charu. So what I want to do is take the NBSGW, which is equivalent to the NXGC kit, and actually become an end user of an animal like this. What would I want to do? And how would I go about taking a humanized mouse and actually using it in the lab? Why and how would I do that? Now the first step here, Lily mentioned they already sent humanized mice. So this slide actually goes one step before that. You take CD34 mice in these NBSGW. So NBSGW mice have absolutely no immune system. In such a scenario, you have to lethally irradiate the mice before you engraft foreign, in this case, tumor tissue. But if you mutate c-KIT, then you've created a genetic background where these mice are now susceptible, ready to take up tumor tissue on a humanized background, which is humanized by CD34 in this case, and their entire immune system is human. So you are able to look at the interactions between the tumor and the human immune system in the mouse. So you would do that, and then using flow cytometry, you would have to ensure that the CD34 cells that you put in actually represent the immune repertoire of the human immune system before you put on the tumor tissue, right? So that's step one. Step two is to either to make sure that the compartments, the myeloid compartment, the leukocyte compartment, all of the proportionality of different cells is appropriate. And you can do that by taking peripheral blood lymphocytes and running them through a scatter of percentage of the immune cells. So that's step two. That's your validation that it is close to a human immune system. The third step is then to take a clinically derived tumor tissue piece and inject that onto this background. There is a wait time between step 1 and step 2, like she said, it's about 2 weeks. And then once you put the tumor onto these mice, that you have to let that tumor grow. You have about 14 to 16 weeks to do that in these mice. And once that uptake is done, you are ready to then treat this mouse with different drugs. These could be discovery, these could be some validation, these could be combinations, these could be dosing, these could be toxicology, a plethora of different questions that you can ask of these. But one aspect is where targeted therapeutics or an underlying mutation is already defined and the drug response to that tumor is already established, should be your validation set of mice that you have utilized against the tumor that you're interrogating. For example, you would have to validate this entire process for TNBC, however, there is no targeted therapeutic in TNBC. So you would have to exhibit a known tumor. This would potentially have to be a cell line derived engraftment of the mouse, say A549 lung cancer cell lines, which respond to anti-PD-L1 therapy. So you would have to take a set of 6-8 mice and graft with lung cancer, treat it with anti-PD-L1 and show that this works. And therefore your mice are now acceptable as models for the other types of tumors that you are conducting. So this is your process validation. This is not to say that TNBC will respond to X number of drugs, right? So this is your process validation in your lab. So this is an end user perspective of how you would utilize that particular background to create a model where you can screen drugs or combinations or different concentrations, right? There are several advantages to this. Like we have discussed so far, the tumor phenotype exactly mimics the human pathology in terms of oncology and immunology. The response of the test drugs, functional response and genomic clonal evolution is expected to be similar in these mice as far as the immunology and oncology works against antagonistic to each other. Ex vivo responses are also expected to be similar and recapitulate the interplay between these two systems. The challenges are, like it was mentioned, the rest of the mouse is a mouse. Cardiotoxicities, other toxicities, neurotoxicities, they are expected or anticipated to be slightly different from those in humans. And there is historic data on those parameters for mice. And then unknown compounds, I kind of alluded to this. For unknown compounds against no targeted therapies available, you would have to have a third axis of validation, which may or may not be directly representative of your screen, right? So those are some of the challenges that would have to be addressed. So that's the humanized mouse part. I'm going to talk about cutting edge technology in alternatives, like I promised. So this is more in situ technology. How do we, instead of manipulating the mouse, find a way of testing drugs inside the human tumor? Okay. So currently, the clinical practice is iterative. It's more based on the clinical guidelines that are defined by clinical trials and all the approvals and historic data and the experience of the oncologist, which means that you go from diagnosis and then first line therapy, second line therapy, third line therapy. It would be great if we could fail fast. Know before first line therapy what actually works. More recently, the organoids and tumoroids is the space that has grown dramatically in this area. But what if there is something called a nano nail? It's a tiny nanoscale device that can be loaded with 20 different drugs in 20 different compartments. And you place it on the tumor so that there is localized release of the drug in the tumor. 24 hours later, you retrieve the device with a fine needle, and then the surrounding tissue that comes with that core biopsy can be assayed using cell-based assays. So this is a little, I hope this plays. So this is a little cartoon of how this would work. And you see that there are active drugs in each one of those holes, and they are being released in a 24-hour timeline. You can have different depths to this well. You can put different concentrations of drugs in adjacent wells, and then you retrieve it, and then you can do all kinds of pathology, cell-based assays, what have you, including genomics on these to assess drug response. So that shortens your timeline and gives you a better rate of response in clinical settings. And this shortens your ability to treat the patient with the most optimal set of drugs that he or she may benefit from, right? So this is a little bit of a detail of that nano nail. I only have 2 more slides. 2 more. So like this says, drug 1, 2, 3, 4, you can put all of those. This looks much bigger in the picture. This is really a nano scale device. And then what you see is you can have slow release versus fast release of the same drug to assess response of 2 different formulations of the drug. You could put different concentrations, etc. And this has been validated and published to see more localized and slow release versus fast release and diffusion in the surrounding tumor tissue. The other validation of this is the uniform distribution and dosing that you can perform. This is also published work. So you can perform this as well by choosing which drug, what concentration goes in each one of those little receptacles. So what is the impact of this technology? It accelerates translation of drug development by predictable drug development. So this is what we call a phase 0 clinical trial, because it's not really enrolling patients. It's an existing cancer patient going through through his or her own clinical evaluation and treatment paradigm, and you're trying to get a phase 0 read on that patient, right? So this overcomes the challenge of humanizing PDX, taking a long time to do this same assessment. You take a tumor tissue, put it in a humanized mice, takes at least 2 to 3 months to come up with a drug screen out of that mouse. So this is in clinical setting and accelerates your clinical decision, but then the same platform is also amenable to use for discovery drugs, because you can do this in a humanized mouse as well, right? And over time, the biomarker and drug discovery and development data that will get generated using this device then goes back and allows us to create knowledge graphs across many different tumor types, perhaps unraveling a more unified magic bullet for cancer in the future. So thank you for your time. I promised pleasantries at the end, so I'm very thankful to Suresh. I've known him for a long time. I admire his trajectory of doing 3 hours for the last 4 years. I've been part of 3 of the 4. Happy to be here. Thank you to Mahindra University, Dr. Madhuri, Dr. Chauhan. The 3 of us are bound by University of Wisconsin, Madison. I learned last night. All 3 of us have been there. I've known Manu for a long time. Thank you for the invitation and the kind introduction. I guess it was a little difficult to follow Krishna and Lily. Pleasure to be here. And thank you all for your patience.
Mandar, beautiful overview. My first suggestion is to change that from fail early to succeed early. You know, as a customer, I don't like to hear the words fail early. I'd be much better off hearing, we have technology that'll make you succeed early. So that's one thing. The other thing is when you do the cassette of drugs with the nail that you said, right? One element that's missing is when you give the drugs IV, you don't know which of the drugs will get to the site in what quantity. So that element somehow, I don't know how you would capture, but that's a pretty important component, right? Of how, A, the drug-drug interaction and how they synergize, do they get there in the right amount or not? So some thinking on those lines would be useful.
Right, absolutely, that's a great question. First point definitely taken, succeed fast, better positioning. For the application of the nanonail, that is why they prefer to start in clinical settings first, to make sure that you are able to get a good readout that correlates with the clinical response, right? So that is a more direct application to start with. But then to be able to go back to the humanized mouse, place it there, gives you probably a better systemic readout of the drug metabolism, right? So we're still not there. That is definitely one of the challenges of how to bridge the gap to have a systemic output to the drug metabolism. I guess the liver models are part of that as well. So we're at the cutting edge, and this is a very relevant question.
Yeah, beautiful story. Thank you.
Thank you.
Hi, Mandar. Equally, the way stolidly that you all have put together VCR Park is admirable.
Thank you.
So, I think this is more of a comment on the lack of targeted therapy for TNBC or HNSCC. I don't think cancer is an organ disease, you know. There is enough profiling that is being done. We ourselves have done genetic profiling of TNBC, and HNSCC is not far behind. So these models that we are talking about would actually be even better to validating targeted therapies available for other cancers that could be brought to bear on TNBC or HNSCC. So for example, the poster child for lung cancer, ALK, RET, ROS, crizotinibs, and its follow-on drugs. The percentage of ALK, RET, ROS fusions in lung cancer is much less than that in CRC and breast. And yet, right now, those drugs are not being used for those patients that have those mutations or fusions or copy number variants. So I think we need to use these systems with the genomic information, proteomic information, available targeted therapies, and push the way forward for molecular targeting.
Right, absolutely. Thank you for the comment and the lead. I completely agree with the fact that cancer is not only a genomic disease, obviously, that's very clear. So multi-omics, functional genomics, more secretome, proteome, all those are very relevant. But that still leaves out the interactive part with the immune system, right? There are still limitations. We have spatial genomics, and we'll go there over time and identify that. And as you mentioned, one of the clinical programs or phase 0 trials that we are about to initiate with this nanonail is for head and neck cancer. So we can load that nail with multiple known drugs or combinations that can potentially work, and that's why it's a phase 0 clinical trial. What drug can potentially work on HNSCC? Like that, we could do TNBC, like you mentioned. So absolutely, yes. That's where it's an alternative to going back to the humanized mouse and doing classical target discovery, get a clinical readout, but then you can use the same on the humanized mouse for novel target discovery, right? So that's why it's refinement of the humanized model as well. So it goes to one of the R's of refining the model further to get a better readout.
I would prefer TNBC over head and neck only because head and neck is more clinically approachable and radiation is the first line, which is not the case for TNBC.
Right.
And even in TNBC, people have already observed multiple subtypes,
right,
such as androgen receptor minus, quadruple negative, which are much more aggressive. So I think the combination of the kind of omic information plus availability of drugs, plus humanoid models or tumor models that we have in vitro, combined with immunized, humanized animal models, should be able to get us to that point of saying, yes, this is doable.
Right. So TNBC
and the drugs efficacy and safety is already known,
right,
in human beings.
Right. So
it's just a matter of expansion of the tumor type.
Yeah, so TNBC also has an upcoming target, RSK2, 87% penetrance in TNBC as a potential targeted therapeutic. And all the other points taken, I think I want to leave other speakers enough time. So happy to answer offline.
Dr. Kulkarni, yes.
My
question is beautiful presentation, not on the model. Basic of that immuno-oncology. Why heart cancer is rare?
Why heart cancer is rare?
There is no question of heart cancer, right? What is the immuno-oncology there? One thing to the work also, heart is the only organ takes free fatty acid. Over glucose is the fast fuel.
Right.
We think of cardiomyopathy, but why not heart cancer? What could your immuno-oncological link into it? I didn't work in this subject, but I have a story to know from you.
So one of the theories, it's not there yet, is that muscle cells don't duplicate. And when you have cells that are duplicating, they accumulate mutations. And the inability to correct those mutations then leads to transformation of the cells. So we have only limited number of muscles. They can grow in their strength, but not in number. And therefore, muscle tumors are very rare in general.
Thank you. Thank you, Dr. Manda, for that insightful presentation. We are truly grateful for sharing your valuable insight on the evolution and refinement of immuno-oncology mouse models. Yeah, and introducing VCR Park and its facility. And that was a very informative Q&A session. I would like to invite Dr. Jugnu Jain to present a token of appreciation to Dr. Mandar and his daughter Rishali.
This is a pleasant surprise. Vrishali is my daughter. It's an honor to be felicitated by your own daughter.
Now I would like to invite Dr. Manusmriti Singh to introduce Dr. Kasturi Mahadik, our next speaker.
Yeah, so I would really like to thank Dr. Mandar for the amazing presentation. And I think from personalized felicitation to the personalized nano nails that are being brought, I'm from the field of nanomedicine, and this was a very impressive work that has come up. So I'll also actually read upon that. So our next speaker is Dr. Kasturi Mahadik, and she's a scientist turned science communicator. She leads the Center for Predictive Human Model System in AIC CCMB, advocating for human-relevant research in India. Her efforts involve collaborating with scientists to drive confidence in human-centric technologies. Dr. Mahadik works with multi-stakeholders to reform policies related to human-centric research and collaborates with students to drive educational programs in the field. Additionally, Dr. Mahadik engages with the public to advance perceptions about human-relevant research. Dr. Mahadik's interest in this area stems from her foundational work in health and disease at the IISc Bangalore and stem cell biology at the University Paris site, France. Please give a warm welcome to Dr. Kasturi to talk about how AIC CCMB is engaging with human-centric science in India. Thank you, Kasturi.
Hello? Yeah. Thank you for this warm introduction. It's a real pleasure. The Center for Predictive Human Model Systems. It's a science and policy think tank which advocates for human-relevant science in India, both for fundamental and translational research. We are housed at the Atal Incubation Center at CCMB in Hyderabad and are the brainchild of both AIC and Humane Society International, which is an NGO which strives to protect animals. But I guess in science, you can't really say no animals, because if not animals, then what is the big question. With our academic background, we are the scientific arm to HSI's efforts, and I hope I prove that to you in the coming slides. So recently, Robert Califf, the commissioner of US FDA, was in India, and he remarked how India is the pharmacy to the world. There's so much happening here, right? 9 out of 10 US prescriptions recommend generics, and India is a strong partner in that regard. As Dr. Mandar was talking before, generics are our forte, but then we've really lacked in the innovation space. Nonetheless, there are so many great CROs in India today who are the market for a lot of new drugs being developed all over the world. And it is in this regard that we must question, what are the models that we are using for research today? Are we really making a difference? Are we revolutionizing science and taking the opportunity that is granted to us? And so when we look at typical drug development, I'm sure I don't need to tell any of you, 90% of drugs fail when they transition from preclinical to human trials. And there's a lot of money spent in this process. 10 years are lost at the minimum. And what often happens is you're not able to validate the result in terms of toxicity, and efficacy is the even next barrier. So when we look at what was done traditionally, when Olson et al. In 2010, many years ago, tried to assess what is the relevance of drugs in preclinical to their transition to clinical studies, only 43% of the instances were rodent successful. And only in these top 3 areas, there were so many other toxicities that were completely under the radar. Why? When they're important. And as we move forward, we heard so many excellent talks this morning about xenografts and transgenic mice, but then I'm sure you're all aware there are challenges to these models as well. Mice are much smaller, they have a shorter lifespan, they have a very high metabolic rate, and most importantly, they are inbred. They don't have the genetic diversity that a human population can offer you. But then, is there a right model? That's the question, right? Often not. I think we must all keep in mind that all models are wrong, and then there are some that are useful. We have to understand what is our question. And today, I sincerely urge you all to reassess the work that you are doing and identify what model or what battery of models do we need to answer our question. It is very relevant today. Perhaps you may need a mouse, perhaps you may need an ape, but how can we maximize the results that we get from each of them and help us arrive closer to human predictions? So these are the research models that we propagate today. In vitro models such as organoids, spheroids, assembloids, reconstructed tissues, which are among the only ones that have guidelines all over the world, 3D bioprinted tissues, organs on chip, and then there's also in silico, where is computation. I'm not going to delve into the intricacies of each of these. We have excellent speakers talking about them. But I'd like to tell you what's happening in the world with regards to standardization, regulation, awareness of these models, because that's the work that we do primarily. And so, as was again mentioned earlier, it takes a village, but even more so for these kind of sunrise technologies. There is so little awareness, and this involves talking to common man, to students, and also to scientists, because scientists are often trapped in silos. There are non-experts, and then there are others who are aware of some aspects of it, but not necessarily approaching other scientists for collaborations. Today, we need a cell biologist, a bioengineer, a chemical engineer, a pharmacologist, and even a toxicologist to bring together a battery of these tests, which possibly could replace animal usage. So it's very critical that all these stakeholders are working together. What we also need is the fact that these models should be approachable, and so CROs, service providers, should have a battery of these in their portfolios. We also need the industry to adopt these models, to see value in these, and then we need investors to show the potential. What has been lacking in India is an investment in this field. If you see the NIH, ICVAM, or the JRC, or even NC3Rs, they have been investing tremendously in these technologies. A fraction of India's expenditure goes on stem cell biology, and there's even less on organoids or organs on chip. Nonetheless, we have tremendous positivity coming from BIRAC or DBT, who has begun investment in the recent past. I think we need to accelerate this process. And then, of course, the weakest link is the regulators, because each one of these stakeholders, before even approaching these technologies, asks, is this technique regulated? What is the regulatory opinion on these? The FDA Modernization Act was passed, and it authorized the use of these models for the screening of new drugs. Similarly, India has been only the second country in the world to fall in steps with the US. The question is, this happened a year ago. What has changed since then? What have we done? Have we followed up on the promise that was given? Have there been new guidelines which talk about where can these models be used? How can these models be standardized? What are the criteria that these models must meet? What are the 4 characteristics that my organoid model must meet? How must these models be used and validated? And lastly, do we need animal tests at all when we talk about these models? Are they going to coexist? How long are they going to coexist? So many questions. So this is the work that we do at our center. We talk to various stakeholders, and we encouraged the adoption in India and facilitate really so that you can adopt them. Starting with awareness, when we began in 2019, one of the only ways to talk about these models was to have webinars and talk to people about what is the potential of these technologies. But then now we've grown. We have newsletters, publications, white papers, all on these topics for diverse stakeholders. And in the recent past, we catered to master's students, especially because a lot of them, like you, are interested in these technologies, but they do not know what is the intricacies, what is needed to set up a model, what can the model help us understand, and where can we get from there. So we developed videos of specifically for Indian scientists who worked in these areas, exposing the tremendous success that we've had in India with regards to these technologies. So I encourage you to have a look at these videos and also share it with your classes or your students if they're interested. Another aspect that we've also realized is there's a lack of training in this field. And this is understandable. A lot of the training that is present today focuses specifically on one aspect, be it 3D bioprinting, or it could be more about stem cell biology. But there's no integrative learning. And so in 2022, we had India's first large umbrella conference called the Microphysiological Systems by India EMBO. And the aim of this conference really was to bring together all the experts from the country and abroad to facilitate a cross-interaction and have a common forum where we could address the challenges of India and bring suggestions from abroad, because they've done pretty impressively. Many of the speakers from today were there as well. And I hope it has been a learning experience for all of us, and definitely for us, because we had a workshop there and realized there's tremendous interest in learning about these technologies hands-on. So we've moved on to have workshops with regards to AOPs or 3D bioprinting, specifically catered to this. But then as we've moved on, what we've also realized is there's a need for intensive training programs, because knowledge of these fields is so poor. What we've developed is a proposal with Dr. Reddy's, and here we want to have lectures and hands-on experiences, not only for students, but also for industry professionals who'd like to incorporate these models in some aspect of their work. This could be to do with humanized mice or transgenic mice with xenografts. And so our workshops, the first of which was in December, aimed exactly at this. Again, Dr. Pusala and Dr. Manu were a part of this, and we had a very interesting time because we had advanced PhD students who wanted to transition from 2D biology to 3D. And what we want to do now in future boot camps is to have more industry participation as well. So if you or any of your colleagues would like to be a part of these, I encourage you to visit us, talk to us, and be a part of our programs. We are having the next workshop in IISc and CCamp, and the next one at IGIB in Delhi. As I was talking earlier, a lot of scientists seem to work in silos. There's not enough crosstalk between scientists. What lessons from organoid could be applied to organs on chip? How can regenerative therapy help in addressing key questions which are coming up in the field of organ on chip, for example? And so we developed a database of scientists who are working in these cutting-edge fields in India. It is openly accessible, and it's also informational for students who'd like to seek internships in these areas. More importantly, what we'd also like it to be is a demonstration of how this field has grown in India. Several regulatory partners and funding agencies in India have reached out to us to understand this database more carefully and to understand the growth of this field really. So it's proving a great experience for us as to the different stakeholders that it could bring on board. And if you haven't registered on it, we encourage you to be a part of it as well. Lastly, what we've been doing is encourage the generation of enough amount of data which can be assessed for regulatory submissions. This is something which is sorely lacking in India. And so we've given grants to researchers, to startups, to develop these technologies and generate data. And what we do with this data is to bring this data forward in policy discussions and also generate a good faith very early on in the system to have crosstalks. So we've done several such stakeholder meetings. We had the CRO conclave of which Sci Life Sciences was a part, and I just heard that it was a great interaction. So I hope many of you reach out to us and become a part of these conversations as well. We make sure that we have enough government representation, regulatory representation in each of this so that your questions are heard. Lastly, if there's one point I'd like to take up with each of you investigators, it would be how can you be a part of this growing enterprise in India? BIRAC has a scheme called the Early Translational Accelerator. And here what it does is to catalyze academic discoveries into commercially viable ventures. Perhaps this is something that each of you aspire to do. If you would be interested in submitting a proposal for this, do let us know. We are looking for 10 discoveries, which we will put together in the form of a program, and the remuneration is very positive. So if you are interested in transitioning away from your research, have a proof of concept, and interested in developing preclinical models, let us know. A lot of you may also not be interested in commercialization, but what you could be interested in is pioneering this research in India with your core competency. And so if you would like to lead research and development in India, develop a core facility which can support each and every one of your ambitions. Have working groups which are consistently solving the challenges in these fields and working together to influence policy decisions. We also invite you to be a part of a Center of Excellence, a proposal that we're submitting to DBT soon. So do let us know if you're interested, and we're available all day to talk to you. So all this would not be successful without our wonderful team. I'd like to thank Dr. Madhu, who is the CEO of AICCCMB. Our program is a core program under the incubator, and it's a very unusual program from the usual startup activities that we have. Nonetheless, we are supported strongly by his vision, and it has been tremendous to work with him. I also have two colleagues, Tejaswini and Gautami, with me here today, and they will be at their posters, so if you have time, do visit them. Thank you, and I'd love to interact with you.
Hi, Kasturi. This is Dr. Srinivas here. Very nice presentation. So, I mean, we are thinking there is a lot of buzz going on regarding alternative to animal testing. So, do you have any discussion? And so far, did you have any discussions with CDSUO to approve these kinds of organoid systems for the screening and drug discovery process?
Right. So we are in the process of ideating a workshop with CDSCO and ICMR together, because CDSCO looks at ICMR for guidelines on these kind of documents. All over the world, there is no unified context of use. There are no standardized guidelines that the FDA, EMA has accepted. It becomes very difficult for CDSCO, a team of regulators who are not very well versed in this field, to adopt these technologies. So we are on the path to raise the awareness and improve their engagement, but unfortunately, that's not the case today.
Kasturi, wonderful, and I think you guys are doing a wonderful job in initiating this process and everything. Here, my question is, the academics are not regulatory powerhouses. I mean, I worked in regulatory arena for 30 years. I know what regulatory wants. Now you guys are going directly to the regulatory without industry. How are you guys going to make these documents usable by industry. Because industry is the final customer who develops the product into the market. AIC is not going to develop a product in the market alone. Industry has to get the regulatory approval. So how are you guys doing it without industry?
It's not without industry. It will be with industry at all points. It involves industry participation. In fact, the sponsor is the major participant of these studies. So we do have Dr. Reddy's on board, and we encourage you, if you're interested, to also help us.
Yeah, so we need to talk about that a little bit more, because I haven't seen any of the upcoming new technology entrepreneurs are invited to these discussions. I mean, Dr. Reddy Labs, they're CDMO. What would you gain from that, their regulatory knowledge? You won't gain anything. So you need to understand who's the right collaborator here to bring them on board, rather than you guys just going and say, hey, we'll talk to CDS, ICMR, CDSCO, whatever you want to talk to. Okay? So don't take it personally. I'm just telling my opinion about it. Okay?
Great point, because Reddy's are not Reddy's. Most probably you have to select a startup company, MSMEs, who are working in drug discovery, those who are having a drug discovery experience. That's where you will get more inputs. Basically, if you go for a generic company, what input they are going to give. So you have to take the inputs from drug discovery experienced guys and see how it is going to be correlated with the clinical data. So end of the day, you are going to remove the animals, right? You have to understand this. So therefore, I think the involvement should be more from drug discovery companies, whether it could be startup or MSME, so and so. Then you will get more information, which can submit to CDSO. I guess CDSO may not be having information about 3D organoids. I'm 100% right, but that's the reason I asked you a question, whether they have inculcated about this kind of technology before going to screen the molecule.
Support from industry is very difficult to gather. So it is important that we all work together, which is why I'm standing here today. I think it's very crucial that we join hands. Thank you.
Yeah, just to compliment you, very well presented. And I have one name for you who can spearhead a lot of this. This guy here has done it all. So I think you should use his services, would be my strong recommendation. Thanks.
Thank you.
Oh, thanks, Uday.
Thank you, Dr. Kasturi, for that insightful presentation. We are truly grateful for sharing valuable information on how AICCCMB is engaging with human-centric science in India. And you brought up many questions and gave us food for thought. I thank you. I would like to invite Dr. Suresh Pasala and Ms. Ramya to give a token of appreciation to Dr. Kasturi.
So launching Culture of Care magnetic stickers through Kasturi today on behalf of Oncosic Bio and the entire 3R community.
Now I invite Ms. Ramya to take over.
Thank you, Nikita. So now I would like to invite Dr. Viraj Mehta. So Dr. Viraj Mehta serves as a scientist at Sai Life Sciences, where he leads the development of liver-on-chips and liver spheroid assays while supporting the establishment of liver organoids technology at DMPK. Dr. Viraj obtained his bachelor's degree in mechanical engineering from NIT Surat in 2014 and PhD in biomedical engineering from IIT Hyderabad in 2018. During his doctoral research, he focused on developing a personalized drug testing solution for cancer using organ-on-chips and primary tumor spheroids. Dr. Viraj holds 2 patents in organ-on-chips and has honored with the Gandhian Young Technological Innovation Award in microfluidics. With over 5 years of experience, Dr. Viraj has demonstrated expertise in developing complex in vitro models for drug discovery and development. Please give a warm welcome to Dr. Viraj to talk about integrating complex in vitro models, microphysiological systems in drug discovery applications and industry adoption strategies.
Thank you. Thank you, Ramya, for that nice introduction. And I would like to thank Dr. Suresh and Ramya, Nikita, Dr. Manu, and Mahindra University for hosting this conference. So my name is Viraj, and there is just one correction. I finished my PhD in 2023 last year. And then I joined SAI, where I am an integral part to establish 3D culture labs. And if we are not wrong, we are the first CRO to start that in India for our potential clients. So we are developing 3D spheroids, 3D organoids, and organ-on-chip based assays at SAI. And also I would like to thank Kasturi for setting the stage and giving up the background of this complex in vitro models that made my job easy to, I don't have to define in depth. Last decade was full of, validation. We saw a lot of papers in well-known journals, they validated organ on chips, liver on chips, and countless number of papers we have seen in the last decade. And this decade is going to be decisive. In this decade, we are going to know whether industries are going to adopt these models, where the regulatory stands in this adoption. So this topic becomes very crucial, how to adopt these models, what are the industry adoption strategies for this decade. So that's why I chose this topic for today's conference. So first of all, we will define why we need these models, what are these models. So CIVM is nothing but complex in vitro models, MPS is microphysiological systems. What can be the few priority areas? Now we are all talking about 3 Rs, how to achieve 3 Rs, and there can be several areas that can be defined as the most priority areas where MPS can fit in the current drug discovery framework. Followed by that, how to increase MPS adoption from the current scenario. What are the current challenges for MPS adoption? How does the future look like for drug discovery with MPS models? And finally, what we are doing at SAI, that also I will describe, and followed by acknowledgements. So first of all, I think I don't need to describe much about why do we need complex in vitro models for this audience. They know very well the pros and cons of both the models. But just to give a background, there is species-specific differences. The SIPs, the transporters are different from humans. And as Kasturi also mentioned, the rodents were 43% predictive of human toxicities and non-rodents were 63% predictive, according to one historical data that was published. And of course, there are ethical cost and high attrition rates, as Kasturi mentioned, almost 90% attrition rates. And most of them fail due to poor efficacy and followed by the toxicity that is encountered. And one of the points that I would like to highlight is the DILI, drug-induced liver injury being one of the biggest challenges for animals to predict among other human toxicities. And that can also be seen from this next picture. So as you can see, there are several drugs from the big pharma that is currently on clinical hold due to DILI concerns that were encountered in the clinical trials since 2022. So this shows that there is some missing link for translating the in vitro preclinical data to the clinical data. So animal models have been widely useful, but they are not perfect. Are we missing a component of human relevance in preclinical drug discovery? And how can we reduce such failures, money losses, and attrition rates? So then the complex in vitro models where they came into picture. So complex in vitro models are, they consist of 3D microenvironment, primary human relevant cells, biopolymer of tissue-derived matrix, mechanical cues such as stretch to mimic the breathing motion, and perfusion flow. Also, they can incorporate the micro-patterning aspect in the in vitro models. So when we talk about the throughput and the complexity, we can say that the MPCC, which is nothing but the micro-pattern co-culture that is also known as hepatopac commercially, they and the spheroids, they come on the higher side in terms of throughput. And as you go towards complexity, organoids, 3D bioprinting, and organ on chips, which is the highest complexity. However, the throughput is quite low compared to other models. So now we know what are CIVMs, what are MPS models. However, it seems unlikely that we can completely phase out animals, as we all agree to this point in the foreseeable future. Can we define few priority areas? That is very important to encourage the adoption of MPS in the drug discovery. So one way to look at this problem is to look at the projections. So this is a simple BCG research, and you can see that therapeutic area-wise, oncology is going to lead in the next coming 9 years or 10 years, followed by immunology and cardiometabolic diseases. And when we talk about the modalities, the new drug modalities based on the 2023 pipeline, they are going to dominate 56% of the revenue in the coming 5 years. So this, we can infer 2 points from this slide. First thing is, we can define our priority areas in the top 3 disease or therapeutic areas, and we can also start validating the new drug modalities using these MPS models. And this is very helpful for the MPS developers or the aspiring entrepreneurs who wants to start startup in this area. So this defines few priority areas. So assuming strong backing from the regulatory bodies and systemic reduction in animal usage in future, we can define that MPS developer can focus on cancer models, autoimmune disorders, immunotherapies, diabetic models, or NASH models, all the cardiometabolic diseases. Further, validating for new drug modalities is also important. Now, we talked about from the disease perspective and the modalities perspective, which are the top priority organ models that the MPS developers should look into, which are going to be very much in the focus of the industry, the big pharma industry. So I would like to put up a summary of an IQMPS report. So IQMPS is a consortium which is made up of regulatory bodies and the big pharma companies. The main aim is to promote or enhance the usage of MPS, the non-animal alternatives in the drug discovery, and also to address the challenges that are currently with this kind of models. So I would just like to present a very good summary of this report. So there were 39 respondents. So this IQMPS is made up of almost all the big pharma companies who are actually using this kind of models currently in their internal decision making. So liver is the most priority organ. Most of them responded that they are currently using liver-based MPS models in their companies, followed by gut, kidney, CNS, cardiovascular system, and lung. So this defines the priority organs in which MPS developers should focus, and this has the highest possibility that if in case industries are going to adopt these models, this will be the priority organ models that they are going to use. So that being said, few conclusion. First of all, you must have noticed liver, gut, kidney involved in ADME. For sure, if you are a startup company, you can focus on these 3 models, followed by blood-brain barrier to check the CNS permeability of the drug, cardiac model, and lung models. There were also few respondents that they told that they are also using multi-organ models or multi-organ on chips that we are hearing. And most of those models included liver in the overall model. So liver gut, liver heart, skin liver, and liver kidney were the most models that being used at those companies. Now, after setting which priority organs are currently being used at the industry, now one thing we need to answer is where this MPS model can possibly fit in the drug discovery. So now let us answer that. So first of all the respondents that they told, whoever told that they are using liver models, gut models, kidney models, they are using it for internal decision making. They are not filing any, they are not including this data in the regulatory filing. So first of all, these models are mostly being used for internal decision making. For what? For mechanism of action studies to understand the new target, new pathways. Because these models are the highest complexity models. You can make a disease models out of that, and the omics imaging, the big omics data, imaging data can give you a lot of insights to find out new drug, new target, new pathways. So mostly at a very initial level to find the target. And as I told, they are all exploratory studies and no regulatory filing so far. However, there are two success stories that I would like to highlight which had MPS data in them. First of all, from Hesperos, this is an organ-on-chip company based on US, that included the efficacy data for the rare neuromuscular disorder that was used for the repurposing of a monoclonal antibody developed by Sanofi. And recently, quite recently, CN Bio also, which is a UK-based organ-on-chip companies whose development focus is mostly on ADME, they also included the efficacy data for one of the candidates from Inifarm for treatment of NASH. So these two are the success stories so far. Of course, very less, but we are very hopeful that this decade will be quite positive in this direction. So for the mechanism of action and internal decision making, these are currently being used. So for that, we can use them currently in the drug discovery framework. Second thing is the human target specificity. So all the biologics which has relatively less off-target interaction, because mostly all the companies are using single organ on chips, so they cannot look at how the drug is going to interact with the other organs. So if the drug has relatively less off-target interaction, then those modalities can be tested, efficacy can be tested using these models. So biologics, the MPS models can be very suitable to test the biologics, cell therapy, gene therapy, antibodies. Antibodies are very much target specific. So that's why you just saw a good success story with the therapeutic antibody with Hesperos data. Further, oligonucleotides is also one of the very promising drug modality, and to check the efficacy of oligonucleotide has very high half-life, and it has a delayed onset of the pharmacological effect. So these MPS models actually increase the longevity of the culture. So for example, we have seen that using liver spheroids, you can culture hepatocytes for 3 to 4 weeks. So that enables us to test this kind of modality which has delayed onset or a longer half-life. So that is why these MPS models can be used to target those drugs which are quite human target specific. Further, we also saw the immuno-oncology aspects by many speakers previously. So MPS can also be used to check the efficacy of immunotherapies because they are also very human target specific. So of course, the development has been slow. There are some models that have cultured the human immune cells along with the cancer models in the organ on chips. And one aspect that I would like to highlight is immune cell mediated idiosyncratic delete. This is one of the very challenging to predict, and there are some reports that claims that they can predict idiosyncratic delete using immune cells on the chip. Further, as I told, to evaluate efficacy of cancer immunotherapies. And finally, as I also mentioned before, the disease modeling using the omics and imaging data, you can also define new targets from this kind of models. Now, beyond this, how to enhance MPS adoption? So there can be a few guidelines that we should consider. First of all, defining a context of use is very important. You are making an MPS models for what it's going to be used, efficacy, toxicity, clearance prediction, and so on. So define a COU from the starting and then develop your MPS. This is one of the, this is quite interesting. One of the papers that also advocated that can we file the NAMS data with the with the conventional preclinical data in the regulatory filing to enhance the exposure of the health authorities to review this kind of data, because they haven't been exposed to this kind of complex in vitro models. So further, CROs like us can also provide MPS data with animal studies or conventional in vitro data to generate business inquiries, because generating business in these models is quite challenging, considering it's quite new in the drug discovery. Further, we can replicate the animal studies using animal MPS. Instead of using human cells, we can make animal MPS models, and we can compare how our results are on the animal MPS compared to the animal models. Similarly, we can replicate the historical human DILI or any other toxicity data to gain confidence in predicting. So retrospective analysis of DILI for the prospective analysis of the new chemical entities. Then I don't know whether some of you are aware of this or not. FDA Moderation Act 3.0 has already come. It's a proposed legislation to which calls for making a framework to qualify this NAMS in NAMS data in the regulatory filing. So if you want to use this kind of models in the drug discovery, either it should aim to replace or reduce animals, or it should increase the predictivity, or it should lower down the development time of the drug. Then only your method is qualified and you can use it in your regulatory filing. And if you use these qualified methods, it suggests that your application will go with the expedited review. And finally, value addition is very much important. So the MPS, the organ on chips, the spheroids, organoids, they comes with a high price, low throughput. So how do you compensate for all this? So value addition is very important. One of the aspects is everybody's looking at is, can we also incorporate the biliary flow on the liver on chips apart from sinusoidal flow? So to also predict the biliary clearance, which is quite difficult currently with the in vitro models, and only animal models can do that. Second thing is immune mediated DILI, as I also suggested before. And clearance prediction of stable compounds, the metabolically stable compounds. It's very challenging to predict the clearance. So MPS models, because they enhance the longevity of the hepatocytes culture through all the mechanical and chemical cues, these low clearance compounds and the clearance can be predicted. And there are already a lot of reports very successfully predicting the clearance of metabolically stable compounds. Now let's look at some of the challenges. First is lack of repeatability and robustness of the data of these MPS models, lack of standardization, as also Kasturi mentioned, lack of regulatory guidelines, and low throughput. But when we look at the future looks promising using these MPS models. Let me give a glimpse of the future, how the drug discovery is going to look like with this MPS, and it will also address some of these challenges like low throughput and repeatability, robustness of the data. First thing is now US and Europe, almost all the big organ on chip companies, they are making a PBPK model based on their organ on chip data. And organ on chip, basically the dimensions, the volume, media volume, what is the clearance of the hepatocytes that are cultured in them. And using this, using PBPK modeling and the mathematical governing equation, you can make an in silico or a digital twin of the MPS model. And then this step is required to translate your MPS data to the human, because MPS is very small. All the volumes are very small. To how to scale this data to a human system. So for that you need in silico framework. Further, large screening, nowadays almost several companies are coming up with a 96 well plate format of the organ on chips, where you can culture almost more than 40 tissues at a time in a 96 well plate format. So, and they are also compatible with the automation, the liquid handling robots. So using this rich human relevant data, you can train your AI models using deep learning algorithms. And in fact, there are two companies that are already doing it, Curis AI and Vivodyne. Curis AI from the Tel Aviv and Vivodyne from the US. So they are also training their AI models using the human relevant data that are derived from their organ on chip or the spheroids model. Now let me come to what we are doing at Science. So we are currently focusing on 3 things. Hepatic spheroids culture that we are currently establishing it to validate DILI prediction and clearance prediction assays. We are also parallelly developing liver on chips cultures to establish dynamic co-culture model to represent full complexity of liver organ with NPCs especially for idiosyncratic DILI predictions. And we are also generating hepatic organoids culture, and in fact we have some good images to show you. So now coming to this images, we have this primary human hepatocyte spheroid on day 14 stained with actin DAPI, and on the right hand side you can see an organoid stain on day 14 with size of 400 micron. And we are also doing liver on chips cultures using primary human hepatocytes. In fact, we are at very premature stage and we haven't generated much of the validation data, but at least we know that they are being viable for 14 days. And after this, we are actually testing different drugs for DILI prediction and clearance prediction. And here we are also using dynamic flow mechanism using simple rocker based mechanism. And this brings us to the conclusion of my presentation. I would like to acknowledge the members of 3D cultures and organoid groups at SAI. First of all, Dr. Vamshi, who had this vision to start early in India in a CRO space, who is also HOD and Associate Vice President at DMPK, and he will be there after lunch. You can meet him. Dr. Guru, he has a background in liver regeneration and liver organoid development from some of the well-known institutes like UCSF, Stanford, and he is currently principal scientist at DMPK. Arojit, who has a background in neuroscience, also alum of Jackson Laboratory, and he is also working as principal scientist. And apart from that, I would like to acknowledge all the DMPK team members, starting from Venkatji, Prabhakarji, Vishal, Venkatesham, Sahista, Ajitji, and all the team members for their small contributions and giving lot of insights as to how to go ahead with this kind of models to establish them at SAI. Finally, I would like to leave you with this kind of beautiful images because microfluidics is very close to my heart, and I always like to give an artistic perspective of the microfluidics, and this is one of the artistic perspective of the microfluidics that is generated by Dr. Albert Folch from University of Washington. Thank you so much for listening to me patiently, and I would like to again thank all the organizers for inviting me, and if you have any question, I would like to take those questions, and otherwise we can also discuss while having lunch.
Hey, Viraj, thank you for a very good presentation. It gives you background of all the things that are happening. Excellent. My question to you is that you said you're developing DILI model, right? Can you tell me how many people actually get DILI in clinical populations?
I don't have an exact number, but I just know that 40%, 30% of the drugs fail due to toxicities.
Not liver toxicity, the cardiac toxicity.
Yeah, that is composed of.
All those toxicities are there. Yeah. Liver toxicity is very rare in terms of the failures in the clinic, in the preclinical toxicology studies. You look at the data. I mean, I know all the data very well in that sense. So what are you trying to predict in the absence of the clinical data?
So one thing from the historical data and from the literatures, I came to know that from all the toxicities that you mentioned, DILI is one of the hardest to predict from the animal models.
Yeah.
And if we can, because these MPS models we need to add the value. right? If there are already established models for this kind of other toxicities, then where the MPS is adding value? That's what I'm trying to, that's what my point. So that's why this is very rare area, but there the MPS can add value.
So my friend here, he worked at Park Davis. He knows the troglodytazone history. This was in 2000. Wonderful drug, no animal toxicities, no nothing. Go into the clinic, no delay. It's marketed, 24 people died, and many others have very severe liver toxicities. It was not predicted in clinic, phase 1, phase 2, and phase 3 trials.
Yeah.
It was not predicted in animal models.
Yeah.
So you're talking about taking 2 million people, only 24 people having the toxicity. How do you know what the problem with those 24 people out of 2 million people? How are you going to use that in your prediction model?
So one more thing is all the new drug modalities. For that, we have very limited data, right? We know that for small molecules, this is very rare. But what about the new drug modalities? Do we know toxicity data for that? Do we know any data whether they are highly liver DILI causing agents or not? So that's why I think that this model is going to bring some value to the drug discovery.
Okay, thank you.
Viraj, very nice presentation. So I would like to learn from you because I don't know anything about that. But you mentioned about several times about the clinical clearance prediction. So when you're talking about the liver, there are fundamental parameters which normally influence about the drug metabolism and pharmacokinetics. When it comes to entry in the liver, so there are a lot of physicochemical properties like permeability, lipophilicity, and at the same time protein binding also plays a major role. Yeah. So when you're trying to establish, again, the outflow and inflow, and again, KP rate constant, how do you maintain that in the case of when you're developing a model? Can you please explain that? So I don't know, because when you're developing a model, any organ chip model, how do you integrate these systems into a model to understand the final outcome of the clearance mechanism? Again, the phase 1 and phase 2. Phase 2 undergoes oxidation, followed by phase 2. So conjugation mechanisms, and the structural changes also happens. How do you take that parameters into the consideration? We are developing a liver models. Could you please explain that?
Yeah, so that's a good question. So first of all, all systems will have a different drug binding. So plasma protein binding is something we measure in a tier 1 assays at SCI. That we assume that is going to remain same in organ on chips, but the binding to the organ on chip material is going to vary. So that we measure using, while running the chip without any cells. So we just incubate with the drugs and we measure how much drug is going to bind to the chip material. So that we can correct it for that when we are running the assays with the cells. Second thing is, first of all, using for clearance prediction, what we do is we first need to generate some quantitative data or the functional characteristic of the hepatocytes. For example, what's the albumin production rate time-wise? What's the urea production rate? Whether there is CYPS expression using the using the PCR studies. So after going through all this functional characterization, and we ensure that this model at the threshold limit of the albumin production, urea production, and all the CYPs are expressing, then we can incubate the drugs for the clearance prediction. So first of all, and this is how the workflow is.
What about permeability coefficient, KP, while moving the drug from inside to outside? Are you going to take that into the consideration?
So it's pretty challenging to measure in the chips, but in silico models only we have to look for that.
And moreover, biotransformation is always concentration dependent.
Yeah.
Amount of the drug that is going into the liver and combining with the conjugation mechanisms.
Yeah.
So do you use any standard molecules which are having a high clearance, moderate clearance, and low
Correct.
clearance
Yeah, yeah.
to establish a robust validated method?
Yes, yes.
What are
Yeah.
those drugs? What are those drugs?
So first of all, warfarin is one of the low clearance drug. Midazolam is on the high clearance side. So such, very well-known compound set of compounds we take and then generate some data from what drugs that are already for which the clearance is known. So as you told, low, moderate, and high, all three we need to characterize on the liver on chips. Yeah.
In the same question, you are telling that protein binding data, what you are getting on the basis, but without toxicokinetics, how you know what is the compound inside the tissue? Protein binding data only for blood data, right?
Yeah.
So then free drug concentration has to go, what Professor is asking.
Hmm.
Without that, how will you measure what drug? How much is there without
So
toxicokinetics data?
we can measure the intracellular concentration. That is one way to do. So what we can do is we can get the cells out from the chip. We can detach them, and we can permealize them and get the drugs out. And using LCMS, we can measure how much intracellular drug is there. So that is one way to look at that problem.
So I think that professor is driving at, if I understand very correctly, that, today medicinal chemistry produces brick dust. We call it brick dust because they're not even soluble at nanomolar concentrations. Okay? So with that insoluble drugs, how are you going to measure anything?
Yeah, that's definitely
Right.
that's a big problem with the big molecules. So the solubility is an issue. Yeah, that's
You'll get a lot of false negatives.
Yeah, that is for sure. So that's why we run all the tier 1 assays before that, solubility, permeability, and then only we should go for all these complex models. Otherwise, that's a waste of money. Yeah.
Okay, thank you.
Thank you, Dr. Viraj, for that insightful presentation. We are truly grateful for sharing your invaluable insights on integrating complex in vitro models and microphysiological systems in drug discovery. Now, I would like to invite Ms. Reshma to present the token of appreciation to Dr. Viraj Mehta. Now, let's break for lunch. When we return, we will hear from our few next speakers for the day. Please return promptly in an hour, and you can also enjoy the poster exhibition by our scientists. Enjoy your lunch. Thank you.
Please, a kind request to be here by 2. We'll start sharply at 2.
So shall we start? Yeah. Good afternoon, everybody. Everybody wants to sleep now, right, after lunch? Okay, so I think now it's really tough for you to be awake. So I'll try to tell some more boring stuff so that you can sleep well. So my name is Jagadish Ganla. So I represent something called Federation of Asian Biotech Association. And in the morning, if you see sessions, especially our dignitaries from AICC CMB who presented, we want to do a lot of non-animal tests and everything, but there were questions from Srinivas about CDSO, how they will take it up. So there is a very big gap between industry and academia. So if you see a couple of talks later as well, they will talk how academia thinks, how industry thinks. But there are very few organizations who actually can connect them. So the Federation of Asian Biotech Association is one of the organizations. The main aim of starting this organization is to connect academia and industry. So it started in 2004, has global presence in around 22 countries. And the main aim is how to bridge the gap between industry and academia. So it connects various players. It could be startup, it could be students, it could be big companies. Anybody under the umbrella of life sciences, it can be connected through FABA. So FABA also has many international chapters. So we are headed right now by president who is from Bangladesh. And one of the thing we are very grateful is that the executive committee board members. So many are here, Dr. Uday Saxena, Dr. Suresh Pusala, Dr. Ajit Kamath, Dr. Ranjan Chakraborty, many are here. So we are all, this is the think tank of FABA. So whatever we do, with the help of them only we do. So what is FABA and what we do? So FABA, as you understand that, it's a big network of people, like-minded people. It could be from academia, it could be from industry or from the startup world. So using that network, we have something called as FABA Academy. In Academy, we do human resource development. We train students, we train industry insiders on anything that they require, mainly for their development. And we also have entrepreneurship. In entrepreneurship, we help startups. It could be funding or it could be mentoring or anything what they need. So I'll show you a couple of examples what kind of activities we do. So here is one of the examples that we have done in 2020 about how to take care of supply chain challenges of vaccines. So one of the example of activities that we do. And another activity that we have done with the state of Andhra Pradesh is how to tackle antimicrobial resistance. Have you heard of antimicrobial resistance? Yeah? So that's going to be the next pandemic that we all have to be prepared. So that's how World Health Organization wants us that every state or every country should have their own plan. So we worked with Andhra Pradesh government to give them how to basically implement a plan. So we also have done Hyderabad Life Science Innovation Cluster. So Hyderabad is kind of heavy on pharma, like we have a lot of pharma companies, as well as we have a lot of central institutes like CCMB, CDFD. So this, as we said, as I said, academia industry interaction is our core. So we invite a lot of players to such kind of meetings, and then we make them collaborate. And not only that, we also focus quite a lot on soft skills. As we all know in our academic system, we are trained quite a lot on the techniques. Like, you know a lot about CRISPR, but do you know how to make a good CV? Or how to do interview skills? How to understand whether you are good for academia or industry? So all these things are not taught at the university curriculum. This is where FABA wants to help. And we have something called FABA Fridays, where every Friday we mentor students. And we had around 20 sessions. Now all our sessions are recorded and we put it on YouTube. So if you just type FABA Academy on YouTube, you'll get around 20 lectures. So these 20 lectures are like a gold mine for a student. So we also do help startups in raising money. So have you heard of Shark Tank? Yeah. So we are doing something called Whale Tank. So we want bigger fishes. So we connect startups with venture capitalists. Because in India, if you look at the biotechnology companies, there are around 7,200 biotechnology companies that have been registered so far. And they all get one or the other grant from Department of BIRAC, either 50 lakhs, 10 lakhs, 20 lakhs or anything. But afterwards, where is the money? So we are not US or we are not Europe to get big venture capital investment. That's where FABA is helping out a lot of startups to get money. And this is our event is called as Whale Tank, which is going to happen on August 16th this year. August 17th, sorry. So we also do several events on biosafety level. We collaborated with somebody called Health Security Partners USA, where we train companies working on vaccines or somebody who's working with BSL-3, BSL-4 facilities. We train them how to take care of cyber security or cyber biosecurity. We do workshops on artificial intelligence in life sciences. I'm just showcasing like what are the works that we are doing. And something that you all should be looking forward is the drug discovery workshop that we are doing, which will be from August 3rd to August 16th. I'll basically show you more details. So how FABA is positioned, like what FABA can do for you is, FABA is an upskilling center for many of the students or industry insiders, or it is a business connector if you are a company, if you are looking for new businesses or a startup, or if you are looking for knowledge, then we are a think tank organizer. Our EC committee itself is a big think tank. And we also help you in recruitment if somebody is looking for any interns or employees. So we have big CV database. We are a business development agency. We basically provide network and we provide investment opportunities as well. Something that I want to show is that I'll explain later. So FABA also give awards. So FABA recognizes leaders in the field. So FABA gives awards. Those awards are 3 types of awards: FABA Young Investigators Award, FABA Excellence Award, and FABA Lifetime Achievement Award. So we have given awards from 2010 to a lot of dignitaries, couple of them. After getting our award, they became Nobel laureates. So we are a bit proud on it. And this year also we'll be giving awards to a couple of people. So I'll just run through. So this year, like the take-home message that I want to, or what you need to carry later is that we are going to have one meeting. It's called FABA Innovation Summit. So FABA Innovation Summit is going to happen from 3rd August to 18th August. So from first will be from 3rd to 16th, a virtual drug discovery workshop. So every day from morning 8 to 2 o'clock, we will be teaching a lot of concepts about drug discovery. So we'll have speakers, around 40 to 50 speakers from all over the world. They will be teaching you everything about drug discovery. I mean, we can debate whether drug discovery can happen in India or not, whether academia is ready for that or industry are ready for that. But we at least need to get trained, and we are here to train. And the second one is FABA Well Tank, which is going to happen August 17th. It will be at University of Hyderabad. And the third event is the Life Sciences Innovation Cluster Meet. So this event, you all have to come. This will be at University of Hyderabad, where we will have a couple of international consulates, CEOs of pharma companies, directors of central institutes, many of the ecosystem players, they will be there. So this is a drug discovery workshop that I think outside you have flyers for that. And Whale Tank 2, if you are a startup who are looking for more than $1 million investment, so you can connect with me. And the Life Science Innovation Cluster Meet. So in a nutshell, what FABA has done. So we usually don't track numbers, but just we try to track from past 2 to 3 years. So we helped about 20,000 individuals with more than 120 plus workshops and webinars. And we are proud that we have around 25 plus executive council board members along with international chapters. So there are a lot of upcoming programs. You could touch base with me or my team. We'll help you out, and thank you. I would like to request my FABA team to come up, all the EC members. We'll have a picture. Please. Ranjan sir, yeah.
Okay, appreciate.
So on behalf of the 3R Advances Conference and the organizers, we want to give a token of appreciation and a souvenir to FABA, who has been a partner with us from the year one. So thank you, Professor Edena, for supporting us every year. Thank you.
Let me first congratulate Dr. Suresh for the sustained efforts for promoting alternatives to animal testing and making FABA as a partner. And I wish that he conducts these kind of workshops every year in different parts of the country and also in other countries also. And FABA will be a partner. We'll be very happy to be a partner of such kind of events. Thank you.
Next, I would like to invite Mr. Sanjaya from Acuprec Research Labs to give us a brief on their services and capabilities.
To hold like this. Yeah. Keep it inside. Good afternoon, everybody. My name is Sanjaya. I represent Acuprec Research Labs. I just quickly go through the capabilities of Acuprec, what is Acuprec Research Lab is all about. So Acuprec Research Labs Private Limited is a globally reputed contract research organization located in Ahmedabad, and we have two branches. One is in North America and Canada, these two for business requirements. So Acuprec Research Labs Private Limited was founded on August 9, 2012 with the goal of offering high-quality solutions for a range of testing services needed by various multiple sectors. We focused on pharmaceutical, chemical, phytochemical, herbal, food, and medical device industries. And we are more focused about medical device, end-to-end complete services we provide for the medical device industries. The word Acuprec is derived from the 4 letters of accuracy and precision together that we follow in all our testing services which we provide to our clients. Acuprec Research Labs is a contract research organization. We have been providing services with various different areas. So all put together, we have identified around 14 different sectors we serve, and these mainly includes analytical testing of pharmaceuticals and cosmetics, microbiological services, all range of microbiology testings, food, water, and this thing. And the entire range of preclinical services, in vivo and in vitro services we provide, and it includes mainly for agrochemicals, biomedical devices, and all other vertical sectors. And we also provide services in food testing services, dyes, pigment, plastic testing services. We have a separate R&D, and we also support regulatory dossier preparation to our clients. We do the complete range of biocompatibility services of medical devices. In our case, the biocompatibility services, it includes the entire range. It's starting from the biocompatibility studies for all the devices. We also do the risk assessment part also, including the chemical analysis, assessment and identification of the range of leachables, chemicals which will come out from the device. So we also have biotechnological services wherein we carry out a lot of in vitro toxicology studies, all this thing, and we also carry out phytochemical services, stability testing services, formulations and development services. We do provide services for the clients on various different types of formulations, and we do carry out clinical services and IPR management services. These are the various different sectors. And Mr. Mulubhai Kondaria is our managing director, and our mission is to provide ultimate solution to our clients without compromising the quality. And Dr. Reena Gokani is our director and CSO. So we always want to be an extended enterprise to all our clients, these studies what we provide. And Dr. Manish Rachach is a director and CSO. So we want to become, make it the research to the revenue. And our motto is uncompromised quality and prompt services. And he is our Mayur Kondaria, is a director and CMO. So these are the four directors and promoters of this company, Acuprec Research Labs. Coming to the accreditations, accreditations for us is very important. Being a CRO, it is very much important to maintain, get accreditations quickly at the earliest, and to maintain over a period of time. So we in the history, if you look into the accreditations roadmap, so this is the complete landscape of various accreditations we got. Immediately after the inception, 2012, we got a FDCA approval, and followed by this FDCA approval, we started our testing services, followed by that CPCSCA for animal testing services and animal related. FDCA GLP is a Gujarat State GLP certificate. With that, we initially started farming studies, and then followed by the ISO 2017, and we got a NABL 2017. So initially, when we started our testing services, we were only having around 25 different service scopes in the year somewhere 2016. But over a period of time, in a journey of around 6 to 7 years, we have added, now as on today, we have around 700 plus NABL scopes. That includes all the different 14 verticals services which we provide to our clients. We have Ministry of Ayush, DSIR, SEDESCO approval we got for all our medical testing services, FDCA, and recently in the year 2023, we got OECD GLP. With that, our data, what we generate for various different clients is accepted to worldwide for all the OECD countries. In addition to that, we also have CIBRC for agrochemicals. We have registered with them. And recently, we also got ALAC accreditations. And in addition to that, recently, we also revamped the entire quality system of quality systems. And then we also have registered for USFDA, and this is for ADQC sample release testings. So these are the various facility infrastructure. This is the analytical department, wherein we have all the high-end equipments, HPLC, ICP, OES, and many other things are in place. And this is our in vitro laboratory 1 and 2, wherein we have all the basic equipments, activities to perform the basic activities of complete range of genetic toxicology studies, and also the various other biotechnological services which we provide to our clients. Coming to the battery of genotoxic testing of pharmaceuticals, we provide the complete range of battery of genetic toxicology studies. It includes the entire range of assays. So these are the three different spectrum of mutations usually happens, the point mutations, structural, chromosomal, and numerical aberrations. And there are specific assays which are specifically focusing on particular endpoint. So we do cover all the various, the spectrum of mutations, spectrum of genetic events which usually happens. So thereby, so we will be having the complete range of genetic toxicology study services, and most of these services are all in vitro based study services. And this is actually the complete standard battery for genetic toxicology studies. It all started with bacterial reverse mutation study, wherein we use Salmonella, Typhimurium, and E. coli. We have in vitro mammalian chromosomal aberration study, cell gene mutation study, wherein HPRT and thymidine kinase, these are all the enzyme involved in salvage pathway. We are extrapolating those enzymes which are involved in the salvage pathway for the selection of mutants. So thereby we will qualify and quantify the mutations or the genetic events which usually happens in a particular test samples. We also carry out in vitro mammalian cell micronucleus studies, and we also have two animal studies, bone marrow chromosomal aberration study, and erythrocytes micronucleus studies. So in addition to that, we also carry out alkaline comet assay and transgenic rodent somatic germ cell gene mutation assay and mammalian erythrocyte PGA gene mutation assays. These two are under validation right now. Coming to the in vitro diagnostic area, so there is a lot of scope for in vitro diagnostic medical devices. So wherein there are very few labs are involved in testing the performance evaluation of IVDs. So we have recently initiated to take up the IVD testings. Mainly we have identified 2 hemoglobin monitoring systems and HCG pregnancy test cassettes. So wherein we'll be doing it the complete range of performance evaluation. It includes accuracy, precision, repeatability by using repeat studies. The experimentations usually will be conducted for a period of 20 days or 30 days on the basis of CLSI standards. We use 3 different CLSI standards. It includes clinical laboratory standard institute documents, for the precision of quantitative measurements, and for the testing of clinical chemistry, and for evaluation of detection capability for clinical laboratory measurements procedures approved by this thing. And in addition to that, we also use SEDESCO guideline document for carrying out the performance evaluation of various IVDs. So coming to the in vitro assays for cosmetic testings, so we do have cosmetic testing services as an alternative to animal testing for safety testing of cosmetic products and cosmetic ingredients. There are 4 different categories we have identified, and we do provide the services under the genetic toxicology studies, skin sensitization study, irritation study, and eye irritation study. And in that, we will be performing all these studies under UV visual spectrum, and including photo irritation study and photo sensitization studies we perform, and also identify in the process the endocrine properties, dermal absorption studies also. And thereby, by conducting all these alternate models, we will find out the margin of safety study for all the cosmetic products. In addition to the in vitro models, we also support physical chemical studies for most of the cosmetics and cosmetic ingredients. It includes the physical characteristics of a cosmetic: pH, viscosity, density, flash point, mass. Active substance identification and quantification also we do it, mainly antioxidants, aluminum salts, preservatives, etc. Restricted substances, if at all, if anything is there in the cosmetic and their ingredients, and the possible contaminants, if it is there. So that also we will be identifying using the spectroscopy and chromatography. Various analytical techniques are available with us. Yeah, coming to the in vitro, as such, the whole range of animal testing alternatives, we have identified the 5 different groups, and wherein we carry out all the in vitro alternate models. So in earlier days, we were performing all these studies in the animal models. Mostly rabbit was one of the recommended models. So right now, with the adoption of this, so we will be carrying out most of these in vitro alternate testing models, and thereby it is involved in the testing purpose, mainly for the labeling purpose of various different chemicals, REACH and agrochemicals we carry out. Most of the time we will be using reconstructed human epidermis tissues, which we procure from different vendors. We will be performing the assay and submit the results. In vivo skin irritation construction, this also using the reconstructed human epidermis. Coming to the in vivo skin sensitization study, we also carry out HPLC-based in chemico skin sensitization method. Here, no animal or human cells are used. We evaluate the simple binding of a chemical to a receptor. In vitro skin sensitization, there is another one more assay is about genomic allergen rapid detection for the assessment of skin sensitizers. It is also called as guard skin study. Right now, we are in the initial stage, and Sanzagen in Sweden is the only organization where they are having monopoly for this type of studies. And we are having collaborations with them, and through them, through the Sanzagen, we provide these services to our clients. Coming to the eye irritation and corrosions test, so these are the various test guidelines recommended by OECD, which we also identified, and we have done our complete full range of validations. And with that, we also started providing all these things. Bovine corneal opacity and permeability studies, isolated chicken eye test methods, and short-term studies, and reconstructed human epithelium cells. So these are all the various models. In addition to that, we also perform dermal absorption study and phototoxicity studies using 3T3 cell lines. So one of the biggest challenge for us in Indian CRO to carry out all these in vitro testing methodologies is the cost of these tissues, reconstructed tissues, which we procure from outside, is exorbitant, and we will not be, most of the charges what we make, so it will goes to the developers of these in vitro tissues. And coming to the biological derived pharmaceuticals, we have also identified various biological derived pharmaceuticals. There are only few labs, and most of the labs are all the government labs. They provide testing services for the biological derived pharmaceuticals. And we recently started providing this service. We have initially started with providing heparin sodium injection, enoxaparin, filgrastim injection, and streptokinase. And for all these things, based on IP and EPA, we will be providing potency studies. Filgrastim, we do this cell line based assay, wherein we will be performing the potency of a Filgrastim strip with a reference item in response to that using various statistical models. And in addition to that, we also carry out a lot of ELISA methods. Using a ELISA method, we will be carrying out bovine spongiform encephalopathy and TSC, transmissible spongiform encephalopathy in various test samples by ELISA methods. And for most of the food industries, there are many allergens is major concern. So these allergens, we have identified around 16 or 17 allergens. So we cashew nut, hazelnut, nut, walnut, pecan, pesticans, almond. So these are the various allergens, possible allergens in the food samples, which we also routinely perform using the ELISA method. In addition to that, microbial contamination studies we perform, mainly the DNase, RNase, and protease in the various test samples. And our clients are present in around 60 plus geographies across the globe. So this is about the services what we provide to our clients. So if I request if any information, if any further this thing is required, please contact us. So we will provide the services. And we are open for any sort of collaborations with academia, with any other industries for new ideas, development of new test methods, development of new alternate methods. We do provide services. We do provide our facility, our expertise, and our client base. So that would also help in getting that. And thank you very much. Yeah. Yes, we carry out GLP studies. Most of the genetic toxicology studies and all other toxicology biocompatibility studies are all performed as per the OECD GLP. 28-day study, in vivo toxicity studies also we carry out, 90-day studies and all. Biocompatibility studies includes the complete range of studies. It includes all the animal toxicity studies as per the ISO. Yes. Yes.
You offer that?
We do provide IND enabling studies also.
And that can be filed with US FDA and CDSCO both?
Yes, because it's we have OECD GLP, and OECD GLP is accepted worldwide for all the OECD countries.
Yeah. You're located in Hyderabad or?
We're located in Ahmedabad, sir.
Ahmedabad. Okay. Yeah. Okay. Thank you.
Pardon, sir?
Have you been audited by US FDA?
US FDA for ADQC sample release, we have registered, and we are waiting for their inspection.
Yeah, the reason is following up with his question, if there's a preclinical toxicology studies are done and enabling studies are done for a US client, OECD GLP will not be accepted. US FDA will come and audit it. They don't need GLP certification.
No. Usually, whatever OECD GLP certificate, whatever studies which we perform as per the OECD GLP, it has been accepted all over actually, all the world. And in fact, even US FDA will come and inspect only in case if there is any triggers, if there is any particular product triggers, if there are any queries comes, then through the NGCMA, they will come and audit our facility.
We'll talk later.
Okay.
Thanks.
Thank you, sir.
Yeah, thank you, Dr. Sanjay, for giving us a glimpse of the services provided at AcuPac Research Labs. Now, I would like to invite Dr. Suresh Pusala to present a token of appreciation to Dr. Sanjay. Next, I would like to welcome Dr. Subramaniam Vangala. Dr. Subramaniam did his PhD in biochemistry from Memorial University of Newfoundland and a postdoc in molecular toxicology from the University of Colorado and the University of California. Dr. Subramaniam is the CEO and founder of Reagent Biosciences Private Limited, Bengaluru, and co-founder of Candor Life Sciences, USA. Dr. Subramaniam is a seasoned leader in the field with 30 years of experience in drug discovery and regulatory preclinical development and has worked at Pfizer, Purdue Pharma, J&J, Sai Advenus Eurofins. Dr. Subramaniam is an expert in DMPK, clinical pharmacology and toxicokinetics and has led successful IND programs, filed multiple INDs and developed 6 marketed molecules. Dr. Subramaniam is also an experienced investigator and molecular toxicologist with expertise in environmental toxicology and risk assessment. Currently, one of his main research focuses is 3D microphysiological systems. Please give a warm welcome to Dr. Subramaniam Vangala to discuss why drugs fail during clinical trials, case examples in oncology, and how can 3D models improve success rates.
Can everybody hear me? Great. It's been a long day. I was almost falling asleep right there because of the food I ate, very good food. Thank you, Suresh, and all the organizers to invite me here. This is my real passion for the last 15 to 20 years after working for pharma companies for 20 years. The reason is that, I come from in vitro background. Uday knows, I've been doing in vitro toxicology, cancer drug discovery, not drug discovery, but cancer risk assessment for environmental pollutants, etc., from 1981 onwards. You've seen me working in the lab, cooking up microsomes and NADPH, etc., for a long time. But you have to transition. So I'm a molecular toxicologist background. When you join into the pharmaceutical industry, you have to transition. So like everybody else, I've also started doing animal trials. Start with mouse, rat, dog, and monkey maybe, if needed, and do the pharmacokinetics in all these species, and do the allometric scaling, and predict what is the human dose. How do you predict the human dose? That should be a safe dose. It's not be any dose that you can just go and give it to the humans. It must be reasonably safe. It may not be efficacious dose, but it has to be reasonably safe dose. And that's what FDA wants you to do. When you propose an IND dose to the clinic, it has to be a safe dose. That is first consideration. You cannot just go to any dose. Yeah, my efficacious dose doesn't match here, doesn't count there. Because you don't want to kill patients in the, not patients, actually, in first-in-human trials or in healthy volunteers. That's another concern I have many times. So I've been learning. You keep learning all life. You're a student. As soon as you come from one place to another place, you keep learning new things. So there's a huge disconnect. We go, I probably managed, you say, out of 25 preclinical studies, IND-enabling programs a year. I worked for 20 years. At least 4 are nominated for the phase 1 FIH trials. They could be US IND or European IND also. European IND is not as extensive as US IND. So you take them, four molecules every year, and many of them I'm a project leader too. I manage the entire preclinical development program. But there was a huge disconnect. You go there and you see some things that you didn't see in the preclinical development. We started off with taking, okay, let's do a species comparison of the microsomal metabolism. And we see huge differences in metabolite profiles. That actually triggered FDA guidance document. Hey, your in vitro metabolism shows that your human metabolite is more than a rat metabolite. They say that now you have a metabolite that is more exposed in rat, could be more exposed in human than in rat. That's a trigger, because they say that you have to do another IND program for your metabolite, toxicology program for your metabolite. There's a metabolite safety assessment guidance is also there, FDA. So you need to be extremely careful before you go to the FDA, because once you have submitted to IND to the FDA, they'll come up with the questions you don't even have answers for. So you have to be very, very carefully package your IND package and then submit it to the FDA. So many times I was turned down, saying that no, we're not going to do that, because FDA tomorrow is going to ask me more questions about it, and they'll put the program in clinical hold. And this is the regulatory aspects that we have to deal with. My mentor at that time was Fred Tonelli, and he said, Manny, if the regulatory don't ask for it, don't do it. This is how the mindsets work. So, but we have come a long way. So because we are failing more in the clinic, 90% are failures. So you all know about it, that you have to have a benefit versus risk. Every drug carries some kind of safety issues, and you have to have benefit versus risk is a great idea. Here is the overall phase 1 through FDA approvals based on therapeutic area in 2016 to 2000. This combined with all therapeutic areas. You can see oncology is the least one, 5.1% of all. If you take all the drugs, if you take hematology indications, they are the highest approved, 26%. So it's also therapeutic area involved where more approvals are. Oncology is the toughest area to get more approvals on. And if you look at the 2013 to 2020 oncology drug development clinical success rates, typically, as I said, in preclinical, in animal models, you do fail. I mean, as I said, 15 to 25% success rate there. But leave that out. Once you come to the phase 1, your clinical PK matches the predicted PK. All your preclinical data is thrown out. You're only looking at the clinical data now. Because the clinical data is more important for FDA than the preclinical data. So you're designing your phase 2 trials, phase 3 trials, based on the phase 1 data, not on the preclinical data. You may use the safety margins a little bit from the animal data, but that's what happens. So the molecule that enters phase 1 is the molecule that enters the market. So not the one that enters the preclinical animal model data. Okay? Then if you look at from phase 1, about more than 50% are failed to go to the phase 2. So what happens in phase 1? 90% of them are PK failures. The PK doesn't match from what you predicted from the animal models, period. Whether you use humanized animals or used rat, dog, monkey, and standard animal models, doesn't match. And then you go to the phase 3 trials. There's more risk there, actually. Phase 2, you have a reasonable efficacy there, right? It's already some value there, efficacy. But when you go to the phase 3, you have N increases. Means more patients you're recruiting in the phase 3 trials. Here the statistics doesn't match, and majority of the phase 3 trials fail. And this is where you have a problem that, hey, my drug worked in phase 2 trials. No, it didn't work because phase 3 trials is failing here. So your N is, so in this case, you have to, what do you call, bring the patients down to a level that you have to use some biomarkers like genetics or something to strategize the patient recruitment here. Okay, so phase 3 trials, now more and more people are using genetic markers to recruit patients so that they'll not fail, they'll succeed. And then you file a submit to FDA and FDA approval. Finally, 5.3. As I said, 5.1 in oncology trials, it's about 5.3. Yes, it's the same ratio that you're getting now also. So what are the factors that influence? As I said, PK in the phase 1 trials, but phase 2 and phase 3, it's efficacy. That's our toxicity that is a major issue. Let's look at the first thing first, right? Why are phase 1 50%, more than 50% are failing? Your PK is not matching. So there's some problem with the animal trials. You're doing the way you're designing or something, it's a problem. There you're not designing these animal trials probably very well. So your calculations or your mathematical, statistical programs that do the allometric scaling are faulty. I mean, they don't predict the right thing. So you take any therapeutic area, right? If it is an oral drug, you have to have a drug metabolism and transport in the liver or even in intestine, and a circulation, protein binding, and metabolites. As I said, if you have more metabolites in human than in animal, then you have triggered FDA alert. And then many drugs are cleared renally, and of course intestinal excretion too. Many drugs are excreted intestinally through the enterohepatic recirculation. They just continuously recirculate in the body. So my journey, only oncology programs, I can say these are the ones that I've been supporting for the last, maybe since 1993, 30 years. The first one I was exposed to is the doxorubicin. Doxorubicin is, it's a chemotherapeutic drug, and it's a quinone. It's actually intercalates the DNA, and is the reason why many people think that causes the anti-cancer activity. But the doxorubicin usually is more and more resistant, became resistant. And people initially thought it was P-glycoprotein, one of the efflux protein. It just as soon as the drug enters the cancer cell, it throws it out. And that is the reason. So I worked on a lot of cell line experiments in those days in vitro, trying to see which cell line doesn't have the P-glycoprotein, is that more efficacious than that. Then the KCO2 and MDCK came along, which are perfect models for P-glycoprotein. But even today, we don't have a doxorubicin resistance drug, because the reason is because it's not just P-glycoprotein alone is responsible for resistance. Now BCRP, breast cancer resistant protein, some other MRPs, many other transporters. There's something called now, people are talking about FAB. So there's a multiple mechanisms of resistance, not a single mechanism of resistance. So it's a redundancy. What happens is the cancer cell trying to find out ways to get around the resistance. Every time you have this problem. So I'll talk to a little bit about PDGF kinase, CDK1 kinase. As we were talking in the earlier talk, this was a drug that was spent for 2 years at J&J. It should not be spent. It's nobody's fault. The drug was highly bioavailable in drug discovery. They had an amorphous drug. Now they pushed it in the preclinical development. I actually was there, one of the guys who approved that drug to take into the preclinical development, IND-enabling programs. Now we have some problem. We were doing toxicology studies. Toxicokinetic data doesn't match the pharmacokinetic data we got from the animal models in the drug discovery. Then we back and looked at it. What it is the CMC, process scale-up groups, generate the crystalline polymorphs. These crystalline polymorphs are more stable. They get, even in DMSO, they turn to the stable form, and they're very, very less soluble than the amorphous compound. You cannot develop the amorphous form in the clinic. You have to have a stable polymorph. So we've done, in my group, actually spent about 100 formulation trials in dogs. We had a formulation company giving us formulations with screening, left and right. And all the 100 formulations, none of them increased the bioavailability more than 15%. So finally we parked it. So CDK1 kinase, that was the story. And then the PDGF kinase, I'll talk a little bit data about it. And then also I have a P38 kinase. I'm not putting any data there because we don't have enough time. And I worked on other targets too, but I was not part of the clinical programs for it. And ginger, today nutraceuticals are becoming very popular. And I had a collaboration with Georgia, Dr. Ritu Aneja. They showed some efficacy in the animal models for ginger, I'll give you a little bit data on that. But we had some very interesting. But here, what we try to do is ginger reverse pharmacokinetics, because there was no pharmacokinetic data available for ginger. People, yeah, ginger works. That's all we know. But is there how it works? What is the mechanism of action? Nobody has a clue about it. So that I'll talk about it. We just in January had a publication on zinc oxide nanoparticles. Zinc oxide is a gas molecule, and it is actually used in a lot of medical devices. I had a program with MS Ramaya Biotechnology Institute in Bangalore. So we got a couple of interns from there. And they had zinc nanoparticles. They're synthesizing and characterizing them. And we tested in 4 different cell lines and looked at the cytotoxicity. Wonderful data. So I'm thinking maybe zinc oxide should be promoted for anti-cancer status. And I'm trying to look at other ways of doing more studies on zinc oxide. Right now, I don't have any money for it. But so I don't want to go through this. So Uday can explain it better than me. This is the pharmacology of PDGF kinase activation and how that's involved in the cancer reaction and everything. Same thing here. So coming to metabolism, what we found was it was metabolized by glucose, glucuronide B and C. It's an amine, okay, aromatic amine, so it forms a glucuronide. There are 2 glucuronides, and you can see that there are species difference. Rat liver microsomes versus monkey liver microsomes or human liver. There's a different profile. Basically, there's a, but it's heavily glucuronidated. That suggested to me that most glucuronides, even take the estradiol or any other bilirubin, etc., all the glucuronides are excreted into the intestine via biliary excretion pathway. It's and then some part of it gets deglucuronidated and goes back into the portal circulation, comes back into the intestine. So it's circulating between the liver and intestine and portal circulation. It's not going into the tissues. Whatever goes into the central circulation system, systemic circulation system, is very minimal. So this is where the problem came in. And we also went into the clinical trial. We had the same problem. We're seeing more glucuronide metabolite there. So I got an idea here. So if you look at the enterohepatic glucuronidation, this is what the pathway is. That glucuronides or the parent drug are excreted more in the feces than in the urine. In this case, we have seen a radiolabeled compound getting excreted about 70% in the feces and 30% in the urine. So there's definitely extensive enterohepatic recirculation. So I got an idea there. I said, let's see actually what happens if it is a glucuronidation is the major problem here. How do we eliminate it? So I got just an idea. This is not clinically applicable. So what we did was I told Gary, can we add glucuronidase into the animal along with our drug while orally giving it? Gary got excited about the idea. He took forward it, and I had no time to supervise him, but he really did a very good job. I left J&J, but in 2012, he sent me a publication that we published it, the data. Very good data, actually. So what happened was, so we did beta-glucuronidase. I don't want to describe the entire protocol. You can find it in the publication if you really need more details. And we also looked at the bioavailabilities of the parent and the glucuronide with the glucose. So there's another slide here. Yeah. So glucuronide administration not only increased the systemic bioavailability of the parent compound, it also increased the systemic bioavailability of the glucuronides. We didn't have enough money from J&J to pursue what more is happening, but the bioavailability is definitely increased. To add to the story, that glu-B metabolite is also pharmacologically active. So we actually increase the parent and also the metabolite bioavailabilities by this method. But this is clinically not applicable, practically applicable model. But we got a good data that glucuronidation is a major problem. So Dawn of the Zombies, I'm going to talk about it because we need more in vitro models that can predict these kind of things, especially human models. So at the same time, our Belgium colleagues, Peter Anhert, he came from academia, and I met him, and he said, hey, Manny, I can do a sandwich culture of hepatocytes. Sandwich, what you do is matrigel was typically used to load the cells. So what he does is that he puts a matrigel, loads the cells, and puts another coat of matrigel. So these are sandwiched. Now what happens is within 3 to 4 days, you form biliary canaliculi and bile secretion. Very beautiful model. But there are lots of practical problems even with these models. But this is what he developed, and I used it for my product here. But anyway, they also validated quite a bit of transporter expressions, because the biliary excretion models also need transporters that are expressed on the canalicular side as well as on the plasma membrane side of the hepatocyte that is getting exposed. So quite a few transporters he just validated, showing that there is an increased expression in humans as well as in rat and so there's a very good clearance. This product that he used, which is a model compound, you could see there's a very good linear intrinsic clearances in vitro in the hepatocytes versus the in vivo clearance that we got a very good, I mean in sandwich model and in vivo clearance in the animal. Now, the fun starts here. These models you cannot just use for one day. Because when you do a toxicology study, you need at least a month, 28-day toxicology study. So you need to keep that for 28 days going, right? Now what happens is the cytochrome P450s, they have highly expressed on the day 1, day 0, when you isolate the hepatocytes. But the expressions go down over the 4 to 7 days. It's a typical problem. They also turn into a fibroblast in more cases that you keep them for longer. They differentiate. Then we add the inducers, cytochrome P50 inducers. There are quite a few, so 3A4, 1A2, etc. You add them, and you can bump up the. But that's not practical in clinical situation. You cannot just keep adding inducers to improve the cytochrome P50 expression. And the transport expressions are actually increasing from day 1 to day 7. So what you have is you don't have a co-expression model ideally to work with both cytochrome P450 and transporters expressed, because majority of drugs that we use today are substrates for both cytochrome P450 and transporters. So there's a problem that we don't have a real good model yet, and people are still working. I just looked at a review article in 2018. They're still struggling for it, how to validate this model. So I had a collaboration with a company called Hurel at the same time. So we wanted to get a better models into this. Hurel came up with a microfluidic chip with a prototype model, and what we decided, we actually, J&J funded it, and I actually worked with them very closely at the time, was we had 4 compartments in the microfluidic chip, and where you have a target tissue, like lung, and adipose tissue, because it takes out all the lipid molecule, and a liver, and in and out, you take this. This is a very preliminary prototype model, where what I used here is rat hepatocytes, not rat adipose tissue, not even human, just to validate the model to a level that we could use it. And if you look at we had a Petri dish, and hepatocytes were plated, and we looked at all the cytochrome P450 enzyme activities. Now we took the hepatocytes for 3 to 4 days. We cultured them in the 3D with that model, and we looked at in the flow system. Look at the activities. This is what I need, right? You need a flow system not a static system that will improve the expression of these cytochromes P450s. Even biliary sandwich model is a static model. It's not a flow system model. So you do need, so how do you combine these models? You have static models, have very good, giving you at one stage, giving one good data, and flow systems are giving you a different good data also. Now, a lot of people are working on these models today, and we also did some toxicology experiments, but I don't want to go into this, but it worked very well in that system. So I'll just touch a little bit on ginger. Ginger has been proposed to be anti-cancer for many of the cancers, but I think it's more for GI cancers than any other cancers. If you look at the major constituents of ginger, there are at least 6 we have identified gingerols. And in fact, Ritu Aneja, a professor from the Georgia University, actually did some anti-cancer studies with the ginger. And she did some studies show that actually ginger phenolics do have anti-cancer. But she doesn't know which ones are going to be the more active and which ones are, because you don't get consistent data with the same mixture that you use. So she wanted to do pharmacokinetics. I call it reverse pharmacokinetics because we want to get, for all these metabolites of ginger, we did quite a bit of studies. We published about 4 or 5 papers on it. You can see there's variability, a lot of variability in ginger phenolics, and they also get glucuronidated. And so we did quite a bit of in vitro metabolism, in vitro glucuronidation. And, but I'm just showing the data just to show that. We also looked at the various tissue distributions of these ginger phenolics with LC-MS approaches. This is all done with Edwina's that when I was there, we did all this work there with Georgia State University. Interesting data, but still, I mean, she didn't get an NIH grant, and we don't have grants. So this is where we had to stop the programs. But what I think is that going into the future, I want to put them into the sandwich culture models or other in vitro 3D models and see if we can come up with, Uday is very much interested in nutraceuticals. You want to take this kind of nutraceutical programs into a clinical program, viable clinical program. So, so this is the hypothesis that, it's only a hypothesis that you actually have intrahepatic recirculation, but these metabolites keep coming back into the circulation, and that's what the anti-cancer activity is. But which are the right metabolites, we still don't know yet. So now coming back to reagent, right? So I met Uday in 2016, right? And I said, hey, Uday, we need to develop these 3D models. He was excited. And what we want to do is that we want to develop efficacy models rather than the toxicity model, because quite a few other people are already doing toxicity models. We talk about DILI model and other models. Emulate has done quite a bit of DILI validation already. And now Inspero is also trying to do some DILI validation. So let's move away from it and let's do the models that are not there existing. I like in Uday's idea that you do efficacy models because that is where most drugs are failing in phase 2 and phase 3 trials. You need models for them too. So this is 3D bioprinting for process at reagent. And this is one of the anti-cancer thing that what we did was we got a 3D structure where we have a 3D bioprinting with intestinal cells, hepatic cells, and target cells. We used lung cancer cells here. And we got very good data using the cell morphology, drug activity, drug metabolism, cell viability in a 12 to 24 well platform. So format. So what it means is that this is a viable model. You can fine-tune these models the way you want to get the right data for you. So the question now comes is that how do you fine-tune these different models that you have? This is the data that's generated by Uday and his people. So the future, it's not going to happen in the very near future, but everybody wants to get a pharmacokinetics, putting all these organs on a chip. I wouldn't call them organs on a chip, but as a misnomer, what we are doing is a 3D cell culture models. So I would say that organoids are also not organs, because you don't have the same cell population that there is in organ. The other problem people use is organ on a chip. Organs do not absorb drugs. Organs absorb drugs through the blood vessels. So you don't have blood vessels here. How are you going to organ and organoids are going to penetrate to every corner of the liver or organ? So people should not say that I'm developing an organ on a chip for drug discovery. Organ on a chip may be for transplantation purposes, not for drug discovery, because it's a very misleading in terms of developing an organ on a chip for drug discovery. So my vision is that we want to develop a PBPK model with integrating the physiology, other in vitro models, multi-dimensional human in vitro models, etc., ADME, toxicology, combined models. You still have to do the traditional in vitro models. You cannot say that I don't want to do them anymore. This is a microphysiology. But no, I cannot see how the cytochrome P50 metabolizes in an organoid. I still need microsomes to do that. I need to get that data. So great with his help, we developed all these models here in the vascular lung, cancer in a dish, diabetes in a dish, leaky gut, liver. So it gives you a different perspective. We are looking at pharmacokinetics and toxicology, but this guy comes in, let's also look at efficacy models. So these efficacy models, what he's thinking about, have to mimic the human pathophysiology that actually a disease has. For example, vascular lung was beautiful model. He developed it for COVID-19. COVID-19, 3 to 5% patients died of thrombosis. You look at any animal model out there so far, there is no thrombosis. So the thrombosis is the ultimate point leading to the death. There's no model animal model out there. Oh, yeah, you have hamster ACE2 expressed models, but they do not really serve the purpose. So he created showing that cytokine storm, inflammation, and thrombosis, all three can be measured in the vascular lung model. That is the kind of model mimicking the human pathophysiology is what we require. So, great, Uday, thank you. I'm out of words right now. I mean, because we have come a long way, and we need to progress this to a different level. And this is where we need to scale up, and we need to get more money and do more models. There are people coming to me asking for osteoarthritis model. People are coming to me and asking for blood-brain barrier model. We don't have money to develop those models. Unless somebody pays for it, we can't do that. So, guys, spread the word around. We are doing an excellent work. Viraj, you should cite these papers in your LinkedIn, because I've seen your LinkedIn thing. You haven't even cited a single paper from reagent biosciences that came in. You should do that. This is India. We are doing this work in India, in your neighborhood, right? Yeah. Right? Okay, thank you. So I have Uday and of course Saranya is here, Arpita is here. They all work with Uday to develop these models. Arpita, are you here? Saranya? Yeah, there she is. So we are a great team who are very enthusiastic, who wants to really learn things and develop things, and we are blessed. Thank you, and any questions? I'm ready to help.
Yeah, so the point that you raised, definitely I will revise it and include these papers. But my focus was mainly on organ on chips based studies. So that's why now I'm planning to make a 3D bioprinting spheroids, organoids, all kind of studies that has been done. That's why I didn't include, but thank you for pointing that out. Yeah.
Okay, sure, no problem.
Definitely.
So Manny and I worked together at Reagene for about 4 or 5 years, and his vision now is to develop it as a CRO. My training is not in the CRO business, so I'm kind of taking the back seat. But one thing I have to tell you, he's probably one of the finest PK/PD toxicology guy in India now. Bar none, really. I've met a lot of people, but this guy, it's all up here, so you just need to pick it. He's got it all. So anybody who needs help with PK/PD, toxicology, toxicokinetics, he's the guy to go to. So thanks, yeah.
Thank you.
Thanks, Manny.
Hello. Thank you, Dr. Subramaniam, for that insightful presentation with informative case studies in oncology, discussing why drugs fail in clinical trials, and your journey supporting oncology R&D since the last 3 decades. Now, I would like to invite Dr. Srinivas Maddi and Sridhar Voleti to present the token of appreciation to Dr. Subramaniam Vangala.
Many come this way. Many? Yeah. Thank you. Give the certificate. You give the certificate. Yeah, this one. Huh?
So we are very active.
Yeah.
We enjoyed your talk.
Thank you.
Excellent.
For the next talk, I would like to invite Dr. Manu Smriti Singh. Dr. Singh obtained her doctorate from the University of Bonn, Germany, and conducted postdoctoral research at the Tel Aviv University, Israel. Dr. Singh was a visiting fellow at Encore University Hospital, Dresden, Germany. Specializing in the 3D tumoroid technique, she received the power grant from the SCRD DST in 2022. Dr. Singh briefly served as a visiting professor at the Hebrew University of Jerusalem, Israel in 2023. Currently, Dr. Singh holds the position of Associate Professor at the Center for Life Sciences, Mahindra University, Hyderabad, India. Her research area includes cancer nanomedicine and photodynamic therapy targeting non-cancerous cells in tumor microenvironment. Please give a warm welcome to Dr. Manu Smriti Singh to talk about the 3D tumoroid model as a predictive platform for preclinical drug nanomedicine screening and personalized medicine.
Good afternoon, everyone. I hope that we are up a little bit. I know it's past lunch and a long day, but we still have a panel discussion and very nice talk coming up from Dr. Charu. So please stay afloat. I'll try to be as terse as possible. So my talk titles 3D model as a predictive platform for preclinical drug, nanomedicine screening, and personalized medicine. So this is a work, it's in continuation, and also most part of it is published, and I did it in Tel Aviv University with my postdoctoral supervisor, Professor Dan Peyer. So I'll talk a little bit about ovarian cancer because those are the 3D tumoroids I've developed before, and I've tested my nanomedicine there. And then composition, because this is a bit of nanoparticles, so probably I'll tell you briefly about the nanoparticle composition, the cargo that is there for anti-tumor activity. In vitro activity, 2D versus 3D model. In vivo model, so my talk would be, let's say, a bridge between 2D and 3D, as well as connecting it to the in vivo model, because that's the glimpse of it we can see in 3D, and that's why 3D is very important. And the conclusion. So just touching upon ovarian cancer a bit. So I've been working on ovarian cancer for a bit now. So
usually the signs and symptoms are very mixed. The patients can't really figure out that they have ovarian cancer unless and until the women are visiting for a checkup where they get ultrasound, and by chance they can get detected in the first stage. Most of the detection, 70% of the cases happens at stage 3, which is quite late. So there are general symptoms, very general, so no precursor lesion, no useful screening, bloating, cramps, etc. So most women come with swollen abdomen, not all, but most of it, and that is represented here in a Nature Review graphics. So this swollen belly basically has the stage 2 cancer cells which are shedding from the ovary. Ovary are hanging in the abdomen. These are very small, so they can't accommodate too much tumor. So from stage 2 onwards, the cells start shedding as clusters. These clusters, if you see under the microscope, this is how it looks like. So they look a bit like 3D tumoroids, what we are growing. So this was actually initially the prompt to be testing our drugs or nanomedicine on the 3D tumors, actually. So we were working closely with the hospital with the head of gyneko-oncology. So the amount of fluid can go up to 5 liters. They are in extreme pain. So only after the doctors remove the fluid, then only they can go for either chemotherapy or surgical removal. So here it is shown. So as I said, actually I should have. So the ovaries are really small, so they start shedding, and this is your 3D clusters of cells. They may have fibroblasts as well. And then en masse as a 3D tumor, they go and start metastasizing and start sticking to abdominal area. The most prominent one is omentum, which is a fat pad behind all of us, which supports all the cushions, the background organs actually. So after this, so basically I'll talk a little bit about nanoparticles. So we are all familiar with RNA therapeutics at the moment. So the 2006 Nobel Prize was given for siRNA discovery. And now we know RNA more for the mRNA therapeutics, the vaccines that got approved on one hand by Pfizer, BioNTech in Europe, and Moderna in US. So our group at the time now, they are working on mRNA, but at the time I joined, we were working on siRNA. And very few labs at the time were working on siRNA therapeutics. So the advantage of using RNA therapeutics, as we know, are that they do not integrate in the genome, that they do not enter the nucleus, and it's a transient and rapid effect. Whether we want to do therapy using mRNA or siRNA, it's a brief intervention, and then the therapeutic effect can be observed. So just jogging behind to this molecular biology. So basically, if you have an mRNA, it can result in protein. So either if you have a lack of gene, you can provide mRNA, as in the case of COVID vaccine, they were making spike proteins using mRNA as a vaccine. Or in my case, I used siRNA to basically block an overexpressed mRNA, which was causing the cells to proliferate, causing cancerous growth. So these are very small nucleotides, 18 to 21 nucleotide long. They destroy messenger RNA, disrupting protein formation. And we were using lipid nanoparticles, which are the same nanoparticles for delivering the messenger RNA in the vaccine. So I used 2 of the siRNAs. One was polo-like kinase 1. This is already in stage 2 clinical trial for treating liver cancer. We've got very good results, the company, Alnylam, and a couple of siRNA therapeutics have been approved for other liver conditions. And besides that, so this I used because this was already approved. So one of the siRNA I used was the one which was already clinically validated. And another was SIEIF3, which was found out by one of my previous lab colleague. So this is basically one of the eukaryotic translation initiation factor 3C. So basically, when the translation is happening, you can block the overall, because we know that in cancer there is an increase in global translation, global protein production. So the idea was to basically target 2 different therapeutic pathways to exhaust the cancer cell so that it doesn't have time to repair or fix the gene and fix the problems and they die off. And using nanoparticle would deliver both the genes in the same cell. So we'll be sure that the nanoparticle, if it is delivering, it would have delivered both the siRNAs. And the research has shown that even as many as 15 to 20 siRNA molecules are enough to knock down the gene expression. So we developed the lipid nanoparticle using microfluidic machine, which has a history of its own actually. So another thing that we did, we coated the nanoparticles for targeting. So we coated them with hyaluronic acid. Why hyaluronic acid? Because it's a natural ligand for the CD44 receptor, which are overexpressed in cancer cells. So CD44 are expressed in normal cell, but way more expressed in cancer cell. So this is a natural ligand, and the idea was that if we are delivering, it would go more and target more to the cancer cells present in the peritoneal cavity. So what we did was basically, this is your cell, it has the CD44 receptors, and we coated the nanoparticle using hyaluronic acid, and inside the nanoparticle are your two siRNAs, siPLK1 and siEIF3C. So this we called as the targeted lipid nanoparticles, or TLNP. The uncoated ones were called ULNPs. So this is the overall design of the nanoparticles. So I'm sorry that we shifted completely from the models, but we will come there. So then we characterized, so you can see here, these are very nice nanoparticles without HA coating, and then we did with TEM for the coated ones, and you can see a flower-like appearance. So this proved that it had got the covering of hyaluronic acid, and also the size increased a little bit between uncoated and coated. So you can see slightly around 10 nanometer size increase. So originally if they were 60, now they became 70 nanometers. So these are all part of the characterization of the nanoparticles. And because hyaluronic acid is negatively charged, the charge of the nanoparticles slightly decreased. Sorry, it's positively charged. So basically these were our target siRNAs. So we always have control. So the main therapeutic molecule nanoparticle was the one with both the combination, but every time we all are aware of this, that we have to do screening, we have to take the each and every control. So we had EIF3C alone siRNA in one nanoparticle, PLK1 alone in one nanoparticle, and also we had to take scrambled siRNA. So when we are testing drugs, screening drugs or nanomedicine, we know that there are too many controls. So we have to take into account all those things. So the number of groups increases, whether we are doing an in vitro study or an animal study. So, and on top of that, I also had coated. So there was one set which was uncoated and another set which was coated. So total, I had 9 batches which I had to test from in vitro to in vivo. And then I had one more set where we didn't put any siRNA, just Cy5 to visualize. It was basically fluorescently labeled nanoparticles to track this, the presence of nanoparticles in cells. So we started with a basic, so this is a basic 2D cell culture, everyone is familiar. The walls are stained with wheat germ, agglutinin, green in color, DAPI's nucleus, and Cy5 are your nanoparticles. So I'm not sure if it's visible, but this is the uncoated and this is the HA coated group. And there was slight more penetration or uptake with the coated one because there was more CD44, but not really significant. So you do see in cytoplasm some of the red dots if you look closely. So these are ovarian cancer cells, OVCA rate. So then I moved to 3D culture. So because I was setting it up, as I said, it is very similar to how the ovarian cancer is disseminating. So these were OVCA rate cells, but they were M-Cherry labeled. So we were doing some GFP or M-Cherry labeling of the cells so that they're easy to track in vitro as well as in vivo. In vitro in the instrument InQSight, which is now being sold by Sartorius. So this was so basically what we do in animal studies, we inject the cells in the mice, let the tumor grow, and then we start giving therapeutics on different time points. I followed exactly the same pattern, but this was in a dish. So we had these 3D tumors and I was trying to monitor the growth kinetics. So on day 0, I seeded the cells and gave them 3 to 4 days to form the tumoroids or spheroids of these ovarian cancer. And then because siRNA nanoparticles, siRNA usually takes some time to inhibit or knock down the gene. So it takes roughly 3 to 5 days to see a very good functional effect or death of cells. So I gave 3 treatments on day 4, 8, and 10, and I monitored them up till 12 days. And every day, I think alternate day, I took images in the instrument InQSight, and it also has a fluorescence, relative measurement of fluorescence. So you can quantify based on your treatment. So if there is more fluorescence, that means it's a control group that it's growing, and if you have a good knockdown effect, if you have a cell death, you see less fluorescence. So I monitored the growth kinetics, and this is what I see. So this is how. So this is, if we all, those who have seen papers with tumor growth kinetics, we measure it with vernier calipers, the round tumors on the mice, right? So similarly, you see here growth, tumor growth kinetics, and as I said, I had 9 groups, 1 control and 4 for each of nanoparticle, 1 set which was uncoated and 1 was coated with HA. Okay. In all these groups, what I saw was, for example, if I'm seeing this scrambled, of course, we didn't see anything. So this is the size, 800 micron spheroid size on day 12 of spheroid formation, spheroid volume. So control and the scrambled siRNA, scrambled is non-targeted, so basically nothing happened. It didn't affect the tumor growth. But if you see, for example, the EIF3C gene, uncoated, coated, I always, in all the groups, what I found was that with the targeting, that is with the HA coating, I found smaller volume. That means it was showing less tumor growth. So I thought it's a good idea to actually, in 2D I had already seen the difference between CD44 expression, it's no way you can compare. But then I decided, why don't I compare the CD44 expression in 2D cells, cells growing in a layer culture, what we are traditionally doing, and then CD44 expression in cells which are grown as 3D. So I checked the expression and I found 1500 times more expression of CD44. And probably that was the reason why my targeted nanoparticles with the HA coating were sticking very nicely to the spheroids, right? So this was much more similar to what we would have expected in mice, but I still, I didn't know how it would, that's why I was doing a lot of screening using these machines before going to the animal and asking my supervisor to give a lot of animals. So this is how the 3D and 2D look. So 2D, so these are M-cherry cells. That's why you can see they're fluorescently labeled. So here are 2D cells, ovarian cancer, and here they are not in a round bottom, but they are in a flat bottom. So they are growing as clusters, not one cluster, but couple of clusters. And I did also the expression profiling. I knocked down again in 2D and 3D, and I saw very good, over 80% knockdown of the gene in both 2D and 3D. So basically, we can do growth kinetics in 3D. We can also check the expression of genes, or if we are also doing drug treatment, then also we can evaluate the expression of different proteins using the 3D model. Then I had to do some functional activities. So this was done mostly in 2D, but for example, for the gene EIF3C. So what is a functional activity? So functional activity is the gene that is supposed to knock down translation protein EIF3C, whether it is still, because now it is in nanoparticle, maybe its activity is affected. So we still want to make sure that it is retaining its original activity. So we checked in 3D, and there were some tests we did. So in 2D and 3D, we saw very good activity, very good overall protein expression decline, and that was resulting in a slower growth of tumor. So basically, and also for the PLK1, the gene, it basically arrests the cell in G2 to M phase. It doesn't let it go to the mitotic phase. So we saw that with the PLK1 treatment, most of the cells retained in the G2M phase. So this was in 2D. So this still needs to be optimized. This is one of my goals still to optimize, because with 3D we know that there are layers. So you need to repeatedly trypsinize or do acutase treatment to get the cells out and see the FACS results. So this is a bit of harsh experiment to the cells. So that's why all this is basically going to the direction that we need a lot of optimization still in 3D, and it has huge potential. Then I went to the in vivo ovarian cancer model. So this was a bit of advanced metastatic model that I had developed. I had screened a couple of cells, and then I had optimized the dose. So this was in the nude mice, female mice. So basically, even before going to the main trial or main experiment, I just wanted to see whether the
fluorescently labeled nanoparticles, the coated ones are going more to the tumor area. So basically this was, I injected cancer cells to the mice on day 0, and on day 48, the mice had these full swollen belly, not extreme, but the beginning of the swelling. So I was monitoring them continuously using the IVIS system in vivo imaging because it has M-Cherry, so it was easy to follow up. And then I injected them with the Cy5, the fluorescent nanoparticles, and then sacrificed and then imaged them after 2 hours. So this is how it looks like. So these are this is the omentum, the fat part, because that is the primary site for the tumors to migrate after metastasis. And this is the ovary. So this is just to show how it looks in vivo. And this is the entire mice with the image. Yellow color is M-Cherry and red color is Cy5. So you can see the HA coated had more red on top of yellow. So they were going and attaching more, the HA coated nanoparticles were going and attaching more to the cancer cells. And we quantified the same thing in ovaries and omentum after taking out the organ, and we saw the same thing, that the coating, the targeting was working very well. So this was one of the, so why was I doing all this? The same thing I did also with just the PLK1 just to see the knockdown in mice. I'll just quickly go through it. I just injected again the only SIPLK1 just to see in the organs, different organs, ovary, omentum and ascites, whether there is better efficiency with the targeting, that is HA coated. Once I obtained this, then the main benefit was that instead of having 9 groups in animal study, I only had to have 5 groups. I excluded all the uncoated groups. So I had reduced the number of animals that I needed in my experiment. And also because so this was for n is equal to 10, but I did it in a batch of 5. So you can imagine you're just using double the number of animals if you're just going blindly with all your formulations. So you need to optimize. So that's why 3D models are really good for screening beforehand. So overall, I got some really good results because this was advanced case of ovarian cancer, at which stage the patients are usually coming, stage 3. And as you can see with the control and NC5 mice, most of the mice, they died by day 62, in fact 52, 54, all of them died. This was the median survival. But in case of the single siRNA, they stayed well up to 68 days in case of PLK1 alone nanoparticle. But with the combination therapy, you can see 60% of the mice survived. So this is a very aggressive tumor. So this was very interesting and good results. And I harvested the organs, ovary, omentum, and ascites, the main places for tumor. And I saw the knockdown of the gene, and this was also, this is in the tissue, not really in the 2D or 3D. So this is actually mouse tissue. And we got fantastic gene expression knockdown. So the key point is basically that I could minimize the number of groups, reduce the number of groups, which is one of the aim of the 3R conference, that 5 animal groups instead of 9. So this got published in the journal Small. This is the cover image. This is the ovary with tumors, and these are my lipid nanoparticles with the genes. So go 3D. This is my suggestion. I'm getting very good results, and this will actually eliminate the chances of failure in case the anti-cancer drug doesn't work out in the end. So you get better screening results in 3D due to tumor heterogeneity, which we are familiar now, because in the 3D tumor you have an outside proliferative zone, inside hypoxic and necrotic area, quiescent cells are there, cancer stem cells are there. You can do multiple assay readouts. Most of the labs, even if they're doing 3D, they're just checking whether cells can form 3D or not. They're not really using them for screening because it's a bit more, much more expensive and needs a lot of money for it. And reduction in number of treatment groups during animal study. And it is a very good bridge between in vitro and in vivo, because as I showed the kinetics of the tumoroids, it is quite resembling to the tumor that we implant in mice and monitor it with vernier caliper. So this is from the group where I learned actually the 3D tumor. She's based in Oncore clinical, it's a university clinic, Dresden, Germany. This is from one of a review article. It's based on actual results and very good results, which shows that the different parameters, oxygen gradient decreasing in the spheroid core, tumoroid core, lactate accumulation in the tumoroid core, the DNA double strand break. So all those parameters which are really making tumoroids as a very good model to study, because your anti-cancer drug would probably kill only the outside cell but won't affect your inside cell. That's where you really need to kill, because this is what is happening in patients. The inside cells, quiescent cells are not getting killed. Now, we are currently working on patient-derived tumoroid. This is also a data. This is a patient data I worked with. And this was actually initially the aim, that we would screen on patient-based tumoroids. But eventually, I got a couple of patient sample, ovarian cancer, but they don't grow beyond a certain point, or there are a couple of problems. This was one cell line, one sites from a patient which was showing very good growth, but then when we tried to form 3D tumoroids, it didn't form. You can see that there was just a lot of variation. We couldn't really work with it. And we gave up on using patients, and we decided to go with the cell lines, because they're more homogeneous relatively as compared to patient. So the idea is, hopefully me and Dr. Suresh, we have almost finalized the RAG grant, and we would be working on patient-derived tumoroids to be used for drug screening. And I recently was reading this paper a couple of days ago. So here, it's a fantastic paper. They've used basically the fluid which is present in the metastatic colorectal cancer, and use that gel or fluid as a replacement for a matrigel to grow the patient tumor. But again, already patient tumor, there is so much heterogeneity. And then if you're using a gel or a patient-derived matrix to grow the tumoroids, there is further heterogeneity. So to understand the development or the genetic profile of tumoroids, this is a good study to use patient material to facilitate the growth of tumors. But for screening, this is not really good. We need some biomaterial, something which actually supports the patient's cells, which right when we get them from a hospital, we are able to support them in terms of some matrix. So with this, we hope that we do more patient-derived tumoroids and also humanized mice. We reduce the number of mice in studies. And thank you very much for your attention. And I would like to thank all my previous institutes who are the reason I'm here today with all of you. OncoSeek, which has been phenomenal partner, one of the first companies doing 3D in India, really doing very well. And Mahindra University for taking me as associate professor and also hosting this conference. Thank you very much for your attention. Are there questions?
Thank you, doctor. Actually, there is no question.
There are questions. Or if we are short of time, I can take later.
Hello. Okay. Thank you, Dr. Manusmriti Singh, for sharing a very informative and fascinating finding from your work on nanomedicine screening on 3D models and streamlined in vivo models. Now I would like to invite Dr. Lily and Dr. Manda to present Dr. Manusmriti Singh with a token of appreciation. Hello. Now, I would like to invite Dr. Uday Saxena to introduce Dr. Akela Venkateshwarlu and present the IEFR Lifetime Mentorship Award.
Yeah? Okay. Yeah. Thank you so much. I first want to thank Suresh for allowing us to present this award. This is the first award, first time that we are giving out this award. I also want to thank a lot of my Dr. Reddy's colleagues who have come specifically to see Dr. Venkat actually. So you will have a chance to interact with him at tea time. The award is being presented on behalf of IFR, which is Indira and Ishwar Foundation for Research, This is a foundation my family created in my parents' name. Both my father and mother were scientists, and they're not with us anymore. So we thought that to carry on their legacy, we should start a foundation. So this is a one-year-old foundation now. The purpose of the foundation is to train and educate young women scientists specifically. My mom was a sociologist. My wife, who's here, is also a scientist. She has a PhD in electrophysiology from Columbia University. So there's a lot of women scientists in the family. So we thought we should encourage women scientists. So the foundation trains women, as I said, and we train them for free. In fact, we pay them a stipend, and we take cohorts of 3 young women. These are fresh master students who are looking for guidance in their career, who really don't know whether they should do a PhD or go to industry or stay at home. So that is a gap we are trying to fill, and the first 3 have graduated. In fact, they actually got a publication. And there's a poster out there you will see. That was the work of these students along with the manager, Sharanya. So it turned out to be really good. We trained them in actual translational research. They get to do their own research. This is not a cookie cutter program where you come and learn HPLC, then you learn, this is not one of those. Here we give you a problem, you have to work on it and try your best to get it published. So leadership and entrepreneurship are the other 2 things we train them. We have had lecturers come and talk to them about leadership and how to think fearlessly. In science specifically, it's very important to be fearless. We also train them in entrepreneurship. So this has been ongoing in my parents' memory for 1 year. And for the first time this year, which is the 2nd year, we thought we should also institute an Lifetime Achievement Award. And I'm very honored to say that the first Lifetime Achievement Award from IFR, we requested Dr. Akela Venkateshwarli to receive it. I'll tell you a little bit more about him. But he was extremely humble enough to accept this award. I really thank you for your humility and, honoring my family really by accepting this award. It's a great honor. I wish my parents were here to see you. They knew you quite well. They saw you and all that. So it is wonderful that you were able to, and one phone call and he accepted it. There was no nakhra, no nothing. He said, when is it? What is it about? And then said yes. So I'm really thankful to you. And it's very consistent with how I know you. I just want to say a little bit more about Dr. Venkat. I see that Dr. Radha, Dr. Venkat's wife is also here. She's also a physician scientist. She's actually a geneticist who's still very active, has done a lot of wonderful work, and I also see their grandson Vishnu that is here. So it's so nice to see all of you. I was just saying, Vishnu, that God doesn't make people like your grandfather anymore. So you spend as much time as you can with him and learn. He's just unique. So just to give you a background of Dr. Venkat, he's actually a builder. I mean, he has a PhD and postdoc from UPenn and Harvard, and one of the few people who came back to set up shop in India. Why is he a builder? He's a builder because from when I knew him, he started building Dr. Reddy's Research Foundation. He was the founder president of that. And then he built Origin. Any of you here from Origin? Yeah, the founding director for Origin was Dr. Venkat. Then he built Dr. Reddy's Institute of Life Sciences. Anybody here? Oh, there you are. So, and throughout the course of this buildership, he recruited some top-class people. I also see Rajnikant, Dr. Venkat's son-in-law, who's also a scientist, and Uma, who's also a scientist at the back. Thank you so much for coming. So he, during the course of building these institutions, he knew that he couldn't do this alone, obviously. He went around the world recruiting the best talent he could. Sincerely, I met him first time in 1992, I think. He came to Michigan, where I was working at Park Davis Pfizer, specifically to try and recruit me. I couldn't join at that time, but he persisted. For 10 years, he persisted, and in 2000, I joined. So the depths to which he went to get good people on board was just amazing. I mean, I have not seen anybody do that. As a result of which, today we have people who graduated under his mentorship who are presidents. We have a president who runs Zydus, for example, who came from us, Arnie Chatterjee. Arnie, are you here today or no? He's the president there. Many senior vice presidents all over, many vice presidents. We also have a few CSR directors who trained under him, as well as many other people who are professors. I mean, the number of people he trained and mentored must be running in a few thousands. If, sincerely, yeah, that's the kind of impact one gentleman can have. One gentleman with foresight and patience to be a mentor can transform an industry. I can honestly tell you, all the leadership positions in the industry today have some linkage to Dr. Reddy's Research Foundation or not. Some means there has to be some linkage. And it's all because of Dr. Venkat. I was a benefactor. He was my mentor. Now I see him as a mentor and a friend. I often call him, we get together for coffee. The humility is amazing. The humbleness that I see in Dr. Venkat, I really want the young leaders today to absorb that. You know, people achieve a lot. There is, I mean, if you're bright and lucky, you're going to achieve. But to achieve it together with humility is something that is very, very rare. You see a lot of arrogance in leaders today. And people tell me that's the new generation, you're the old generation. That's fine, but humility never goes out of fashion. So be humble, stay grounded. I think that's a very important message. Besides that, I think the builder part and mentorship, he's also a very spiritual individual. My wife and I have benefited a lot from his spirituality in some very difficult times in our life. The first person we always called was Dr. Venkat. And he always has a way out for you. Within 10 minutes of talking to Dr. Venkat, you feel great. You know, the spiritual connection he has. It's the whole package, like I was saying. God does not make people like these anymore. We are very fortunate to have interacted with him and been blessed by him. And now you have honored my family by accepting this award today. So I'm very grateful to you. And in all his humility, he said, I will not say anything today. He does not want to speak. So that's the, I mean, he could have easily come up and said, I did this, I did that, but he's refused to say that. So Dr. Venkat, I would like you to come to the stage and receive the award now, please. Can we have a standing ovation for Dr. Venkat, please? Suresh, you want to give the flowers? Yeah. We'll take a picture. You can, that's fine. Please. What is the sequence? I give this first, you give the
Vishnu, Rajni, Uma, please come. Nina, please come.
Just take one quick picture.
While that is happening, I do want to say that this is an annual award, so if you have any suggestions for next year, please. I'm looking for people who have selflessly mentored younger generation and brought them to a position of leadership. So if you know of anybody, please let me know. Thank you. Thank you so much, Dr. Venkat and family. And Suresh, thank you so much. You're really thankful for providing this forum for us. So, yeah, why don't we will take some pictures outside. There is another speaker now from the US that's going to talk. So we'll go outside and take pictures? Yeah, okay.
Good evening, ladies and gentlemen. It is Uday's generosity on behalf of his family that he has chosen me to give this first mentorship award in memory of his parents and on behalf of the foundation IEFR. Uday and I have been colleagues of late, worked together in equal capacity, but at one time I was his mentor. I was after him for 10 years to bring aboard Dr. Reddy's to head our research program in Atlanta, Reddy US Therapeutics. Apart from scientific mentorship and interactions between us, we have become close friends, became very family friends. He mentioned about my spiritualism, and he is a beneficiary for it. I'm grateful about it. God has given me the strength. But apart from this, I have recruited 67 PhDs, about 250 MScs, dozens of BScs. My mentors both were Harvard chemists. Professor M.P. Kawa gave me a graduate fellowship when he was in Ohio State University. I was to join him in the fall of 1965, but I got delayed. I joined him in 1966 fall. But then he moved from Ohio State University to Wayne State University, Detroit. The reason why he moved was his wife was a psychiatrist. She got a job in Detroit. So by the time I joined him, I went to Wayne State University, Detroit. 6 months later, he didn't like Detroit. He moved to University of Pennsylvania, Philadelphia. That's where I completed my graduation. Thereafter, I joined Professor E.J. Corey, also a Harvard MIT chemist, became Harvard professor, and I was one of the many 475 researchers who were trained by him. So under these two professors, mentors, and the researchers who worked in those laboratories, stalwarts have worked with them. Subsequently, 4 or 5 of them have got Nobel Prize. Interactions with all these people have given me some special flavor to build research organizations and develop teams and make them perform at high levels. So, best opportunity came across. Although I was working in Sibagaigi in the beginning, now part of Novartis, then Smith, Klein and French, which is part of GSK, and thereafter I worked in a collaboration with DuPont and Atul Products, which is headquarters for American Cyanamide, Sibagaigi, and Imperial Chemical Industries. With all these international collaborations and meeting with professionals, I became perhaps proficient in judging people and their qualities and abilities. That has helped me a lot. And best of it was performed, demonstrated in Dr. Reddy's Research Foundation, a story of which will take hours together. I will stop here and thank Dr. Uday Saxena and his family for their generosity in selecting me for first mentorship award. Thank you very much for patience.
Thank you, Dr. Akila and Dr. Uday Saxena. Last but not least, our next speaker is Dr. Charugupta. Dr. Charugupta earned her PhD in molecular medicine from Hannover Medicine School, Germany. Her doctoral research focused on IDH1 mutated acute myeloblastic leukemia, providing mechanistic and therapeutic insights. Dr. Gupta's work contributed to the development of several clinical trials for AML. During her postdoctoral work at the University of Michigan, Dr. Gupta identified and characterized adhesion receptor crucial in neutrophil excavation. Apart from research, Dr. Gupta volunteers as a career podcast host in online science communication. Currently, she serves as a technical information scientist, leveraging her expertise in mouse model disease modeling and hematology and oncology to contribute to global research goals. Dr. Gupta has shared her talk with us, where she provided her how-to guide for selecting xenografts and models for preclinical cancer researches. Following which, she will join us via Zooms to answer any questions you might have.
It's not showing here. Just a second. Excuse me, can we have some help, please? Can we have? Wait, let him do it. It's not showing. The video is not showing. This is the one which has. 1 second.
Extend more on.
Okay, thank you.
Sound is connected, right?
Yeah, it's connected.
Recorded. Yes.
I check once.
Sound is there. Yes.
Information scientist at the Jackson Laboratory. I am here as your guide for selecting xenografts and models for preclinical cancer research. Before we begin, I would like to take a moment to also introduce to you the Jackson Laboratory. The Jackson Laboratory, JAX for short, is an independent, non-profit biomedical research organization that leverages a unique combination of research, education, and resources to achieve our mission to discover precise genomic solutions for disease and empower the global biomedical community in its shared quest to improve human health. JAC strives to empower scientific excellence through innovation derived from discovery, research, and education. We have our own internal grant-funded research faculty who perform groundbreaking discovery research. Additionally, we offer mouse models, preclinical services, and education to support the worldwide biomedical research community. JAX has a global presence with multiple sites represented by the dots shown within this map. We provide preclinical research services and have shipped mouse models to more than 64 countries around the world, represented by the light blue areas within the map. Now that you're familiar with the Jackson Laboratory, let's discuss our topic for today's presentation. Nearly 10 million deaths are caused by cancer each year, of which many can be prevented by early detection and development of viable therapies. Perhaps you recognize this need and have a potential cure at hand. Selecting an appropriate preclinical model for cancer research is crucial to ensure the relevance and reliability of your findings. Considerations to guide your choice may include defining a clear research objective, understanding the tumor type, heterogeneity, and complexity, the immune system interactions, drug testing and efficacy, limitations of cost and scalability, ethical considerations, as well as validation and reproducibility efforts. You have likely answered several of these considerations and resonate with now being ready to evaluate the efficacy of your therapeutic in vivo, essentially taking your idea from bench to bedside. This guide will help you narrow the 8 considerations down to just 3 key considerations needed to design your ultimate preclinical cancer research model. With the model in hand, we will explore sample study designs, evaluating therapeutic efficacy in a tumor-bearing as well as immune-humanized tumor-bearing mouse, leaning into the advantages of including the immune tumor microenvironment. Each sample study will be followed by a literature example showcasing how JAX mice helped answer real scientific questions. Finally, we will explore the multitude of ways in which JAX can support your translational journey by providing mice, xenografts, consultancy, and other study support. Let us begin by understanding how to create the perfect model for your discovery research. The 3 key considerations here are the type of xenograft, so the understanding of how best to model the cancer, the host mouse for this tumor, as well as the incorporation of human immune system to create a more accurate and predictive study model. While you certainly want to choose a tumor that closely resembles the specific cancer type you're investigating, it is important to keep in mind the purpose of your research. Are you studying tumor biology, drug efficacy, metastasis, or immunotherapy? Different models excel in specific areas, and it is this clarity that will help you choose between human cell line-derived and patient-derived xenografts. Let us explore the characteristics of these xenograft types further. Cell line xenografts are established via serial passaging from human patient samples and therefore become homogeneous in cell types. Patient-derived xenografts are human tumor specimens that are able to be passaged in immunocompromised mice, and their clonal heterogeneity offers a closer mimic of actual human tumors. CDXs are useful in high-throughput screens and are cost-effective, while PDXs help understand tumor biology and further personalized medicine research. However, they are costlier. CDXs are widely available and cultured in many labs. PDXs are somewhat elusive and offered via hospitals or some repositories such as JAX. In summary, while PDX models retain the patient-specific characteristics, CDX models provide a simpler and more standardized approach for cancer research. Researchers choose between these models based on their specific research goals and requirements. So having settled on a xenograft makes it suitable to now choose the host mouse. While the host needs to be immunocompromised to accept a xenotransplant, the degree of immunodeficiency is also influenced by the type of cancer being studied. For example, breast cancer cell lines will engraft in athymic mice lacking adaptive immune components, but leukemias and lymphomas require further immunodeficiencies in the adaptive as well as the innate immune systems. There are several immunocompromised mice ranging from the earliest nude mouse models to the most immune-compromised mice, not-skid gammas, to choose from. For the purpose of our discussion today, I will introduce the two ends of the immunodeficiency spectrum, the nude and the NSG mouse. On the least immunodeficient end is the athymic nude mouse that lacks mature T cells and therefore shows diminished B cell responses. The spontaneous FOXN1 nude mutation also renders the mouse hairless, making it easy to visualize the growth of subcutaneously engrafted tumors. This well-characterized model has an intact innate immune system, making it suitable only for solid human cell line xenografts and not for PDXs or primary human cells. On the most immunodeficient end is the not-skid gamma or NSG mouse. Alongside a complete loss of an adaptive immune system and innate system components, a key advance here is the absence of NK cells, which are the greatest impediment to human tumor and xenograft establishment. The NSG mice also show enhanced engraftment of human hematopoietic cells due to a mutation in the SERP alpha gene in the NOD genetic background. Excellent for primary human cell and PDX engraftments, these mice have become fundamental workhorses for oncology drug discovery. An added advantage is the high fidelity preservation of patient tumor characteristics in these hosts. In this experiment, patient lung tumor fragments were engrafted subcutaneously into the not-skid gamma and the bulb C-skid mice. As you can see, the tumor engrafted into the NSG mice retained a typical epithelial carcinoma-like appearance very similar to the original patient biopsy, whereas the engraftment in the bulb C-skid mouse has a more fibrotic appearance. The NSG sample retains human HLA staining and also shows very little infiltration of mouse white blood cells. This study exemplifies that the NSG mice better approximate the human tumor in an engraftment setting over other previous generation immunodeficient mice. An important deliberation, however well the tumor may be modeled, all immunocompromised mice lack the ability to replicate a human tumor microenvironment. Tumors are never lonely. They are surrounded by immune cells in their microenvironment, as well as have infiltrating lymphocytes. This immune microenvironment can affect therapeutic kinetics. Immune modulation of this cancer-sustaining microenvironment is called immunotherapy. It is therefore important to consider adding components of the human immune system into our host mice to create a more holistic and robust preclinical model. Speaking of immunity, we reach the final consideration in creating the ideal cancer study model. The xenotransplanted host mouse can be built to incorporate the human immune system, providing a more realistic study setting to identify potential adverse effects, efficacy, and safety profiles. Immune humanization is also driven by the therapeutic being tested. Immunotherapies rely on interactions between immune cells and tumor cells. Additionally, patient-specific responses vary due to individual immune profiles. It is certain, though, that a fully humanized immune system predicts responses to all therapies, be they small molecule or CAR-Ts, more accurately than models lacking human immune components. The human immune system engraftment at the Jackson Laboratory is performed in two ways. Peripheral blood mononuclear cells, or PBMCs, are extracted from leukoplaques and engrafted into NSG mice, recapitulating a human-educated immune system useful for short-term cytokine release therapeutic safety and GVHD studies. CD34 positive hematopoietic stem cells derived from umbilical cord blood are engrafted into myeloablated hosts to create a mouse-educated human immune system ideal for long-term target validation and efficacy oncology studies. The hematopoietic stem cells often need transgenic aid in the form of human growth factors or cytokines to engraft elusive cells. While abundant in T cell frequency, not all hematopoietic cell types are equally engrafted in the NSG mice, similar to human peripheral blood. To allow for greater engraftment of these other cell compartments, the Jackson Laboratory has developed transgenic NSG mice. For example, the NSG IL-15 mouse has physiologically relevant IL-15 levels required to engraft a higher number of natural killer cells. We also humanize the NRG mice, which offer better chemotherapy and radiation resistance than NSG mice. If you wanted to know which model was best suited for your research, you can compare and contrast their immunophenotypes using this handy table. Now, with the right immune complement in mind, let's define our immune-humanized xenotransplanted mouse. Tumors, be they PDXs or CDXs, can be engrafted onto immune-humanized mice. Based on tumor growth kinetics, you may choose to first engraft the tumor and then PBMCs, or vice versa, to generate the OncoHue PBMC mouse. With HSCs, the tumor is almost always engrafted post-humanization, since this is a longer-lasting model, generating the OncoHue HSC mouse. These OncoHue NSG mice are the bridges that cover the gap between preclinical studies and clinical outcomes, enhancing our understanding of immunotherapies and advancing cancer treatment. With deep insights into the xenograft closely modeling the cancer, an appropriate immunocompromised mouse to host it, and understanding of immune roles, we have created a powerful preclinical model for unraveling the mysteries of cancer and developing effective therapeutics. Remember that no single model is perfect. Each has its strengths and limitations. Consider a combination of models to address different aspects of your research. The best way to recognize how many such models you may need is to understand the data output from a designed experimental study. To realize a study outcome, let us look at some sample JAX study designs. These are studies we routinely perform for our clients and match them with a similar published example to truly grasp the wealth of knowledge one study can result into. We will talk about both onco or tumor only with a small molecule efficacy example, as well as OncoHue, that is immune humanized and tumor engrafted studies, with a monoclonal antibody effect example. Designing an onco study to evaluate therapeutic efficacy on engrafted CDXs or PDXs is very straightforward. At JAX, we begin by engrafting tumors subcutaneously into immunocompromised mice and group them for synchronous tumor growth. We can begin prophylactic dosing almost immediately or wait for palpable tumors for therapeutic trials. A typical 5-arm study with 10 mice per arm includes a vehicle control arm, reference compound arm, experimental therapeutic arms dictated by number of, for example, small molecules being evaluated. In this study, we chose to evaluate 3 molecules. Compound dosing can be administered through all common routes, that is, IV, per oral, IP, and subcutaneous. Animals are monitored biweekly. For body weights, clinical scores, and tumor sizing using digital calipers. A typical study duration is about 60 days or until individual tumors grow to about 2000 mmQ. In-life and terminal samples such as serum, whole blood, and tumors can be collected for further analysis. In such an oncology efficacy study, researchers can evaluate the effectiveness of a treatment in combating cancer by noting tumor regression, overall survival, biomarker problems, and pharmacodynamics. An example is the impact of naturally derived compound curcumin on the growth of chronic myeloid leukemia, CDX. Nude mice were injected subcutaneously with CML cell line K562 that is able to form solid nodules. Mice were dosed intraperitoneally every 2 days with vehicle corn oil or compound curcumin. As can be observed through the graph and the representative images of the excised tumors at day 22, the vehicle-treated mice reached large tumor volumes while the tumor growth was inhibited by a curcumin treatment. Curcumin is known to inhibit tumor growth by increasing ROS levels. The authors also noted average tumor weights, with the vehicle group being much higher than the treated. Now, while a nude mouse engrafted with a cancer line will develop a large tumor, the tumor that develops does so without the normal microenvironment expected for an actual human cancer. It is necessary to note that this and similar onco studies are unable to describe the effects of the tumor microenvironment on therapeutic efficacy and avidity, which rely on immunohumanization for evaluation. Designing an OncoHue study to evaluate immunotherapeutic efficacy on engrafted CDXs or PDXs begins with creating the OncoHue mouse. In this study design, hematopoietic stem cells are engrafted into irradiated NSG mice. HSCs from 2 donors were used to account for variability in patient-specific responses due to individual immune profiles. This is called donor balancing and is highly encouraged for both HSC and PBMC engraftments. Mice showing greater than 25% circulating CD45 positive cells are subcutaneously engrafted with the desired tumors, and twice-weekly checks are performed till the tumor reaches a dosing size. These mice are then enrolled into the safety and efficacy study. To evaluate immunotherapeutics, such as monoclonal antibodies, a typical per donor includes a vehicle control arm, the reference monoclonal antibody. In this case, it is pembrolizumab, since an anti-PD-1 therapeutic was being tested, and experimental arms. Mice were randomly split between the study arms, assessing either IV or IP dosing routes. Animals were monitored 3 times per week for body weight, clinical score, and tumor size. If the body score is less than or equal to 2, or tumor size is larger than 2000 mm3, mice were sacrificed. In-life and terminal samples, such as serum, whole blood, and tumors, can be collected for further analysis, such as cytokine release screening. Such immuno-oncology studies hold immense promise in revolutionizing cancer treatment by empowering our own immune system to combat tumors. A literature example demonstrating this outcome is the T-cell mediated breast cancer cytotoxicity modulated by anti-PD-1 monoclonal antibodies. NSG mice humanized with HSCs from 2 donors were subcutaneously engrafted with a triple-negative breast cancer PDX. Mice were dosed with either vehicle, standard of care cisplatin, or anti-PD-1 monoclonal antibody pembrolizumab. Graphs from both HSC donors show tumor regression upon pembrolizumab administration, albeit with varying trends in tumor size reduction. This exemplifies the need for donor balancing. The binding of PD-L1 to PD-1 keeps T cells from killing tumor cells in the body. Blocking the binding of PD-L1 to PD-1 with an immune checkpoint inhibitor such as pembrolizumab allows the T cells to kill tumor cells. To understand the difference between immunotherapy readouts in an onco versus OncoHue model, let's look at this CDX TNBC example. Naive or HSC engrafted NSG mice were orthotopically transplanted with the triple-negative MDA-MB231 breast cancer cell line-derived xenograft. OncoHue mice treated with pembrolizumab showed a tumor regression in comparison to vehicle-treated group. However, no treatment response was noted in the naive non-humanized mice. This is because pembrolizumab leverages the host immune system for anti-cancer efficacy. No immune system means no response. Immuno-oncology or OncoHue studies explore how the immune system can be harnessed to fight cancer, helping researchers evaluate therapeutic efficacy and model clinical immune response variability. Now, with the knowledge of a robust preclinical model and an understanding of how experimental studies can be designed to answer key research questions, let us take a peek into how the Jackson Laboratories products, services, and scientific assistance can help your discovery research. With over 13,000 strains housed at our repository, the Jackson Laboratory is an ideal stop for research mouse models, be they naive, immunocompromised, humanized, or tumor engrafted. JAX can run antibody evaluation, pharmacology, and efficacy immuno-oncology preclinical studies with high finesse, leveraging our scientific expertise and PDX database. Above all, in the spirit of constantly supporting the global biomedical community, JAX has a team of PhD-level experts as well as freely available educational modules to help you on your preclinical journey.
Thank you, Dr. Charugupta, for joining us all the way from the USA for an informative this webinar. And she's joined us through the online for any question and answers you have.
Hi, Nikita. I can. How are you?
Questions? Please ask Dr. Charu. She's joining us from all the way from USA. Any questions?
Hi, Charu. Suresh here. I heard
Hello,
this talk.
Suresh. How are you?
Good, good. I heard this talk several times, but still have a question. I don't know.
Yes.
So, from the perspective of the size of the tumor, you said 2,000, right? As an
Yes.
endpoint.
Mhm.
How do you differentiate that for various tumors? Because some of the tumors may not or may cross that size also, right? Do you face that challenge with several tumors? Tumor types, I
So
mean.
that's the upper limit for how much we would like a tumor to grow. That is based on our IACUC and our animal approval boards. And so we have the permission to work with tumors up to 2000 mmQ. There are facilities who can only work with tumors which are about like 1,000 or 1,500 mmQ. So that's something that should be tumor size at the time of sacrifice. If you in your control colony, could really be determined by what your institutional animal board approves. One, and of course, all tumors have a different kinetic. There are some which are really slow, and usually in studies you would want to do a sort of comparison. It's like even if it is a smaller tumor, if you start prophylactic dosing at like 50 mmQ or therapeutic dosing at 200 mmQ, you have a comparative chart of if the treatment that you're using is actually working. So at the end of the day, there is a max size of the tumor, and that's the humane way of doing the experiment on the animal. But if your tumor, for example, does not grow beyond 600 cube and it's super small and it's end of life for the animal, that's perfectly fine as long as you're able to get relevant data of your treatment.
Okay, thank you. So the endpoints need not reach, you don't wait until you reach that major endpoint of 1,500 or 2,000, right? There's constant monitoring.
It's not always necessary to reach it. That is just the upper limit.
Correct. So the 3R says that you should monitor it continuously so that anytime there is a body mass size of greater than 10% or 20% loss of and flank movement is missing, the animal is not able to reach the food, and also that there is necrotic centers, and you can also employ imaging in between your studies. So I think you do all those things. I just wanted to ensure that the audience realizes the value of endpoints by this question.
Yeah, it's a good point that you raise, and we do typically do, like, as you mentioned, all of the characteristics of the clinical score. The body score should be about 2. If it's less than 2, it's something that it's an animal we would have to sacrifice. You have to be really careful about the body weight changes, about the posture of the animal, about the coloring of the feet, especially with leukemias. The feet become really wide and these animals are really anemic. So you have to kind of be very careful about if the animal is feeling undue stress and undue pain, then that's the time then when you should sacrifice the animal. So you should be careful about these. And I encourage everyone to contact their veterinary staff and their facility administrators, and they have really good charts and really good trainings usually about all of these things. And if they would like some more information, definitely we would, at the Jackson Laboratory, we would definitely be able to provide some more resources.
What's the major tumor tissue that gets transplanted into your most possible nude mouse or skid mice or any mouse that is immune compromised severely? What is the most common tumor type you use? I think we know the answer for that, but what is the one tumor type that cannot be performed using these humanized animal models?
One tumor type that cannot be engrafted?
Yeah, correct.
I think we've been able to engraft majorly every tumor model. It's just that we haven't done all of them orthotopically. So at the Jackson lab, we do subcutaneous in the hind flank. We don't do orthotopic engraftments except for mammary tumors. And I think it really, at the end of the day, there are, if you're talking about patient-derived models, about 25% of them get engrafted anyway. So there is a large population of patient-derived tumors that does not engraft. And there isn't really, at least to my knowledge, there isn't a specific reason or a set of reasons that have been pointed out for that occurrence.
Okay, I have a quick question for you. We are a startup company that is developing drugs for chronic and acute myeloid leukemias. And it's being developed in the US, but we also want to develop in India for Indian patients. One of the things we've been thinking about doing is PDX models using Indian patient-derived cells. Do you think that's a good idea? Is it necessary?
Do you mind repeating the second part of your question, like facing the laptop? I think I can only hear through the...
Oh, sorry, I shouldn't be facing the laptop. Okay.
Sorry about that.
I didn't know that I was on the laptop. So we are developing 2 products, one for AML and CML. And the products were discovered in the United States. And now we are trying to develop that in India for Indian patients. So one of the studies we want to do before we take it to the clinic is use Indian patient PDX samples and then do in vitro and in vivo studies. So the question is these are turning out to be very expensive. So the question is, do you think it's a value add, or is the AML, CML, same all over the world?
So I know that the FDA does encourage a community-based approach now, that people from different races and different communities are included in the clinical trials. That is a push forward because there are obviously some racial differences. There are some differences in the characteristics, in the diets, in the epigenetic influences. So if this is this clinical trial and the preclinical studies, if it is going to be translated, say, from the US to India, there should be a trial in India with Indian patients in the Indian conditions, with the epigenetic factors that are influencing these particular set of patients. I completely understand the fact that these models are very expensive. They're expensive all over the world.
Right.
And part of that expense really is from the fact that they're so elusive. And a majority of them are under restrictions, and leukemias are not the highest occurring cancers in the Indian subcontinent.
Yeah.
But I would definitely encourage you to try that. If you, and I mean, if you are running into trouble, if you're like, well, this is becoming too much, then try to obtain samples from other repositories. Like our repository has about 7 AML samples, and you can start working with them and start optimizing the experiments, at least at your facility, and then whatever Indian patient samples come by, you can continue to use those. But I definitely encourage that you do your clinical trials with Indian patients as well. That does make a lot of sense. Because we do not know the patient experience, and the patient experience, we know, plays a huge role in how they receive treatment.
Then one last quick $1 million question. We go ahead and do the in vivo PDX and it fails. Should that be a go, no go decision experiment, or should we just ignore it and say, let's go ahead anyway?
So I'm not really aware of the particular study and the product that you're referring to, right? So there are multiple aspects here to consider. Is it an immunotherapy? Is it, if it is an immunotherapy, then even if you put it into a cancer model, at the end of the day, you want a healthy immune system also to be activated to work against that particular system. So there comes in the variability of having multiple donors. So if it doesn't work with one, if it doesn't work with two, but it works with 2 others, then how do you look at your data? Is it 50% effective? And then there are all drug approval boards in India as well as in the United States, the EMA and PI in Germany and Europe. They all have particular things that they ask you for. And there is a checklist. And when you go into clinical trials or you file an IND application, they ask you to complete a portion of it. You're asked to have a percentage of positive results to move this therapy into phase 1. I mean, preclinical trials only give us so much. At the end of the day, if it is more than 50% promising, we're like, maybe we should try it in the clinic. And that's where phase 1 comes in. And then phase 1 determines if it moves forward or not.
Yeah, thank you so much. Yeah.
Most welcome.
Yeah, Jugnu wants to ask.
Yes. So I would go in vitro first. And there are multiple subtypes of AML. So once you have checked different subtypes of AML, you know it works in M2, M4, it doesn't work in M1, whatever. Plus you know the genetic background. You refine the ones that you want to put into an animal and then go forward. So I don't think you need to necessarily repeat the PDX with Indian patient samples, but PDCs of Indian patients we can do for you. What you get for free will not be worth it.
No, I completely understand. $1 million question as management is, if those studies fail in vitro, do you make a go, no go? For whatever reason, you never know, right? So
That is a nuanced question.
it's a very important because you're spending millions after that, so.
I have to give you a long answer. These are not quick yes, no answers.
Okay.
We run the 3 approved drugs,
Okay.
cetarabine, arsenic
So you control
trioxide,
the experiment
doxo.
and all that.
Then
Okay.
you see, even these drugs, doxo, it doesn't work in all AML patients.
I know, I've gone through the literature. I'm not a big fan of the hit and miss type of things I'm seeing.
So that's why I'm saying you need to look in aggregate.
Okay.
You need to look in aggregate.
Okay, we'll talk offline.
Correct.
But thank you for offering, the cell lines and, yeah. Cells, yes. Thank you. Okay, you're going to introduce? Yeah.
For joining. Thank you, Dr. Charugupta, for joining us all the way from the US. And it was a very informative Q&A session which was happened. And the award will be handed over to Dr. Krishna on behalf of Dr. Charu. And the panel discussions will happen now. Bye.
Thank you, everyone. Thank you for having me. Bye.
Thank you.
Thank you, Nikita. Appreciate it.
Okay, I'll invite Dr. Suresh Pasala. Yes, yes. I'll invite Dr. Suresh Pasala to present the award to Dr. Krishna on behalf of Dr. Charu.
I wish she could have been here.
Yeah, she left.
Yeah, she just locked up. I'll tell her.
Yes. Reshma, take a photo.
Yes, I send that to her.
Yes. Yeah, okay. Now that we have gained insights from our speakers, it's time to deepen the conversation with our us versus them first ever debate-style panel discussion on the pros and cons of animal models and alternative models. Our panelists today are Dr. Suresh Poshala from Oncosic Bio, Dr. Radharna Pallu, FABA, Dr. Jugnu Jain, Sapien Biosciences, Dr. Ajit Kamath, Pandorum Technology, Dr. Subramanyam Mangala Reagent, Dr. Ranjan Chakrabarti, Dr. Akbar Shah, Society for Alternatives to Animal Experiments, and Dr. Uday Saxena, founder of IFA, who will be the moderator for today's panel discussion. I cordially invite the panelists and the moderator onto the dais. Now I will hand over the proceedings to Dr. Uday Saxena.
Yeah, thank you so much. Is there only one mic that will go around or? Yeah, okay. Yeah. All right, maybe we can have the 4 animal people sit on the right-hand side and those who are not for animal on the left-hand side. So which one are you? You're not for, you're for alternative, right? Sit here then, yeah. Yeah. What happened to Dr. Akbar Shah? Yeah, he's here. There's only 2. Ranjan is coming here, right? Yeah. Ranjan, come here then. Yeah. Left side is alternatives to animals and right hand is for animals. So we are going to stop strictly at 6 p.m. because Ajit has a flight to catch. And the format is we will go through the questions for each side and then there will be an audience poll to tell us which side has won. So keep your buzzers ready whichever side has won. And we will allow for questions for about 10 minutes or so. All right? Everybody is happy with that? Yeah. Okay, great. So my first question, since Jugnu is sitting to my left, immediate left, I'll ask her. Jugnu, how much do you really trust the emerging alternative to animal models? And the way I'll pose that question is, are you willing to sell your home and put all the money into alternate to animal technologies? Do you really believe these things are here to help us?
Selling my home, okay. So I think when they are used intelligently and they are interpreted realistically, yes. The example I would give is of cystic fibrosis, which is my previous company, Vertex Pharmaceuticals. They used patient lung tissues to look for CFTR channel potentiators and correctors. There was absolutely no animal model at that time for efficacy. And so 3 drugs have come out of that, and even FDA accepted that model, in vitro model, not in vivo model, for efficacy, followed by the standard tox that was done in 2 species. So we are not looking to eliminate, but we are looking to decrease the use of animals by using patient tissues, cells derived from patient tissues, to go beyond inbred mice and homogeneous cell lines.
So, I mean, you've cited a great example that I don't think many of us knew. So if I had to summarize, make an assessment of where you stand, you're saying you would be willing to put all the money you have on these technologies. Yeah? Okay. Thank you. Ranjan, I'm going to ask you the same question. You're sitting to my immediate right, and you've done a lot of work on in vivo and PPAR. So of anybody else in this room, I think you've done the most amount of work, and you have produced clinical candidates. So clearly the models worked for you. Would you say that that's the type of work I want to continue to do, and or shift over to alternate to animals, because those are emerging? I mean, where do you stand on the use of animal models to predict human biology?
See, I have that unique advantage of working first 12 years in drug discovery and the last 11 years of my professional life in US pharmacopoeia in biotherapeutics, I will say. Now, there's a big difference there. As far as animal usage is concerned, small molecule area, I think in our time itself, we have started using for ADME PK the alternate models, in vitro models, enzymes, micronucleus, and cell-based in vitro assays. But at the same time, we also started using transgenic mice models. And I will tell transgenic mice is a refined model instead of normal rats and mice. But when I moved to USP, and especially when we were in constant touch with FDA while developing guidelines, for biotherapeutics, actually using this kind of models, animal models, especially I think besides chimpanzee, most of these are a big questionable data. So there I will suggest that alternate models give a better, especially in the case of biosimilars. See, what happens is biosimilars, you have a reference product where we know the characteristics of that. So if your similarity is very clear, you can even, regulators are also slowly accepting that in vitro like immunogenicity, which is one of the major toxicity. Today we have very good models to predict this immunogenicity, and then maybe you can do some bridging post-marketing or pharmacovigilance study. So I will not say directly yes or no. I think based on the area where you are working, based on the disease what you are working, you have to choose.
Thank you. From your answer, it sounded like you should be on the left-hand side. Yeah. Ajit, since you're next, the drug industry has been around for over 100 years now, and we have had many blockbuster drugs that have come out purely using animal studies. the statins, I mean, the work that I did was done on liver homogenates. There wasn't even a target isolation. So, and human quality of human life has improved, longevity has improved. So clearly the animal model-based paradigm has delivered. So why fix something that's not broken? Why should we go to alternates?
No, it's a very good question, Uday. I think things have moved on. Things have evolved a lot. I mean, you worked in pharma, I worked in pharma. We were working on yeast enzymes. We were working on rabbit enzymes. We were working on guinea pig enzymes because there was no human enzyme available. Okay? We were doing structures of proteins from guinea pig, trying to see how that would help in developing drugs for humans. Okay? Because there was no sequence information available, no structure information available. But things have changed, Uday. I think everybody wants to do something that is very, very close to the patient. In this particular case, it's related to humans. So we would rather work on cell lines related to humans, or tissues related to humans, so that the translation becomes very, very fast. Okay? And I think industries have adopted that very well. Rarely you see pharma industries using, rat enzymes or rat cell lines. Everybody is moving towards humans. And in this particular case, I think if we adopt anything related to humans, I think translation becomes easy. Going to the clinic becomes a little bit more justified, and you'll have more confidence moving project forward, rather than having something in the back of your mind saying that I hope it translates. I hope it translates. Okay, so from that point of view, I think there's nothing like using cell lines, tissues, enzymes, and anything that is related to humans.
Yeah, I mean, you've been in management positions in large pharma. Would you be willing to make a go, no-go decision based on, let's say it was an animal-free program, and the president says, Ajit, you got to tell me whether we should spend the 100 million or not, purely based on alternatives. What would your answer be?
I would say definitely very much towards that, because there are examples within Pfizer and in the industry where people develop molecules, including Pfizer, and ran all the animal experiments and everything else, didn't see any data at all, like Jugnu said, and then realized that such a gene is not there in that particular animal, and then they said, oops, we lost a few years of work. Okay, so in this particular case, going back to your question, I think if anything related to alternate is there, I would rather go with that.
Yeah. Okay. So you strongly believe that these technologies have the predictive value that the animals may not have, even though the animal paradigm has produced billion-dollar products.
Right. Yeah. If it is anything related to a model very, very close to humans, that's the best one. And in this particular case, if we have all the gene information, we have cell lines available, we have tissues available, then nothing like using that particular tissue and answer the questions there, than working on animals and then trying to understand whether it will translate or not.
Okay.
I again want to just add, I think efficacy is where I see these models going best.
Yeah.
Toxicity, I think, will take some time for in vitro systems to reflect full animal tox paradigms. But another very positive point about in vitro system is the number of compounds or biosimilars you can test in vitro is very high. Whereas in vivo, you are restricted by cost of goods. You have to make 1 gram, you have to have good PK, you have to invest a lot to get a molecule which is testable in vivo. So you lose so much of your diversity by the time you get to the animal.
Yeah, I think generics and biosimilars are a different breed. I just want to focus on new chemical entities, innovation, and all of that, where the risk is very high, right? It's all about de-risking the program. So those are very, okay, thank you. Thanks a lot. Suresh, you organized a fantastic conference, and I heard a lot about humanized mice being the next big thing. Do you really think it's going to replace any of the animal studies we do? I mean, sitting and listening to that, I think the humanized mice, and I am neutral, we are at least 5 to 10 years away from the FDA saying, don't do any tox studies, don't do any PK, just show me the humanized data and you're in. where do you stand with the use of humanized mice?
Well, that's definitely a good question, Uday. So it's on the path of acceptance. The pace with which it is moving is something that is not acceptable, coming from the perspective of speaking for animals. But if you looked at the trajectory that humanized mice have taken, we heard the liver, the PBMCs, we heard the tumors. So step by step, it's getting conquered. So definitely 5 years for many things, but oncology is right in the top of the alley right now to be accepted by FDA pretty soon, simply based on the data that is coming up from mice from big labs like Jackson and Harvard's and all this. There's a lot of data getting accumulated as we speak. And also the consortium of pharma that is doing this for alternatives have been doing this for animals for a long time. So in my view, the alternatives is a recent child, and this one is almost a teenager. So by the time they reach level of acceptance, it will be some time. Having said that, the humanized mice data can be readily accepted by FDA if these models are representing rare diseases that don't have any other model, including alternative
That's
or
an
animal model.
appropriate citation rather than biosimilars and generics. Yeah,
Exactly.
that I completely agree with that.
Right.
Yes.
So that is number one. Number two would be the humanized mice also now getting so sophisticated, they don't show the leakiness or the challenges that we faced. Some of them will have NK cells later or B cells later, antibodies later. Those challenges are being addressed by many, many labs right now. So the humanized mice are probably going to be ahead of alternatives for acceptance of data, in my view. Just speaking for the panel today, but if I had to say I'm from OncoSeq Bio, my view would be different.
Okay, thank you. Dr. Akbar Shah, same question. You know, all said and done, the humanized mice is still an animal model. So where is the alternative there? I mean, all you have done is still use the mice, and you have expressed a set of genes, but at the end of the day, you're still relying on the mouse physiology and metabolism. So what's the alternate there? I didn't quite get it as to what is this brouhaha about we have come up with a huge alternate model. It's still an animal model.
Now, at the time when Suresh was talking about the humanized mouse, to his expression, the very concept I was developing within myself before you raised this question. Well, the very issue in the alternative and the animal model, the difference is basically in terms of to what extent do the animals mimic or take care of the biology of the human. So you take, for example, humanized mice is good. There are certain contexts, very rare diseases, where humanized mice could be the alternative. I would put that as the alternative when there is no other approach for you to do that. But again, the issue is that supposing you are going to develop a diabetes model in the humanized mouse, can you say that that diabetes is the same as the diabetes 1 or diabetes 2 in the human, who is 75 kg weight and whose life is so many years, and the mouse lives for only 2 years, and its weight is only 30-40 grams. So the diabetes in the mouse, which is developed due to some genetic modification, or the humanized mouse, can it be treated equal to the pathology in the human? If that was true, by this time we would have discovered a drug for diabetes. We have not done it so far. We have done it only the palliative medicines. We don't have drugs or therapeutic drugs for the diabetes. Well, humanized mouse is good for certain specific contexts, but not for all instances of pathology or disease management or drug discovery. To your question straight, what is then when you say that, what is the alternative model for that?
Right.
Right now, I don't have one.
Okay.
But the point is, if you look at the evolution of animal experimentation as a branch and alternative, I don't agree on the alternative, to be very frank.
Right.
This is now your NAM, novel approach methodologies, new approach methodologies. People are not happy with the term alternative. They want to remove alternative from the whole scenario and call it only new approach methodologies so that there is no conflict. So this is an evolution of scientific concept and practice. we had animal. Now, when human material was not available, we were comfortable with animal material. But now technology has greatly improved, science has greatly improved. We have now animal material, human material. When human material is available, then why do you hang around the animal material to go for the human material? Why it is available?
Right.
If you look at Russell and Burke's 3 R's, you will see that they mentioned about replacement, reduction, and refinement. Emphasis was on replacement when he said that today's technology for replacement, I can tell only about the tissue blocks, tissue slices, not even cells. But they said that in the course of time, the science will progress very rapidly. Newer technologies will come up, and those technologies will take over the animals, and the replacement will become complete at that stage. And therefore, today, well, no alternative. Where there is no alternative, go with animal experimentation. But be prepared, in 5 years, 10 years, 15 years, we will have total replacement for all the pathologies, including all kinds of toxicology, where alternatives, not alternatives, new approach methodologies will be available. Thank you.
Okay, yeah. You should enter politics because your answer, I'm still not sure which side you're on. So.
Alternatives, sir. My side is go for new, approach methodologies. Okay. Where you don't have one.
Yes.
You can't keep the disease keeping quiet for until you have an alternative.
Yeah.
I am not criticizing animal experimentation.
Yeah.
I am alive because I am maintained by at least 15 drugs every day.
Yeah.
Brought through animal experimentation.
Right.
I am thankful to them. But doesn't mean that technology should be taken to the side track. You will have to follow track with the development.
Okay.
Where new technology has come up, necessarily we will have to give place for that and leave the old technology.
That's my point. Just because it's new doesn't mean it's better. Right?
It should be proved.
New is new, but what has yielded results for you past for the 100 years, that suddenly cannot become bad. That's really my viewpoint. I'm not saying new or not new. I'm just saying as a management making $1 million calls, you're not supposed to be enticed by saying, wow, this is brand new versus this is old. You're supposed to make a more balanced decision. So that's kind of where I think the field will go. I don't think the field is ever going to drop animals or completely adopt. It's going to be a mix. For my lifetime, I don't think that's going to happen. And that's because the stakes are very high. This is not academic research, right? I mean, we're talking about spending millions of dollars. Would Manny, as he said, he had to make calls on moving products forward, would he make the product, push the product forward based on just one set? I don't think so. I mean, we'll hear from him, but that's my whole point. My whole point is new doesn't necessarily mean it's going to change the game. Right?
Well, I'm taking a lot of time. I've taken more time compared to others, but still, having raised a point, I would like to answer, to respond to that. Well, 100-year-old, agreed, that's what I'm telling. 100-year-old is really appreciable. What all animals have done for the human welfare, we owe a great lot to them. We don't undermine that.
Yeah.
And when I say new, it's not, I don't emphasize the word new, different from what was already there.
Okay.
These are different. New technologies.
Again, a very politically charged answer.
Improvement over that.
Yeah.
See, we had firewood as our fuel 50 years back. Today we have the LPG in all homes in Rome.
Yeah.
When LPG has come, necessarily we have to brush aside the firewood for the other reason that we will be engaging in deforestation.
Yeah.
Please, I'm not explaining this.
Right.
There
I understand.
Cross currents are there when I make this comparison of fuel. Thank you.
Yeah. Thank you. No, I mean, this is part of the debate, right? I mean, she was very emphatic and said, this is how it's going to be. That's how debate should be, right? You can't, completely understood. My next question is to Professor Redden. I don't know if any of you know, he has done some beautiful work on 12-lipoxygenase and association with psoriasis. I've been part of that when I was at drills. I used to follow. That's beautiful work. Sir, today if I said, I stopped doing that, here is a 3D model for psoriasis, make a decision based on that, which way would you go? Would you still say that I have, I love that 12 lipoxygenase knockout and I would pursue that? Or would you say, okay, this is something new here, let me do this? And what if the data were different in the 3D model?
So anything that we do on isolated cell lines or organoids or even in silico models have come now, that is not going to replace the complexities that are existing in the living system. So liver is not an isolated one present in the body. It is connected to various other, its function is coordinated by the controls that are there in all other organs. So you need that kind of systemic environment in order to understand the complexities that are associated with any particular tissue or organ. So in that, there is no such kind of replacement of the whole animal system with an isolated organ or the tissue. Ultimately, see, we all have gained knowledge on biology of health and disease by understanding, by studying the various information that was generated by using animal models. And that is the one which is now being extrapolated, the thing. Even today also we are not able to understand the complexities that are associated with any particular disease also. So when you are doing an isolated condition, that is not going to reflect the entire holistic system that you see in the animal model. So I strongly advocate for that continuing. They may supplement, but they are not going to be replaced in near future.
I think that's well said. Thank you so much. Please, Jugno.
So again, I would say for efficacy and for mechanism of action, these newer, more human, disease-relevant models will be useful. But for toxicity and understanding the overall impact on a full body, animals aren't going to go away anytime soon. So again, I'm making a distinction between efficacy and toxicity. Secondly, I have great faith in being able to develop predictive systems with the help of AI. So over a period of time, this interconnectedness, which is absolutely true, what is a full liver, what is a full pancreas, how are these things talking to each other? I do think such model systems are being built, will be refined, will be reiteratively made to a point where they can do more than what we can anticipate today. And coming back to animals, we all know, at least in the area of cancer, so many mice have been cured. The inbred mouse system using cell lines selects for highly proliferative tumors. You go in for typical chemotherapy, it works very well, but guess what? Human beings don't act like mice. They fail. So for efficacy and for testing, we need to have better models than typical CDX, mouse, xenograft, inbred mice.
Thank you. I mean, sorry. Ranjan, go ahead.
I thought I will agree on that. No, I think the advent of AI and ML, I think it really can. What we are looking at, the gap, bridging the gap. I have big hope on that as I'm listening and attending some of these meetings. That in it's not ready today, but in near future, most probably, we'll have this tool to really bridge that gap and then predict.
I agree. I think AI ML will take some of the bias out of, saying this alternative is new, so it's got to be better. I think that'll be much more rational right now. So completely agree with you. Manny, I called you the best PK/PD toxicology guy in India today, and somebody came and challenged me, why did you say that? But I believe you are the best. PK/PD is at the core of drug development. If there was one activity that dictates, it's not showing have nothing. PK/PD is the core that translates into clinical. All decisions are made. Do you ever think that alternate models can replicate the PK/PD that you'd really like to see?
I think in 1990, we were doing a lot of benzene carcinogenicity studies. Carcinogenicity means EPA has tons of studies in animals, rats, mice, different strains of rats, mice. They've done. They're trying to do that to put a dose exposed for humans in an 8-hour timed average per day. And they were not able to come up with a dose because of each animal is behaving differently. Metabolism different, PK is different, everything is different. All these strains, strain differences are very predominant. At that time, I was meeting a guy called Freddie Bois. He's a statistician, a mathematical modeler. He came to me, I want to see all your in vitro data. I have tons of in vitro data, microsomal data, myeloperoxidase data, and anything you want. You can even KM, Vmax values of the in vitro I generated. He put together all the data. He wrote a PBPK paper, physiologically based pharmacology. He computed the blood flow rates, protein binding rates, metabolism rates, and everything together. How many metabolites go to bone marrow, where they are going, and everything he put together. He generated a PBPK model that was accepted by EPA instantly because that gave them the best awareness on how to do the human risk assessment. We're doing for humans. This is not for animal. You're not developing a drug for animal. You're developing a drug for human. Yeah. So in which case, you need to understand the human first rather than understand the human. So for example, I'm not biased here, but what I'm saying is that we've had the battle for oil versus the solar for the last 50 years. Even today, people say, hey, solar is not cheaper, so I'll still stick to oil. Because it's not, they want solar, but they want to stick to it because oil is cheaper than solar. In same analogy, somebody who has invested crores and crores of rupees in animals studies, why would he suddenly give it up? He's a suicidal. Same thing happened when Bruce Ames challenged EPA. You're doing all these carcinogenicity studies on the environmental pollutants at very high dose, and they're showing cancer. Right. But all those compounds are not mutagenic in vitro. So there's no correlation. So there are natural pesticides present in plants, do carcinogenicity studies at the same doses, and EPA didn't take the challenge. The reason because those study designs were completely flawed, trying to show that these are all carcinogens and trying to do the risk assessment. He was right. He was the one who came up with the in vitro mutagenicity assay. Even today, that is the first one we do. We don't do carcinogenicity studies anymore unless we go to the phase 4 studies. And it is a really well taken. One reason they don't do it is because if it is a mutagenic compound, they don't want to take it because tomorrow if somebody by any chance get a cancer, he's going to blame the company for pushing that compound through the clinical development, and the guy got cancer, he's going to sue the company. Yeah. So as I've been in pharma industry, you have to be very careful in the public coming back and suing you, especially something happens in the clinic before, which didn't happen in the animals. All these now, one compound, actually, I forget, just 2 days ago, they're going to sue, I think Pfizer or Bristol Myers, somebody. It's because things are going out of hand. So you cannot predict, even Vioxx, they took 7 years to find heart valve defects in the humans.
Yes.
Right? So you really need mechanistic investigative models that are human models, not animal models, because you're not developing drugs for animals, you're developing drugs for humans. And the human risk is more important than animal risk.
So how can you humanize PK/PD? I mean, let's just say we needed to find an alternate.
Right.
And I heard you tell Viraj this morning that the current state of PK/PD, the multi-organ, and I agree with you, is not even close to what you would like to see. And what was especially scary to me is when he said, I put it on a rocker and I got different set of data. I mean, how do I interpret that? What if you put on a rocker at a higher speed? I mean,
so
aren't we moving away from humans is kind of what I wanted to ask him.
Well, I'll give you one example, one target. I'll give you, they had luckily mouse, rat, guinea pig, and few other, the target available. They did the binding acids. The drug binds least to the human target compared to the mouse target. And would you take it to the clinic? I've seen the data. I've seen the data. So why are we don't even know the target. PKPD, you need to know in the human for the same target. For something that binds very hard, the PKPD is going to be very good in mouse. The PKPD is not going to be very good in human. So you need to worry about human PKPD, not a mouse PKPD.
Yeah. So where do you stand?
I think that it's not easy to switch that side to this side because it's somebody who is invested into the animals. Charles River is not going to give it up today.
Mm-hmm.
Okay? Because they have invested so much money, they invested in monkeys, dogs, rats, and everything. They're not going to give up today because they're multi-billion-dollar business.
Right.
So they're waiting for the time, and until then, they're also trying to develop these models in-house.
Yeah. Yeah. Ranjan, you know, one of the things that scares me about the new miniature micro technologies that are coming up is, you know, you're dealing with 10 microliters, 5 microliters. And that's scary because if I'm off by 0.5 microliters, I might get a different set of data. Does that worry you, or you think, because in animals, there's a large capacity to absorb some of these errors that are bound to happen, even if you robotize whatever it is that's happened. So are you concerned about too much of miniaturization and that's why you're moving away from animals? Or where do you stand on the miniaturization platforms that are coming up?
Well, see, it has, I think as far as the, convertibility is concerned, that's an worry. Yeah. Because you are doing it at such a miniature scale, and a big human body when it is going, how much of it will translate, that's an issue. Only hope is the automation, the way it is going today. Mm-hmm. So I think in very near future, we'll see that precision will come. Okay. And most probably that will give us the confidence that it can be scale up to the animal and human stage. Today, it's a concern.
Yeah, okay. Thank you. I think that was very clear answer and yeah, in agreement with what I think. So, you know, Jugnu, the other question I have is GLP tox, which besides PKPD, the other thing that we rely, again, I want to talk to you as management who has to make million-dollar decisions, right? I'm not an academic looking for a Nature publication. That's really not, these technologies to be used by industry will be for go, no-go making decision, right? So I'm asking you, do you ever see the full repertoire of GLP tox studies being replaced by alternate technologies? Because we make decisions, one at efficacy, then GLP tox, in between PKPD, and then phase 1. You know, the clearly defined points, value inflection points where we make decision. And GLP tox is a big one. Do you ever think that we'll be able to replicate the complexities of GLP tox in a rodent and dog that you see?
In my lifetime, no.
Okay.
But I do have hope that, again, we're talking of reducing the dependence on animal systems. Mm-hmm. Instead of doing N of 20, instead of doing 3 doses, instead of reaching the maximum tolerated dose, are there better systems to predict that will come into being? Also, the kinds of mechanistic work you can do in vitro, where you can add additional cell types, you can do drug combinations, you can do sequencing of different drugs. Those kind of things are very difficult to do in animals. Mm-hmm. So, as I said, I can sit on that side. Right. If you will ask me, 100% are you walking away from animals today? I will sit on that side. Okay. But I think the question you are asking, which is why I am on this side,
Yeah.
are the alternative models advancing? Do they have the potential to get to a point where use of animals will become less and less in a very judicious manner?
Okay.
No. I mean, I don't have $1 million to make that decision today, but if I was back at Vertex, I
Yeah.
was leading the projects that I have led there,
Right.
I would not advocate immediately
Yeah.
walking away. And as I gave the example of cystic fibrosis also, all the efficacy studies were done in patient cells.
Mhm.
Toxicity was still done in 2 species.
Yeah. So you're really saying we are not quite there yet. There might be some adjunct assays available to help you, but yeah. Okay,
Yes.
I got it. I think that's a
To
pretty.
take the right molecules into animals,
Yeah.
less animal models for efficacy,
Yeah.
more animal models for toxicity. And for a minute, if I was to step back, all patients, all animals, the example of oncology is with us. If you do a test for drugs that target EGFR in a broad population, your result will come out negative. The drug will get rejected. So what are we doing? Using sequencing, using understanding the signaling cascade. We are already stratifying patients using human knowledge.
Right.
And that's when these drugs that target even 4% patients like Ross, Rett, these are getting selected,
Yeah.
getting approved. So human biology is something we need to understand. For cancer stem cells, if you're going to target metastasis, how are you going to generate human stem cells in animals? You have to do the human systems.
Yep.
You have to understand metastasis from the human being.
Fair enough. Yeah. I think it's a very rational answer. Chugnu.
Look
Thank
at
you.
the way FDA defines GLP tox, right? What they say is that the drug must be safe in 2 animal species. One small animal species,
One large.
another large animal species. They say that what is your estimated PK dose from the pharmacology studies from the animals. So when you put this together and you say, hey, in rats, my compound is toxic at 10 milligram per kilogram dose, in dogs it's 100 milligram per kilogram dose. So now you have a differentiation here. The large species is good, small species is not good. How are you going to make a decision? Then my manager says that, let's do monkeys, right? Each monkey is $15,000. So forget about the cost. What I'm trying to say is that when you have these discrepancies, what FDA comes to say that you have to come up with a first-in-human dose, not necessarily the efficacious dose, should be the conservative dose that you can actually assess the PK that you
Yeah.
predicted
You mentioned
from
that
animals
this
to
morning.
human.
Yes.
That is the only purpose of GLP tox,
Yes.
nothing else. You can do that even using the human PK/PD in vitro models.
Yeah.
You don't need animal GLP tox.
Yeah, I think where we are going with this is again that, there are certain complex decision-making points such as PK/PD, GLP tox, and you cannot solely rely on present day, but they can be used for, as she said, mechanistic, teasing out the role of each cell and all of that. But the ultimate decision will have to be an aggregate largely of the animal-based data, right? Is that accurate?
Currently, that is how it is pushed, but FDA is also saying we don't agree with animals' data at all.
Right.
So that is why they're also pushing industry to move
Alternate,
from to the
yes, I
modern
understand. Yeah. Great, thank you very much. Suresh, I have a question for you. I know you're developing a product for human aging, understanding their biological age versus chronological age. And you've been asked by the regulator to do a set of patient samples. Did you ever think of an alternative and said, I don't want to do that's going to be very expensive, it's going to cost me money, can I just do this? Did you ever think of an alternate to such? Because I think diagnostics is a low-hanging fruit where you could potentially introduce alternate studies.
Absolutely, Uday. I think that there is no need for animal models, especially when we talk
Yes.
about these kinds of models. Having said that, what what I have been banking on is the retrospective data that is probably available for so many people who have died at different ages.
Right.
And there all the demographics are available. And we can go back, dig in, and also do artificial intelligence, computer-aided
as he
surfing
said, yeah.
and identifying these things. So that is a very big possibility. We have actually proposed to the regulatory that we don't have to do the biomarker estimates on the path forward for people, but we can go back and look at the retro data.
Yeah.
And they said that we should do that. We should actually do that. But it comes down to some of the biomarkers which were never tested in those people and what they have taken that confounded their data. So some of the data is not actually available retrospectively for aging part. But if you go into oncology, I think there are biobanks, there are databases now available with not only the patient demographics and data, but also biomarker, gene analysis, epigenetics. A lot of data is falling through into the system where it's a funnel where it gets narrowed down, and then your level of confidence goes up when you say, okay, this is something that we have done. So one example for that is for Alzheimer's, nothing to do with aging, but aged people do get diagnosed with Alzheimer's. There is an infectious reason. The cause of Alzheimer's is an infectious disease, is one of the hypotheses right now.
Prions,
right?
I think.
Porphyrominus gingivalis is the bacteria that's known to cause black tooth decay in older people. But the path to brain from the nasal cavity is very short. So these bacteria secrete gingipains, and they go and create a pathogenic reaction where there is accumulation of glial multiplication and so on that can contribute to Alzheimer's.
Right.
But this, nobody is going to do it in an animal study because the animal brain and bacteria, everything is different.
Yeah.
But there are so many animal models available for Alzheimer's, having said that.
On the flip side, there's probably $5 billion that have been spent on
on so many things, right?
Yes.
So I think the retro data that is available for almost all diseases actually calls for not using either alternatives or animals. So we are not digging into the retro data that is available. So that's something that the regulators should think about. The NAMs, which is, I would like to call as non-animal methods, need not be alternatives. It could be in silico, it could be many things, along with the animal data that is available, and most importantly, the retro data that we can dig by the things that are available right now in the fast-growing computer science field, machine learning, tools, and artificial intelligence. If we can combine all these three, we can create algorithms. In fact, our aging assay is based on algorithms based on some of the biomarkers, not just one biomarker. So I think I am a big fan of retro data now, along with animal data, and if available, non-animal methods.
Yeah. And you think, based on your interaction with Cedesco, they would appreciate this line of thinking?
Yeah, so this is a low-hanging fruit. The point with Cedesco is diagnostics is a low-hanging fruit because unless there's a disclaimer that says, okay, consult with a physician before you draw some conclusions and take some medicines. If that goes with a disclaimer, Cedesco has no problem with diagnostics because a physician is not prescribing anything without looking at. So these are predictive, which is even lower than the actual diagnostic. So predictive biomarkers actually contribute to, for you as an individual to make a decision on understanding. So you understand your age is 60 or 62, but you're not going to do anything about it. But if I look at some biomarkers and tell if it is not 62, it is 68, then you're going to react and do something about it. That's predictive value that we bring to the table.
Are such products approved? My very quick question, more as a learning. Are such products approved anywhere in the world where you can look at your biological age versus chronological age, even though a physician doesn't have to?
I'm happy to say that we are probably one of the first pioneers in India to do this. But in US, there are at least 5 to 10 different companies that are looking at epigenetic markers for not only measuring your age, time clock or the aging clock, which also tells how fast you are aging, not just what is your current age. It's going to tell as a subscription, this year, next year, the year after if they measure, then they'll tell you are aging 10 times faster than you are supposed to. So there are availability in US, but our market is very fresh.
Astrology. Dr. Akbar Shah, my question to you is, what do you think will it take a regulator to accept this alternate? Right now,
it's a case by case. And I think in most cases, they still prefer the old paradigm of animal GLP tox, the typical toxicokinetics. And what do you think it's going to take them to say, yeah, I don't need to do 2-species tox anymore, just go ahead and enter clinical trial? You think that's ever going to happen? And if so, what tools do we need for that to happen?
Well, that's a very good question. Well, even the regulator is actually looking for a change where the cost, time, effort in tackling issues in toxicology and biomedical science will be minimized or lessened. They are looking for that. So they are very keenly watching the development in the context of in vitro approaches, starting from 2D culture to microphysiological systems, or even more than that. Well, they are also keenly watching that. And that is the basis on which the USA agreed to go for the modernization. And the government of India has also announced that in addition to animal experimentation data, you could provide data from cell culture or in vitro approaches. So what I'm trying to emphasize straight to answer your question is that in as much as somebody puts an effort to convince the regulator to change from animal method to non-animal method, spontaneously, automatically, there is a change of thought in the regulators to change from the classical approaches. I don't say condemn, I can't condemn that. The classical approaches to the modern approaches, less expensive, more, it's a faster, less labor consuming. That's good for the country by and large. Less investment on that, quicker result, and diseases are taken care of, chemicals are managed appropriately.
Yeah.
So they are looking for, and therefore, little bit of, we invite them to conferences. You invite them to conference. I invite them at least 4 or 5 of them to every one of my conference.
Yeah.
We try to educate them that techniques are available. So once they get convinced of that, it is taken care of, and you get this transformation.
Okay.
Yeah.
Okay, thank you. Ajit, I kind of, asked him before I asked you, but the black box still is when the molecule hands off from the preclinical team to the clinical team, right? That's the big black box. We basically, even today, we pray that it works out all fine, as you have probably prayed in Pfizer secretly, saying, my promotion depends on this, so please make it happen. You think the alternate will ever change that type of thinking of, oh God, make it work, or?
No, it's not a question.
No, I was going to ask Ajit first, and then, so yeah.
I think it's a challenging question. I think, the industry has to adopt, and we see the change. Okay?
Yeah.
So I think they will be more willing as long as you have a justification saying that, these are the reasons why we think, we are more confident with this particular system. But, it's always a risk, right? And analyzing the risk and taking that risk. I'm sure they'll say, okay, let's just go ahead with this, but can you run few animal experiments to make sure that, it falls within that range? Okay, I think it's just a matter of time, Uday, before they all say. But I want to agree with what Professor Redana was saying. See, we are all working in silos. We're talking about models. People are developing, human on a chip, right? With various organs and all, microscopic sizes and all. But animals give a holistic picture.
Yes.
Right? So from that point of view, I may be sitting there for this particular answer.
No, you're just being fair. Yeah.
Yeah. Right. Yeah, but things are evolving, absolutely. I think we have to, keep up with the time and move into systems that are very, very close to, in this particular case, humans.
Yes.
Right? And then even in humans, like Jignu said, there are hundreds of mutations. We have access to all those mutations. You test all of them. So then, that is the place where you need to play rather than saying, okay, let me just go with the animal model. So holistic, I agree. I think, there's a lot more that animals have to offer. But
moving forward.
Correct. Thank you.
Thank you so much. One question for Professor Redana is, a lot of this save the animals, reduce, a lot of it is also sentimental. At least in the US, there is such strong lobbying, And, the government has to, bow to that. So my question to you is, how would you convince these people saying, I know we are sacrificing animals, but we may be saving your children one day? I mean, how do you argue with people who are completely blind to the fact, the value that animal studies have brought in developing human medicines?
I think they have to see the history, how everybody is enjoying today the medicines that were developed by using animal models. And even then also we are not able to answer all the problems that we see, specifically with the aging kind of things. So unless we come up with a kind of, so using animal models, we could always, the cell lines or organoids or other in silico models could be supplement in reducing, refining those kind of things.
Yeah.
But ultimately, you need to evaluate. So even the drugs that were developed after testing in humans,
yes,
they are not showing the same efficacy across all races and other populations. So there is still diversity.
Yes.
So one drug for all concept has gone.
Yeah.
Now we are moving to personalized medicine.
Yep.
So in such a scenario, so we need to rely on the animal models until there is really foolproof alternatives are available. So that's the scenario.
I think one of the ways I think is to stop you. Yes, sorry, go ahead.
To your point, I think we need to educate that also to common man, because we are working, scientists are working in isolation, but not conveying the message to the public.
True, totally true.
And
I agree.
that is one of the reasons, misconceptions, that needs to be removed.
Yeah, 100%. I mean, one of the things I see, I'm a pet owner now, and a lot of these people are worried when they say we use beagle dogs in our research, right? So, like you said, I think it has to be conveyed to them, that it's not just beagle, not because it's your favorite pet we are sacrificing, it's just that that breed happens to be, so that message of why the value that brings has not gone out. All this, so that I think I used to, in Atlanta, we were running a big lab. Kasturi, you just made a big question, which is, there is so much sentimental pressure on not using animals. How do you convince those people? And I think Professor Redana gave a great answer for that. So, I think we're not communicating well. So to the point that you guys are, working towards communicating to the lay people as to the value of, using animals has to be part of your agenda. So.
In our country, it is still to a large extent acceptable. But if you go to US and then Europe I think is the most aggressive activist there. And from Dr. Reddy's, when you have to go with our collaborator lab, they were like Bhulbhulaiya. Where they put the animal house. If they don't take you, you can never go to animal house, even you enter there. So I think that's where most probably what Professor Redana is telling, it's important that you have to teach them there. But again, the vaccine hesitance we have seen during COVID. So that's I think a separate debate.
Sorry if there's any animal activist here, I don't want to offend them here, but I'm just putting my point here, that all animal activists I've seen fight for dogs and monkeys. They don't fight for rodents.
Nobody fights for chickens. That's why we are working on chickens
right?
now.
I mean, so the question is that what is life? Is rodent is not alive? Only dog and monkey are alive? So the question is that where do you put the brand? I mean, I kill mosquitoes every day. Right, I mean, as soon as it comes from... So,
why are you killing a mosquito?
I have to kill because it's going to suck my blood, right?
Right.
So the question to this is that the ethics versus the science. Here, I'm only a scientist. I'm not talking about ethics of animal research. In a science, how do I get a best drug into the man the best possible way?
Yes.
Right? And I have worked on animals for 30 years in pharma industries, and I've done quite a lot of studies. Toxicology, PKPD, everything I've done. But I'm not convinced a 5% approval rate for oncology drugs from 100 drugs in the clinic, you're talking about how many billions of dollars? I mean, you're wasting money. So can you bring it down? I mean, the cost is also very important. If I go and ask somebody to give me 10 crores to do my alternatives to animals, people look at that side.
Yeah.
Why? Because the perceptions.
Yeah.
We have perceptions, wrong perceptions for wrong reasons.
No, the
We have to
thing,
change
Manny, is, and I agree with you what you're saying, the lobby in US for, just like the gun lobby, right? Everybody knows the killing can only happen from a gun. You don't need to own an automatic gun to, hunt whatever you want. Yet, it's the same type of lobby on animals ethics also. People have no clue what they're talking about, thinking, but there's such a strong lobby, and ultimately the FDA has to listen to that because, elections and won are lost on such type of things, as you know. So I don't know what's going to solve. I think we will stop now because I know Ajit has to catch a flight. We will open the floor up for questions for another 15 minutes or so. But can you just show of hands, as I said, which side do you think has won? Is it for animals or the alternate? Just show of hands, please. For animals, how many people are here? 1, 2, 3, 4, 5, 6, 7, 8. Great. 9. And alternatives, 10. Alternatives, how many people?
Oh, come on.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Oh, I think so. It's a tie then. Basically, you're both tie. Yeah, that's I think fair enough. Yeah, I think somebody from this side has bribed a few people. So that's.
I can phrase this question a little bit differently and see the show of hands. How many people are willing to take a drug if they have, let's say, chickenpox or something? The drug has not been tested on an animal, but only an alternative. How many people are willing to take that? And there's another drug that has only been tested on alternative, never tested on animal. So it comes down to the confidence level of which one do you accept, and that's where the data is important.
That is today.
Today. We're talking about today. So today we are.
Not in my lifetime, probably.
Yeah.
But I know we are evolving towards that. If I can give an example, predictive example. So I agree with Suresh about retrospective data and samples. In diagnostics particularly, people are using that very effectively. So at the time of surgery itself, again, example is oncology, there are more than 4 diagnostic tests I'm aware of, which we have helped develop with that company, where at the time of diagnosis of the surgery, diagnosis and resection, you can have a marker that can predict whether that person is going to respond or not to a therapy. And so the saving of money, saving of cost, saving of side effects is enormous to not give somebody a drug that they will not respond to. So what we are seeing in diagnostics may or will come to drugs also.
Yeah, thank you. For a minute, Suresh, I thought you were asking the audience, how did the panelists do? I mean, the moderator do, so I'm glad you didn't ask that. I think we'll open it up for a few questions, please. All those who raised hands have to ask a question, so. Yeah, go ahead, please. I see.
Come here.
Yeah, please. You have a mic or just come here? Yeah. There's no mic, so just, you're not mic today, you'll be somebody else. So. Yeah, we can hear you.
I'm just wondering about the narrative of this debate.
Okay.
Where we are talking about benefit of animals versus an alternative system.
Correct.
What if the narrative could be changed that your alternate system is as sloppy as your in vivo system, and if the alternative system can prove that it is as sloppy as in vivo, is it the right data to stop using animals?
I think there's no money to do sloppy work, to be honest. Nobody's going to fund you to do.
But that is happening anyway, right? We are saying
It
that
is.
decades of animal study has
Yeah.
not given us better drug candidates. Now we are saying that these alternative methods will come up with a magic wand that
Right.
will probably give us better throughput in terms of drug discovery.
Right.
So we are anyway, we have made that distinction already,
Uh-huh.
right? Right now, the alternative guys has to prove is that their system is at par to the in vivo system in animals rather than better system of human.
Yeah.
Right? So why
Yeah.
don't we put that narrative in place? Because I, being a scientist, I have always seen that we do null hypothesis. But here we are not using null hypothesis. We are saying something is better unless otherwise proven. What if we go from saying that nothing is better unless otherwise proven otherwise?
Yeah. But to change something that's already been used for last 100 years, you have to show that something is better. Otherwise, nobody is going to mandate change, right? That's the whole thing is you have to say we are better than what's been used for 100 years because of the following reasons. That was very well articulated by both sides today, I think. The challenge really, as you very rightly said, is to one side to show that they are better than what's been used for the last 100 years. So
Right.
that's
and
what
that
it
might be
is.
a case for phasing one thing out, right?
The narrative, how sloppy can you be? I don't want to be part of that debate. Thank you so much. Yeah. Any other questions? Sri, I see you have a burning desire to ask a question, is it? Or? Yes.
I am neither from this side nor from this side. I like both of you guys.
Okay.
But more than that, allow me. The only thing I can talk about.
She immediately reacted.
We also like them. I should interpret
It's not the burning question with respect to who is good, who is bad.
Right.
Or who is improving or not.
Yeah.
I think that's a better way of saying.
Yeah.
I think I have one, your question to Ajit. Where,
Yes.
if you have $1 million and what would be a go, no-go decision?
Yes.
I know, I would say spend all the money.
Yeah.
And who will decide to go and who decides to stay will be decided. That is first thing. And as what Professor Redana, your question to Redana was actually, can animals be spared? Of course, there is a presidential pardon every year.
Presidential pardon.
One turkey gets saved every year.
Okay.
At least that's an animal to be saved.
Yeah.
Thank you.
Yeah. Thank you. Yeah. Any other questions, please?
Can I just...
We had so many questions in the morning. What happened now? People are tired and shy. And oh, there's Srinivas. Yes.
I'll ask.
Next question will be by Lily. I can see she wants to ask a question.
So, I mean, some points I don't disagree with. Efficacy only, we cannot only extrapolate, we can extrapolate only to efficacy. Toxicology also very difficult ball game. So when you go to GLP toxicology, as you mentioned, there we are doing 7 days, 14 days, 3 months, and 6 months. See, if FDA want to revisit that, they can reduce at least 2 studies. One is, suppose what is the difference between 7 days and 14 days? I think we need to understand dose linearity. So I think only one study we can keep, and instead of 6 months or 3 months, we can keep only one. But at the end of the day, the information what we are acquiring from the animals is lot as compared to in vitro. I would say in vitro is only pertaining to proof of concept studies to understand what is the mechanism and the toxicity to a certain extent.
Yes.
But when you are correlation with the human data, at some point of time you need to understand the pharmacokinetic and pharmacodynamic relationship to understand the first in human studies, dose. So I mean, again, there is a lot of confrontation, definitely. So end of the day, how we need to reduce the utility of animals is very important, rather than more relying on in vitro, rather than more relying on animal studies. So especially biomarkers are playing a major role when we are moving towards first in human studies after toxicology studies.
Yeah.
So whether we can reduce at least 2, 4, instead of 4 GLP toxicology studies, we can come down to 2 and get more information as much as possible, and then reduce the animal size.
Yeah.
That's what I'd like to tell.
Okay, let me answer you one question here. There was one compound I worked with, which actually very interestingly gone into clinic one way. The problem was that when they did rat and dog toxicology studies, in 1 month toxicology. It's not 14-day study, 28 days. It's a 28-day toxicology studies. In rat and dog, in dogs, they're all going blind. They couldn't see, they were hitting the pens, this side, that side, everywhere. So the question came in, nothing happened in the rats. I know the biochemistry of the mechanism of action and also the toxicity. And I did suggest something, but anyway, it was ignored because they thought that I'm wrong. But in any case, what happened was they pushed it to monkey. And then pushed it to monkey, and 28-day toxicology was clean, very clean. And the senior VP comes, see, Manny, you're wrong, right? And I said, yeah, I'm wrong, that's okay. And I walked away. So they started doing, because they already went into the clinic. And they started at-risk 3-month toxicology studies, because when they have to do the efficacy studies for a longer period, they have to have a 3-month data of the toxicology as well. Now, in monkey 3-month toxicology studies, monkeys were going blind after 1.5 months. It's just the time. It was not toxic in 1 month study, it was toxic in, they were all going blind, monkeys. Okay. Now, who do you blame for this? It's already in the clinic.
Yeah.
People are already exposed to it, right?
Yeah.
So these decisions are very, very difficult to make, because one compound in one animal may cause toxicity in 30 days. It may cause in 3 months in another animal.
I think that's a good point.
So,
right. You don't know where to draw the line, basically.
I'll add
you can respond, and then we'll move on to
I'll
other
add
questions.
that point. So, Mani, I think you're raising a good point, but that is where we need to integrate AML model, and along with that in vitro models for 3 months toxicity, whatever the observations we are doing. So if you conclude the mechanism, what is the responsible for the toxicity, and then I think 6 months toxicity would not be required. I mean, end of the day. So where the biomarkers will play into the picture. And especially most of the times toxicity driven by metabolites, which are generating from the compound.
Yeah.
So if you address that one, I think by 3 months model, we don't require to go for 6 months model. Is very, very harsh model. I mean.
Anyway, brilliant discussion. Manu, you have a burning question to ask, or?
No, actually not a burning question, but I do have like a comment on it. So basically, I'm again, because I've worked extensively on animals, and I know, as Suresh sir said, who all are willing to take the drug which has just been tested on the in vitro or just the lab on chip model.
Right.
But then again, if we are able to show a correlation between the lab on chip model, let's say, like I was discussing with Viraj as well, that for example, he has taken the control drugs, he's tested warfarin on his lab on chip, liver on chip model as a positive control, another drug as a negative control, and we can see the unknown drugs, where are they falling in the correlation part, whether they are towards the better performing or lesser performing, and if we can tally that data with the human performance.
Right.
And if there is a correlation, we can show it, then we can possibly reduce, as I said, reduce the number of animals that have to be tested in a bigger study. So that is already refining of the study. And
very political, nuanced answer. I like that. Yeah, that's good. Yeah. Lily, you have a question, or?
No.
No, okay.
No, but I have a comment which is on the same line.
Sure.
That, there has to be sides, but at the time, the importance is to actually, like we said before, to create bridges. Because both systems are complementary, sorry.
I think that's a good word to use, complementary,
Yeah, and like examples like this, you can actually use some system to eliminate, to fail faster, or success, improve your drug, or make the sorting of which compounds or which drugs or whatever are going to go to the next step using less animals. The other point is selecting the animals that's adapted to your study and to the test that you're doing and to the disease that you're analyzing. Even at the preclinical stage, like we said, there's so many different, and there are some tests that are done with the animals that are available here because they're available here, but these animals are not necessarily the ones that are adapted to the test, to the drug, or to the disease so that's a completely waste. And whatever you're going to have results here are useless. And you're going to realize this 2 phases down the line.
Totally.
Right? So it's really, looking back now, like Suresh was saying, looking back at the data for everything, and saying, which animals is more adapted to the test that I'm going to do, to the disease that I'm going to analyze. And this is where also the alternatives or the non-animal models can help. Mm-hmm. Right? So I think depending on what we're doing, it's a synergy, and together we'll use less animals and succeed faster.
Yep. Yeah. No, completely agree. Thank you so much. One last question, and otherwise I will thank all the panelists for, I think, very fair and brilliant discussion, actually. So anybody have a question? Otherwise, a round of applause for all of them, please. Yeah. Yeah. Thank you, panelists. Yeah. Hope to have this discussion again next year, my friend Suresh. Yeah. Thank you.
Thank you, Dr. Uday Saxena, for being an amazing moderator and the panel discussion. Taking photo.
Don't mix it up.
Yeah. Thank you, Dr. Uday Saxena, for being an amazing moderator of the panel discussion and leading the panel discussion with a concise, unbiased approach. I would like to thank the panelists, Dr. Suresh Pasala, Dr. Reddanna Pallu, Dr. Jugnu Jain, Dr. Ajit Kamath, Dr. Subramaniam Mangala, Dr. Ranjan Chakravarti, and Dr. Akbar Shah. This is a topic we can continue the discussion for a long time. We will meet again next year and have similar discussions based on the new technology that will come. Now, I would like to invite Dr. Manu to give a token of appreciation to Dr. Subramaniam. Careful. Next. Okay. And I would like to invite Dr. Lily to give a token of appreciation to Dr. Akbar Shah. Someone taking photos? Okay. Yeah. A token of appreciation to Dr. Ajit Kamath. A token of appreciation to Dr. Jugnu Jain. A token of appreciation to Dr. Ranjan Chakravarti.
Thank you for not changing sides.
A token of appreciation to Dr. Reddanna Pallu. A token of appreciation to Dr. Uday Saxena for being an amazing moderator. Thank you all.
Token of appreciation.
Thank you. Really enjoyed meeting you and hearing your views. Yes, we have.
No.
Yes.
Award ceremony. Thank you. Thank you. No, I've been a part of Augusta for last year.
Right. So, I mean, are you leaving the kids? You're going to stay?
Yeah. Now I would like to invite Miss Ramya for the award ceremony.
Hello, everyone. What an uplifting and productive day it has been. Ladies and gentlemen, distinguished guests and esteemed colleagues, welcome to the award ceremony for the 4th International Conference on 3R's Advances in Research and Progress. We would like to express our sincere appreciation to our sponsors and partners who have generously supported this conference. Without their valuable contributions, this event would not have been possible. I invite representatives from our sponsors to come forward and accept a certificate of appreciation and a souvenir as a token of our gratitude. I would like to invite the platinum sponsor, the Jackson Laboratories. Nobody? Our platinum sponsor, the Jackson Laboratories. Next, our gold sponsors, January Labs and Sai Life Sciences. Thank you to our gold sponsors, January Labs and Sai Life Sciences. Sai Life Sciences. Our silver sponsors, Acuprec, Vishnu Traders, and High Media. Mr. Sanjay from Acuprec. Next, I invite representatives from our knowledge partners to come forward and accept a certificate of appreciation and a souvenir as a token of gratitude for being with us throughout organizing this event. The Jackson Laboratories and OncoSeek Bio Private Limited. OncoSeek Bio, ma'am. OncoSeek Bio Private Limited and Jackson Laboratories.
No, it's working. Medal, where to find the medal? So the evaluation of the posters were done. We had two esteemed judges on the panel. We can't reveal their names because of confidentiality. However, we'd like to announce that the medal was won by a very good scientist. I have known her. Happy for her. Human-like severe hypertriglyceridemia in an avian model, Serenia.
Wish you all the very best. Could I put it on her? Yes. Good luck. All the best. Thank you so much. Yeah. Thank you. Good luck. All the best.
Very good evening to one and all. I am Nikita Naik. You heard from me the whole day. Still, Organizing Secretary for the 4th International Conference on 3 R's Advances in Research and Progress, Patient-Derived Tumoroids and Humanized Mice. It is my honor and privilege to propose a vote of thanks to all those who made this event possible. First, I would like to thank the organizers, OncoSeek Bio and founder Dr. Suresh Posala, for conceptualizing this wonderful international conference and organizing it successfully for the 4th time in sequence. I thank our co-organizers, Mahindra University, the Federation of Asian Biotech Association, FABA, and the Jackson Laboratory for being with us throughout organizing this event. I would like to thank the respected Vice Chancellor of Mahindra University, Professor Yaj Madhuri, for immediately accepting our invitation and presiding over the inauguration function as the chief guest. I thank the special guest, Professor Radhana, Executive President of FABA, for participating in this event. I would like to thank the guest of honor, Professor Rajendra Singh Chauhan, Dean, School of Life Sciences, Mahindra University, for taking the time to be part of the event. Coming to the very essence of this conference, the eminent speakers who have taken all the pains to travel to Hyderabad from various places in the country and abroad, graced us with their invaluable findings and discussion. A very hearty vote of thanks to you all. Respected faculty members and students of Mahindra Universities, representatives from the industries, presenters and attendees, and hundreds of participants from various countries who are watching us live. A very big thank you to all for being our great audience. I want to express my gratitude to the supporting and sponsoring organizations, the Jackson Laboratory, Xavier Labs, Sai Life Sciences, Acuprec, Vishnu Traders, and High Media. This conference would be impossible without your generous support and sponsorship. I also want to take this opportunity to extend a hearty thanks to the Mahindra University staff for the excellent logistic support and cooperation extended to all of us at this conference, including your generosity in using this auditorium to organize this conference. An event like this cannot happen overnight. It requires rigorous planning and a bird's eye for minute details. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues from Oncocyc Bio and Mahindra University. I'm thankful to Dr. Manusmriti Singh and Dr. Bipin Singh for their guidance and support. I thank the PhD students, Ms. Priya and Ms. Pragya, for their invaluable support and contribution. Last but not the least, I want to express my gratitude to Ms. Ramya, the organizing secretary, for being an excellent partner as organizing secretaries in organizing this conference. I'm thankful to Mr. Suresh and Ms. Reshma for their vital contribution in manifesting this conference into a successful reality. Finally, my apologies to all those whose names I am unable to take here due to time constraints. Also, kindly pardon us with a big heart if there is any inconvenience caused or if there are any flaws in our arrangements, like AC. Please follow our LinkedIn page and revisit the conference through the live telecast recording. Once again, thank you everyone for taking the time to be here today. Okay. To be here today and listening to me speak, I'm so grateful for this opportunity. Before I conclude, I would like to invite Dr. Uday Saxena to present a token of appreciation to the representative of Oncocyc Bio. And I also invite Dr. Manusmriti Singh to present the award.
Yeah, before we give this award, I just want all of you to give a very big round of applause for Suresh. You know, I don't know how he does this. Every year the conference keeps getting better and better, and the number of sponsors keep increasing. So he's just amazing. Suresh, you are in line to receive the IFAR Lifetime Award soon. So thank you so much.
Thank you. Let us continue to uphold the principles of replacement, reduction, and refinement in our research endeavors. And may we always strive to create a world where scientific progress and compassion go hand in hand. Thank you and good night.